All right. I hope people can hear me. Just want to make sure that things are working technically. Sound is okay, and then picture is okay. Wonderful. Great. Then well, it's my pleasure really to welcome you to Nanion's user meeting 2022. Well, it has been a little while to do this. It's the 13th time we're doing this, so it's great to see you here. We again, like last year, are doing this in a hybrid mode. That is, we have a lot of folks here at our company in Munich, which is nice. It's a exciting day, vibrant till the morning. Lots of visitors, and we also have a lot of folks just tuning in remotely. I'd also like to welcome all of you, and I hope we make it an experience that is equally fine for both those that are here and those that are doing this remotely. 2022 actually is a special year for Nanion. We are celebrating our 20th anniversary. So we actually had a big party for the 20-year anniversary this summer. That was great fun, great celebration of the good old times, and appreciating the great development that the company is taking, and the great team that we brought together. That was really nice. I know some of you have been there. That was a big party, so very special for us. 20 years, that of course is a long time. Some things are remaining quite stable within our company. Among those are the people. This is just a quick flashback to 2003, one of the early biophysics we did, and the most recent one. And well, it really feels like time is flying away. But you will notice that the Nanion management team appears completely unchanged. There's no notion of us growing any older, just maturing nicely. I put this somewhere else, so you can finally see that. Right, yes, so 20 years has been quite the ride. Just a quick look at the sort of major milestones of our company. Well, we brought along a number of automated patch instruments, scaling from the small powder patch that we still love to, well, all the way up to the 384, the SyncroPatch. Well, over the time, there have been many great steps the company has been taking, not only product-wise, but also, growing a team, growing a real, I would say, great and lasting company. So that is certainly something we are proud of and very grateful for. This is sort of a quick overview of the different instrumentation that we offer. In the upper part is all the automated patch systems, small benchtop devices to full-blown automated high throughput systems. But we not only offer automated patch, which of course remains the core of our technology and the core of our business. But over the years, we've been introducing a number of new devices that are related, but really making use of our expertise in the protein analysis field. On the lower left, you see our suite for bilayer recordings for ion channel reconstitution, the Orbit family, and also the Vesicle Prep Pro, small device to make giant unilamellar vesicles. Something that still, after many years, is very much in high demand from the market. In the center down part, you see the impedance technology that we introduced a few years ago. This is used for analyzing iPS cardiac cells, looking at contractility and basically doing drug safety assessment. And we also have on the lower right part our transporter assaying technology, the SURFER, solid supported membrane technology, sort of complementing ion channel assays with transporter assays. So something that we built to really enable research or enable you to facilitate and accelerate your research. And not only instrumentations, what we offer of course also the consumables and reagents that go with the systems. So that is how we spent the last 20 years, so to say. Well, we always keep things up to date and try to, improve continuously our instrumentation, broaden application range, and then also make new systems or new features. Recently, last year, we introduced our newest flagship product, the SyncroPatch 384. And the new system comes with a number of new hardware features and software features. Couple of things to mention, maybe the 32-well mode, so you can run the system in sort of partial plate using only 32 wells at a time to enable assay development or smaller scale work while still using the large economy of a high throughput system. And the new SyncroPatch also comes with a completely new temperature control system that not only allows work at physiological temperature, but also allows cooling of the system. Right, so we certainly try to stay innovative, and well, that's of course a bold claim after 20 years and even more innovative than ever. We at least try to do that. That is for sure. We're always looking for what is new, what can be improved, and well, also after 20 years, more successful than ever. That of course depends how you define successful. Well, we are having still a lot of fun at the company at Nanion. And well, last year has been our best year ever. We are growing as a company. That is, the business is growing, but also the team is growing and maturing. So that is clearly something that we feel is a big success for us. And well, to sort of keep it that way, we really try to stay on top of things and then engage in research and development. And we are very fortunate in terms of being able to work with work with a number of you guys, the best people doing cutting edge research. I just want to highlight a few of the recent things we've been able to engage in. We had a really great, or we really have a great collaboration with some of our friends in Göttingen, the cardiac group, that we managed to do something really outstanding, I would say. We managed for the first time to get our high throughput system to work with adult cardiomyocytes, this case from swine, pig cardiomyocytes. And then we just recently were able to publish that in Nature Communications Biology. That was really a fantastic collaboration and something that really surprised us that it worked so well. This is just a small image to show you how the shape of these cardiomyocytes look like. And if you see those, you realize, well, those are not easy to patch cells. They are not nicely round and small, but they are really large and big. But working with the teams, we were able to get that to work. We could differentiate the different types from the poor pig heart that we sliced into the atrial part and the ventricular part and analyzed those together with the team in Göttingen that is mostly led by Niels and by Will Seiberts, who is also in the audience. Happy to see you. That was a great collaboration, and they on the way managed to get a system up to Göttingen, and we are still collaborating further there. This is Niels Vogt and Thomas Mager, the two PIs that sort of brought the instrument to Göttingen. Thomas is also here. We will hear from him, and we are working within the sort of cluster of excellence there. Also combining high throughput APC with optogenetics, and we will learn about that in a little bit later. So great collaboration there. Another paper that we had recently was just showcasing the different applications that the new SyncroPatch can do. That was in techniques for physiology, basically showing the whole range of just using cell lines, but also iPS cells and even primary cells. Doing that in high throughput mode, doing a small screen, also dedicated biophysical characterization. Just an example here is some action potential duration measurements on these iPS cells on the SyncroPatch. And we will also hear about iPS cardiomyocytes in the first session. Well, and I mean, with all these applications, we do find a lot of clients and users for our SyncroPatch, both in academia, in pharma, and in CRO. And there was one recent installation that we were very happy about, where we had to develop or further enhance a specific application that was required by our recent client, the FDA, where we have installed the system last year. They were very much interested in having fluoride-free solutions for the recordings. Something we've been using for many, many years. Fluoride in the solution just enhances the seal formation and stabilizes the seal. But we've been working on making that not a requirement anymore. And after putting in some really in-depth research, we were able, together with some of our friends, to bring around an application where we do not require fluoride in the solution anymore, which we feel is a big achievement. There was also a recent publication on that. Just some data comparing fluoride-free and fluoride-containing reagents. The success rate overall is a little bit lower without the fluoride, not surprisingly, but it's still very reasonable in the range of 30 to 50%. So something we are very content with. Right, we had a number of best practice papers coming out over the couple of last years. This is working on the cardiac safety related targets, sodium 1.5, HERG obviously, and for CAF 1.2, there's a manuscript in the making that shall be published next year. And very recently, like 2 days ago, a paper was published where some of our team had a collaboration working on erythrocytes, and they were able to show in that work that the piezo-1 actually is responsible for bringing in an antigen to define a new blood group. Something very interesting, and well, we will hear more about piezo today as well. And that was published in Blood a couple of days ago. And one last thing I wanted to mention, because we just learned this morning that one of the papers of our group came out really hot off the press. There's a paper, we did it together with some friends at Metrion, looking at ASIC assays on APC platforms. So we clearly stay within the research arena and then try to engage with stuff that we find exciting, stuff that we know is important for you to do. And by doing so, we are really also always looking for improvements and additions for our instrumentation. So that really is what Nanion is about, making instrumentation for research, for researchers. And we will certainly stay like that and keep doing that. We will, well, we do embrace the science part. We have always a number of PhD students that we have within the company to keep this going. And well, we will also keep going with these grant schemes that we have been started to do in the recent years, that you can basically giving back that people can apply for getting instrumentation to use in their research in case they have not the funding to buy outright. So by doing so, we take great fun and also are able to bring together such a nice meeting. I'm really looking forward to the talks the next couple of days. I think we have a great lineup, great speakers and great presentations, really cutting edge research. So lots of cool things to hear about. Before I hand over to my colleague Sonja, one last thing, so to say, the what's next thing. We have something that is new that we will want to share with you today. It's not official, not launched yet, but it's in the late stage making. And well, as you are likely aware, Nanion has been, I would say, rather successful in making bulk plate based analysis instrumentation to look into complex assays. And well, we stay true to that. We keep working on instrumentation using that assay format, and we have something new that we will hear about in the next presentation. That is going to be presented by my colleague Sonja Stölzle, who is also running this project and then has been part of the development. And I hope you are excited now to learn what is new, what is next at Nanion. Other than that, I wish you a great meeting and great networking and everything. Thanks so far. And Sonja, please, you can take over.
All right. Thank you so much, Niels. So what's next? Our next system, our new system will be called Atlas. It is a quantitative live cell analytics device. The system is a impedance-based system, and it allows for recordings of 1 or up to 6 96 well plates. The system allows for real-time cell analysis. It runs in a label-free manner, and it does also an automatic graphing of the data, so of the results, while the recording is going on. And one key feature is also a very easy to use software and handling of the instrument. So like the power of impedance-based technology is basically the capability to be able to look at cell layer integrity from like seconds over 2 weeks. And with the system, you can tackle a lot of different research areas. You can investigate a lot of different cell types and targets as well. So for example, for cancer researchers, looking at cytolysis or cytotoxic effects, CAR T cells, or GPCR functionalities, just as some examples here. So the system will come soon. Oh, you don't see that now. It's going to be springtime next year. The system will be launched. And now let's have a look at the technical setup together. So, share my mouse here. So in the incubator, you have the recording station sitting for the 1 up to 6 well plates. On the outside of the incubator, you have a control station where basically all the electronics is embedded. So the amplifier, for example, and you also have a display where you see the status of your experiments, which are ongoing in here. In addition, you have a nice convertible laptop with the data acquisition and analysis software. You also do have a barcode scanner where you can scan cell plates and compound plates. Now, what is the system actually doing? The system deciphers complex cell responses, and it does it in a continuous way. So the measurements are continuously running in a real-time and label-free manner. And therefore, you sort of have big advantages, for example, over endpoint assays, because you can look at the kinetics of your cell growth, proliferation in the beginning, before dosing, for example. Then you can enjoy a walk-away time after dosing, after treating your cells, and you end up with multiple data points and the kinetic effects of your treatment, for example, compound treatment or immune cell application on cancer cells. So looking at the technological concept, we do have transparent consumables. Those consumables have a center electrode and a reference electrode around here. And with this technology, you can measure the impedance of cells sitting on the surface on the gold electrodes. A typical measurement looks like this. So here on the y-axis, you have your cell signal. This is basically the impedance signal that you get over time. And in the beginning, if you have an empty electrode, empty well, you have basically an impedance or a cell signal of 0. Then upon addition of cells, which sink down on the surface and attach on the surface and grow there, you have more and more cells impeding your ion current in between the two electrodes. If you have a full confluent layer of your cells, you reach your maximum impedance basically, and also a plateau phase. And then it's the time to add, to apply a certain treatment to the cells, for example, application of a compound. Now, this would be a typical experiment for a cancer researcher, where you could, for example, add immune cells when you have a confluent layer, and then you detect the kinetics and the time dependence and concentration dependence of your application. Let's have an even closer look to that. So again, here on the y-axis, you have your cell signal, the impedance. And now here on the x-axis, we have different data acquisition frequencies. And what you see is when you capture your cell signal with a higher frequency, you get more the capacitive current through the cells of your cell layer. When you capture your cell signal with a lower frequency, you capture more the paracellular pathway of the current. So you capture more cell-to-cell contacts, barrier integrity, and so on. So basically, this kind of electrical impedance spectroscopy, so spectroscopy because you have the full spectrum of frequency here, really allows you to gain a lot of rich information from the cells that you are investigating. For example, again, with higher frequencies, you look at things like wound healing, proliferation, cell density, cytolysis, cell death. And again, if you have a lower frequency of your signal detection, you look more at barrier integrity, transepithelial electrical resistance, or junctional impedance, for example. Now, to make this powerful technology adoptable for screening labs, what we did is we integrated predefined measurement modes. And these measurement modes basically own the settings that are required for the specific applications. So you get basically all your sweet spots that you would need to investigate your specific target, whichever research area you are looking at. So really, one key focus of the development of the Atlas system is, and was until now, also ease of use for our users, for the customers. So this is why we integrated these recording modes. And these recording modes come with a specific analysis as well. So you do get automatically the graphs, the plots that you need for your specific type of experiments. For example, for these cancer researchers out there, if you run the cytolysis mode, you get your kill time 50 and the cytolysis in percent. So this is what those labs need to report, basically, based on their experiments. At the same time, you can at any time export all the raw data without any analysis behind that. And of course, you can export the final figures and plots as well. Now, still, we cannot deny our Nanion DNA. Like Niels said, we are innovative, we love science, and at the same time, we also want our customers to be innovative when it comes to their own research. So we don't want any limitations regarding the settings. And this is why we implemented a certain level of sophistication into the software. So we do allow that the users can run the full spectrum of frequencies, because some researchers out there, some academic groups might want to look at different other things, cell death and barrier integrity as an example. And we also allow not to only have the impedance value as a signal, but also resistance and capacitance and the phase. So let's have a look at one example experiment here, a first case study. We got those cancer cells, A549, from the University of Munich, plated them on our well plates for like a couple of days. So we were growing them on the plate until we reached a maximum confluence with a plateau phase. And then immune cells were added to the cancer cells in different ratios. And we can see here that the more so-called effector cells are added, the faster and the more of the cancer cells are killed in the end. This would be the analysis that comes along, cytolysis in percent. So with the highest effector cell amounts, you get 50% of cell death on your cancer cells quite soon and quite early. And this is also represented, for example, in the kill time 50 plot, where you can see that this ratio here, for example, killed 50% of the cancer cells within like 14 hours, and the other ratio of effector cells killed the cancer cells earlier, after like 6 hours. Right. So as a summary, Atlas is a system for quantitative live cell analysis. It runs the measurements in a label-free manner and in real time. You get automated graphing of your results while the experiment is running, and it's really easy to use. So stay tuned, and thank you very much for listening.
All right, thank you, Sonja. Thank you so much. Are there any questions? I think we have time for maybe one or two quick questions, if there are any. And if they're not from this room, I'm not sure how they will get here, but I'm sure they will somehow. Maybe while there's Nothing to ask? Maybe. Oh, Lars.
Just a short question, because what you want to do is you want to relate your electrical data to your cell entities, and what is very helpful in making these relations, because there can be 100 things that happen. I mean, I understand that you specified quite a lot, but it would be nice to also have a visual inspection. Does these plates allow transparency from, well, to use it on a microscope?
Yes, it will allow that. So we will have transparent plates. So you can really look at the cells and do some more third-party investigations like imaging. You can do things like RT-PCR if you harvest the cells and so on. So this is, yeah, this is a good thing to do.
Anything further? Do you want to share why it's called Atlas?
Yeah, so the thinking here was, you know, the statue Atlas, right? This guy holding up the whole globe, the world. And the thinking was like, with this system, you can really, do so many experiments and capture so many research areas. So basically, the world of science in the hands of Atlas.
That is about the impedance, right? You do get that. that, right? Okay, thanks, Sonja. Great. Wonderful. Then, yeah, it's online, you mean? Yeah, please. So, yeah, it's my pleasure to welcome Professor David Beech, our keynote speaker for today. And I understand he is online. You can say a quick hello. Do I need to do anything? No.
Hi, good afternoon. Thank you for the invitation. Nice to see you. I'm very frustrated not to be there in person. I'm a big fan of in-person events. I've got 8 conferences this year across the world, but I've got a clash with another event and I just couldn't sort out the travel. So I'm in Leeds and I'm not there with you. So very sorry about that. So I've made a recording of my talk, but I'm going to be here for questions at the end if you have any questions. Thank you.
Most wonderful. Yeah, we also saw you couldn't arrange for coming around, David. We'll find another time. Would be great to have you over here at some point. I have a bit of a formal introduction. Let me just briefly do a proper intro of our keynote speaker. Well, David is professor in Leeds, but he started out in Manchester and did his PhD then in London. Did a postdoc at University of Washington before returning to London as a Wellcome Fellow, and then established his group in Leeds. And while his interest is in cellular mechanism around calcium homeostasis and general mechanisms of calcium as one part of his research interest, but his group also contributed to key evidence for roles of a number of interesting proteins, among them the TRPC1, 4, and 5, ORA1, and piezo1, and these and the function of these channels in cardiovascular and metabolic health and disease. And we'll hear a bit about the piezo1. Their work or the identification of some natural and synthetic small molecules modulators around the TRPC1, 4, and 5 channels actually led to a spin-off company called Caltech, or Caltic, that is. Not Caltech, that's well, way to go, I would say. No, Caltic. That was done together with some partners in Dortmund and the University of Leeds and the group in Heidelberg. David is also a fellow of the Academy of Medical Sciences. And apart from all this glory, he also calls himself the first user of the patch liner, which is, well, was 2006 and we launched the system. And David actually bought the first patch liner ever. And to stay to the truth, he also bought the second patch liner ever. And these two went to Leeds and have been serving him there hopefully well for a long time. Yeah, it's my pleasure. We've been engaging more, working together with David in the recent past, which is very nice and I think super productive. And we'll hear a bit about the piezo in David's recording, and then you're available for questions, I understand, right?
That's right, yes.
Wonderful.
Thank you, Niels.
All right, the recording will start now. I guess I don't need to do anything. Okay.
Good afternoon. I remember very well visiting Niels and Andrea at their original premises when they were first setting up Nanion. I was also the first to purchase a patch liner, and it's been an absolute pleasure to see Nanion develop over these years and reach its 20th birthday in a very buoyant state. I'd like to thank you for the invitation to speak here, and today I'd like to tell you about our work on piezo channels, which began shortly after Ardem Padaputian reported his discovery of piezo channels back in December 2010. It's also been a great pleasure to see Ardem receive the Nobel Prize for his discovery just last year. Before I talk about the science, I'd like to acknowledge the funding agencies that have very generously supported this work: the Wellcome, British Heart Foundation, MRC and EU. And all of these superb investigators, PhD students, postdocs and technicians who've supported the work and worked closely with me over these years. Our interest in these channels began with trying to understand the sensing of blood flow by the endothelium. It's been known for many, many years that the endothelium isn't simply an inert structure, but actually it feels mechanical force, and how it feels the shear stress generated by blood flow has been a fascinating topic, and it's really still quite poorly understood. Our idea is that the first event that occurs when shear stress starts or changes is the activation of a calcium permeable channel to start a calcium signaling cascade. Of course, to test that idea, you need to know the identity of that channel, and that was very unclear. We now think that is piezo 1, and I'm going to show you the evidence that makes us think that. We were triggered to do this because of Ardem's work on neuroblastoma cells, which identified a channel of 24 picosiemens, and that was of interest to us because of Joachim Heuer's work on human umbilical vein endothelial cells, which had suggested that the shear stress activated calcium permeable channel had around about that conductance. We hadn't managed to find one of that size, but then we thought perhaps it could be piezo 1, and so we started to study those umbilical vein endothelial cells, and it did look like piezo was really interesting. I'm not going to show you those data here, but what I'm going to do is start by showing you our data on the knockout mouse. So initially we did studies on the global knockout mouse, and then did an endothelial specific deletion. Hopefully you can see here on the left is an endothelial stain for CD31, PCAM1 it's called, at embryonic day 9.5, which is a day after the heart starts to beat. And it's known that what occurs when the heart starts to beat is that blood is pushed into the nascent vasculature, which you can see as a ring-like structure here. And then that blood flow causes the maturation of blood vessels so that they become larger and able to allow blood flow to the organs and the development of the embryo. We knew that this is a shear stress dependent process, so we thought that if piezo is an important shear stress sensor, it should be important for this maturation of the vessels, and indeed that was the case. So in the knockout you can still see green labeling for the endothelial cells, but it was very difficult to find any maturing blood vessels. The embryos were smaller, so they were growth retarded, and after about day 10, 11 or so, then the embryos were missing and presumably readsorbing. We also showed the same thing for endothelial specific knockout. We isolated endothelial cells from these embryos and applied shear stress to these endothelial cells in the direction of the arrow, and these cells are known to align to the direction of the shear stress. And very strikingly, in the absence of piezo 1, then the endothelial cells don't align anymore. We measured intracellular calcium, and initially the signal in response to shear stress here of 15 dynes per centimeter squared is quite small, but then it amplifies up, and we think there's a cascade of events that causes the signal to become larger. What's striking is that in the piezo 1 knockout is there's no initial rise in calcium and no subsequent rise in calcium until we apply a different stimulus, and that is vascular endothelial growth factor, so we can get a calcium rise in the piezo-1 deleted cells, but it's not a mechanically activated signal. Very strikingly and encouragingly, when we studied HEC cells, which are not normally shear stress sensitive, and apply shear stress to them and measure calcium, and there's no rise in calcium, but if we transfect in piezo-1, then we can get a very nice reconstitution of the signal. Because the piezo-1 is important in embryonic development, then its knockout is embryonic lethal. So in order to study the shear stress sensing mechanism in the adult animal, it's necessary to do a conditional knockout, so to allow the animals to develop with normal piezo-1, but then to conditionally delete the piezo-1. In this case, we use a cadherin 5 or via cadherin promoter to do this. These animals are relatively normal, but I'll show you some differences later on. So they grow at roughly the same rate, and we can then study the mesenteric vasculature. That's interesting to do as a vascular biologist, because we know the mesentery is an important site for blood flow, of course, to the intestine, but also for control of blood pressure. So we obtained second branch of the mesenteric artery, and then isolated the endothelium fresh without any culture, and then pinched off bits of membrane using a patch pipette, and had the outer surface of the membrane facing the extracellular medium, and then applied different shear stresses and recorded channel activity. Strikingly, there's some basal channel activity, which is shown on a faster time base here. The unitary conductance of these channels is 25.5 picosiemens, so close to what we were expecting. And when we apply fluid flow, then we see increasing channel activity, so they're fluid flow activated channels, which are then inhibited by gadolinium, which is a known blocker of the piezo channels. Most persuasively, when endothelial piezo-1 deletion has occurred, there's no activation of these channels, suggesting that it is indeed piezo-1 channels that are flow activated. What I've suggested to you is that the first event that occurs when shear stress is applied is the activation of piezo channels. So if that's true, then the channel should be very fast activated by mechanical force, and that is what we see. So we've done this using a variety of patch clamp configurations applied to native endothelial cells freshly isolated from the mouse. When we apply a pressure pulse to cell-attached patches, as is shown in the left-hand part of the slide here, then we see rapid activation of the channels. The activation is sustained, then when the pressure pulse is removed, the channels switch off. Sometimes in patches we see lots of channel activity, so then we observe what are referred to as macroscopic currents, and we can apply increasing pressure steps to the cell-attached patch and see increasing currents. All of these currents are missing in the piezo-1 endothelial-specific deleted mice. What I'd like to emphasize here is the very rapid activation of the currents. So within milliseconds, these currents are activated. So it is a very quick event, and it could be the first event when shear stress is applied. The channels then switch off very quickly when the pressure pulses are removed, and we get a nice pressure-dependent curve, as shown over on the right. I'd like to tell you a little bit about the way the channels might work. We have some insight into this from cryo-electron microscopy studies, 3 of which have shown the channel, at least part of the channel in the closed state, and a recent study shows part of the channel in what seems to be an open state. We've been using molecular dynamic simulations, working with Andreas Carli, and I'd just like to use this here to show you roughly how the channel works. We're incorporating the model into a membrane which comprises lipids that are known to exist in the endothelial cell membrane. In the resting closed state, the channel causes a really interesting inward curvature of the membrane of between 6 to 9 nanometers. There's a little cap at the top here, and this sits quite deep within the membrane when the channel's in the closed state. The channel is comprised of 3 piezo-1s, each of 38 membrane spanning segments, giving a total of 114 membrane spanning segments and a channel of almost 900 kilodaltons. When force is applied, and in this case membrane stretch, but actually it seems the channel is activated by a variety of mechanical forces, one of which is shear stress. Here when there's membrane stretch, then the membrane flattens, the channel flattens, and the cap rises up a little bit like a cap on a bottle, and it tends to twist. And when that happens, then the ion pore opens and ions flow through. It's a calcium permeable non-selective cation channel, And I'm emphasizing this because what these data suggest and this model suggests is that the channel is directly sensitive to mechanical force, that it responds very rapidly. An intriguing feature of piezo channels expressed in hex cells or reconstituted hex cells is that they also activate very rapidly, as I've shown you for the endothelium. But in addition, they inactivate very rapidly, and that was described in the original recordings by Ardem and by many other groups subsequently, and we can also observe that. So here we're expressing piezo-1 in hex cells, applying a pressure pulse, getting very rapid activation, and then in the continuous presence of the pressure pulse, the channels inactivate very, very rapidly. Just as a control here, we've mutated the channel, so it's a loss of function mutation, and then you don't see any current. So this current is coming from piezo channels. So piezo channels undoubtedly have a very fast inactivation gate. But what's really striking for the endothelial currents is that they don't inactivate. And some people said to us when we first reported these piezo channels in endothelium, they said that these couldn't be piezo channels because they don't have the hallmark feature of rapid inactivation. We thought this might be something about the patch configuration that we were using, because some variety in activation has been reported depending on the configuration, but we've done a variety of configurations and also, as shown on the right, recorded membrane potential from sheets of endothelium, applying fluid flow, and then here we see a depolarization as expected for a calcium permeable non-selective cation channel. In the absence of endothelial piezo-1, there's no such depolarization, and strikingly this is a sustained depolarization. So in endothelium, the piezo channels don't inactivate. Now, as I've mentioned, we've been doing molecular dynamic simulations. We're doing that to try to understand how the channels are activated in the environment of an endothelial lipid bilayer. And something that we observed early on was that some of the lipids that we've incorporated into the model associate with what is thought to be the inactivation gate of the piezo channels. That doesn't prove that those lipids actually sit there in the native channel, but in the model, sphingomyelin and ceramide are there near the inactivation gate, and that made us think that maybe sphingolipids are important for controlling the inactivation. We know that sphingomyelin is converted to ceramide by an enzyme called sphingomyelinase. There are several types of them, which I'll mention again in a moment. Not only is ceramide produced, but also phosphorylcholine. So we tested the idea that maybe sphingolipids, including ceramide, might be important for the inactivation process. And so we recorded from endothelium in the presence of an inhibitor of neutral sphingomyelinase 2, and that's called GW4869. Very interestingly for us, when we did that and applied a pressure pulse, we then saw rapid activation followed by rapid inactivation, just as you see for the overexpressed channel, but not normally for the endothelial channel. Also interestingly, if we added ceramide in the presence of the inhibitor of the neutral sphingomyelinase, then we could return to a non-inactivating current. Sphingomyelin and phosphorylcholine didn't do anything. We then wondered, what is the molecular identity of these sphingomyelinase? And the one that's described in the endothelium as potentially being most important is indeed neutral sphingomyelinase 2, also called SMPD3, sphingomyelin phosphodiesterase 3. Mutants of this enzyme have been produced previously, and we collaborated with a group in Toulouse to study these mice. They're called Fro mice. Their sphingomyelinase is still there, but it lacks the catalytic activity of the enzyme. Very strikingly, in the endothelium of these mice, you see rapid activation followed by rapid inactivation, rather like the overexpressed piezo-1 channel in hex cells. So when the sphingomyelinase is inactivated, the current in the endothelium is fast inactivating. So what we suggest is that in the endothelium, when there's no force, there's no activity. So there's probably some basal tension in the membrane that actually causes a bit of basal activity, as I showed you earlier on. Then when we apply force or shear stress, then we see activation of the channels and see a lot of current going through. And this is in the presence of an active sphingomyelinase, which produces ceramide. And then as time goes by, we're continuing to apply force, and the channels remain active, and you get sustained activity as shown over on the right, which is an actual recording from endothelium. But when the sphingomyelinase is blocked by the GM compound or by genetic disruption of sphingomyelinase, then there isn't so much ceramide. the channels initially do activate, but then the inactivation gate is allowed to work, the channels switch off, and so the current decays. So normally the sustained activity of the channels in endothelium depends on sphingomyelinase activity. So what does such a sustained activation of piezo channels actually do physiologically for the animal? Well, we thought it could be important for blood pressure control because we'd seen the channel activity in the mesenteric endothelium, and the mesentery is an important vascular bed for controlling peripheral resistance and therefore blood pressure. We were disappointed initially because when the mice were relatively inactive, then there was not much effect of the piezo deletion in the endothelium on the blood pressure, but we thought this could be important, mechanism could be important in exercise because piezo is all about physical tension being applied to the membrane or shear stress, and these kind of things are known to be increased when there's physical activity. Very strikingly, blood pressure of course goes up when there's exercise, which was previously known, and this is blunted by the endothelial piezo-1 deletion. So this was the first indication to us that actually piezo-1 might be an important mechanosensor, not only for normal cardiovascular physiology, but particularly in the situation of physical exercise. This is indeed interesting because we know that physical exercise is a great protector against cardiovascular disease. But we don't know how this works exactly. There has been suspicion that it might work by causing shear stress that stimulates the endothelium. So our idea is that piezo-1 is an exercise sensor functioning via the endothelium. We've done some further tests of this, and initially the results were a little bit disappointing. We did see the change in blood pressure, but relatively small effects on physical performance. But what we realized is that the length of time of deletion of the piezo-1 is important, and we've recently reported results on this in JCI, an article by Fiona Bartoli et al. Very strikingly, in these endothelial piezo-1 deleted mice, then the ambulatory activity, exploratory activity, running wheel activity are all strongly reduced by at least half when endothelial piezo-1 is deleted. But the enthusiasm of the animals to actually run on the wheel is not changed, so this seems to be a problem with their physical performance. I'm only showing you a brief outline of the study here. We've done a detailed study of the mechanism, and so I'll just briefly summarize what we think is the mechanism. We think that the endothelial piezo-1 deletion results in rarefaction of the capillaries, so fewer capillaries in the skeletal muscle. I'll show you some evidence for this in the top left-hand part of the slide, where we're staining the microvasculature with CD31 antibody, or glucan-1 antibody, and also staining for collagen 4, which provides the outer sheath of the microvasculature. And what we find is that in the piezo-1 deleted mice, that there are some endothelial cells missing, but the collagen is still there. So these are so-called empty sheaths, which is something that occurs when there's microvascular rarefaction. We could also identify, using tunnel assay, apoptotic endothelial cells in the piezo-1 deleted mice. So we think normally when there's blood flow in the microvasculature of the skeletal muscle, that stimulates piezo-1. I haven't shown you the details of the biochemical pathway here, but we think that then stabilizes eNOS enzyme, which generates nitric oxide, which inhibits the production of thrombosbondin 2 in the surrounding pericyte cells. So when the piezo-1 is deleted, or there's less blood flow, which we suggest occurs when there's sedentary lifestyle, lack of exercise, and then there's less stimulation of this mechanism, so less nitric oxide, so thrombosbondin 2 is released, and thrombosbondin 2 is known to cause apoptosis of endothelial cells, hence vascular rarefaction. Because physical exercise is such an important thing in determining cardiovascular health and indeed overall health, we've been wondering about whether it might be possible to enhance this mechanism in people who are unable to exercise, for example, because of hospitalization. Fortunately, in the piezo-1 field, Novartis, working with Ardaputian, screened 3.25 million compounds against overexpressed piezo channels and identified an activator, and we've tried that activator against the endothelium. As I showed you earlier on, flow causes the depolarization of the endothelium. If you don't apply flow, but instead apply this chemical activator, which is called Yoda-1 from the Star Wars films, Yoda for May the Force be with you. When the Yoda-1 compound is applied, and the structure is shown here, there's a very similar depolarization, and it can switch off after the Yoda-1 is removed, but in this case in the absence of shear stress. Both of these effects are missing in the endothelial piezo-1 deleted mice. So we've been working on the chemistry of these analogues. Yoda-1 is a good agonist of piezo-1, but it's not particularly potent. It's not particularly soluble in aqueous solution, and it's quite unstable, so unsuitable for in vivo studies. So we've been modifying the compounds, and I can't show you the structures at the moment, to produce more stable agents that still activate the piezo channels, and we've succeeded to do that. And these are the compound names, KT159 and KT289. They're actually more potent, up to 4 times more potent than Yoda-1, and more consistent in their agonism. But most importantly, shown in the blue table at the bottom, they have better aqueous solubility. They also have better thermodynamic solubility, as shown here, and also microsomal stability. We can show that the compounds are active. In this case, in the portal vein, you get a nice relaxation to the compounds, which is lost when nitric oxide synthase is inhibited, as shown down here, also lost when the endothelial piezo-1 is deleted. We've started a very nice collaboration with Nanion to study these compounds and to try to get mechanical activation of the channels in Nanion's SyncroPatch, which is a 384 well automated planar patch clamp system. And Andrea's group, working with Nadine Becker, have very nice now activation of the piezo channels in the cell lines that we developed for mouse piezo-1 or human piezo-1, and we're able to enhance those responses with the compounds that we produce, particularly KT159 as shown here, which is better than Yoda-1, and this KT157 is an inactive version. The actual currents are shown on the bottom left-hand side. So in the null cells, there's almost no current activated, but in the human or mouse piezo-1 cell lines, then there's a current that's activated by the fluid flow from the delivery pipette. And this is quite strikingly enhanced by the compound, in this case Yoda-1. Note the difference in current scale here, 200 picoamps compared with 20 there. So the take-home messages from my talk are that piezo-1 is activated by shear stress as well as other forces such as membrane tension. It's a very rapid native force detector. It's expressed in endothelial cells. It isn't exclusively in endothelial cells. In fact, it's expressed in a variety of other cell types and seems to be a general mechanosensing cassette. It has a fast inactivation gate, but this is disabled in the endothelial cells because of a sphingomyelinase enzyme, it's SMPD3, which is active in producing ceramide, which we think disables the inactivation gate. These sustained activated piezo-1 channels are important for blood pressure elevation in exercise, and we think piezo channels are physical exercise sensors. The channels are important in exercise performance in mice. They're important for muscle capillary density. It's possible to agonize the channels with small molecule agonists, and we've been developing the Yoda-1 medicinal chemistry for this purpose. And recently, working with Nanion in a very nice study, we found that it's possible to stimulate these piezo channels in their automated mechano patch clamp system. And I'd like to thank you for your attention, and I'm happy to answer questions.
Can you hear me?
Yes, yes.
Niels, we can't hear you right now.
Now it's on. Can you hear me now?
Now it works.
David, can you hear me?
Yes, now, yes.
Wonderful.
Good.
Thank you again for the interesting talk. I'm struggling a little bit to make the connection between the action of the channel in the endothelial cell and the systemic effect that you have sort of concluded, namely that it is a sensor of physical activity, so to speak, because my understanding was that shear stress is enhanced when blood flow in the vessel increases, right? And
Yeah.
that do you think that this results in an increase in in a sort of shear stress induced increase in total peripheral resistance, which increases blood pressure on exercise. That is sort of not the thing that I'm used to. I'm more used to vasodilation in response to increased blood flow.
Indeed, yeah, indeed, it's a good question, and we don't know the answer fully, but we discussed this in our Nature Communications paper on this topic, because shear stress can cause constriction and dilation depending on the vessel type, and in exercise there is a redistribution of blood flow, because we need more blood flow to the muscle, and the blood flow to the intestine, liver, etc. tends to decrease. The brain is stable, and what we found is that there's vascular diversity in the functional effect of piezo-1. This is complex to work out. I did show you an example where the piezo causes a relaxation. That's in the portal vein. It's a nitric oxide effect, but we also observed that there can be a constrictor effect in the mesentery. So I think this will depend on the native state of the nitric oxide system and the balance against other mechanisms. Piezo actually has a dichotomous, contrasting effect on the endothelial cells because it causes calcium entry, which of course will drive nitric oxide production, but it also causes depolarization because it's a non-selective cation channel. And depolarization opposes EDHF, endothelium-derived hyperpolarization. Hyperpolarization is known to cause relaxation. So the depolarizing effect will cause constriction. So how this works in vivo exactly, I think it is not clear yet, but it's clear that both effects can occur. So what we think is, when there's exercise, is that there's increased flow in the mesentery. It causes constriction to reduce the flow in the mesentery. That raises blood pressure and diverts blood flow to the muscle. I hope that partially answers your question.
So that's a hypothesis, but that would assume that simply by exercising you increase blood flow by the increased venous return or something like that. Is that the idea?
Increased blood flow through the increased cardiac activity.
Yeah, cardiac activity. So it would be subsequent to the sympathetic effect.
Yes.
Yeah. Okay, I see. Thank you.
Hi, Dave. Hi, David. This is Marcus from Nanion.
Hello.
I have a related question. Have you ever looked at the endothelial effect in young versus old mice? Because the flexibility of the endothelium is also age dependent.
Yeah, very good question. We only have a little bit of data on this because, of course, the studies of the older mice are more expensive and longer to do. But the piezo certainly is still active in the older mice, but we think its functional effects are different. So the stiffness of the cells and tissues increases with age, so we expect some changes. I don't have more information to give at this stage, but undoubtedly that's an important question.
Hi, this is Bjorn Nolman from Vanderbilt University. Great talk. I was sort of following up on that regulation of peripheral vascular resistance, and I was sitting there similarly perplexed because it sort of goes as opposed to what we'll usually think about physiology of the vascular system. Now, these are all endothelial targeted knockouts from birth, right? So you won't be able to decide which are like more like due to reduced capillary or change in the vascular anatomy as opposed to acute functional effects to a certain degree. So do you, having had the SyncroPatch, do you have an inhibitor of the identified inhibitor of the channel?
Just two things in response to that. First of all, the germline knockout is lethal. So these are not germline knockouts. This is a conditional tamoxifen-induced knockout or Cree-driven system at the adult stage. So first of all, the adult develops normally, and then we delete the piezo-1. So the vascular structure is normal. I did show you some change in the microvasculature, but the animals are relatively normal. On the second point about inhibitor, there's very little known about this so far. There are things like gadolinium and ruthenium red. There's a toxin called GSMTX4, and none of these are specific. We have screened our Leeds library of chemicals, about 11,000 chemicals against piezo-1, and we've got 2 candidate series inhibitors for piezo-1. We're hoping to test those in the Nanion system soon. My PhD student was there recently, and developing the protocols. And we're not quite there yet, but we're hoping to have some answers for you fairly soon.
All right. I would have a non-scientific question for you, maybe as the last question of this session. How for you personally, the research on piezo 1, how did that change after Adam got the prize? Is it a better situation? Is there more interest, more attention to it, or is it overcrowded, or how do you see it?
I think it's a bit early to say. Obviously, it's something really to celebrate that Adam's discovery was recognized in this way. I think us in the field are delighted. I think the ion channel field is delighted, and of course, trip channels were also recognized. So I think this is really great news. There is a lot of interest in the piezo field. It's accelerating. There are still only about 1,000 publications. It's still fairly small, particularly in China. There's a lot of piezo research and a lot of interest in whether it could be a target. It's important in skeletal muscle, bone, also red blood cells as well, in heart failure, all kinds of things. So I think it's a very early phase, and people are certainly joining this field. So I think it's good news in some ways, or competitive in others, but that's exciting. Yeah.
Right. Yeah, great. Couldn't more than agree. All right. Thank you again so much, David, for participating and having the presentation. That was wonderful. We will then move on to our next speaker. Thank you. Right, yeah, it's my pleasure to introduce our next speaker, who comes from Göttingen, Professor Thomas Mager. He is the group leader in the Institute for the Auditory Neuroscience and Optogenetics at the University in Göttingen. He did actually start doing his PhD in biochemistry in Frankfurt at the Goethe University. He did a postdoc at the Max Planck with Professor Bamberg at the time, and then another postdoc with Tobias Moser already in Göttingen before starting his group at the University Medical Center. And he's also part of the Cluster of Excellence as the application specialist for the multiscale bioimaging. I really have to say, we have been working together for a little bit on co-developing instrumentation. That has been great fun and really is pushing the limits. And I can't say less than that I really love the title of the talk, Opto SyncroPatch 384, Next Level Optogenetic Tool Development. Very much so. Take it away.
Thank you for the kind introduction. Hello to everyone. As you said, I will talk about optogenetic tool development using the SyncroPatch 384. And first, what is optogenetics? It combines optics and genetics to remote control cellular function with light. And this shows the number of PubMed entries connected to optogenetics. And as you can see, that since the discovery of channel one absence, that is light-gated ion channels, which are used for activation and silencing of excitable cells, that this number increased substantially, and it stopped here 2010, but I can tell you it increased steadily further up to now, where it's now roughly 1,500 publications published about optogenetics. And the question is why, right? So the optogenetic approach has some benefits, and a clear benefit is that it has a very high spatial resolution. And also the temporal resolution of the technique is very high. And it's moreover also a reversible technique. So you can switch light on, light off, and you can do that on and on, right? And when you have specific promoters, you can also, this technique, this optogenetic approach is cell type specific. The Cluster of Excellence in Göttingen recently purchased the SyncroPatch 384. And we are happy to we have received the device in spring this year. And as you very likely know, this is enables recordings of up to 384 wells simultaneously. And what we did is that we operate this SyncroPatch 384 in synchrony with a prototype of an illumination device. This is currently covering 96 out of the 384 wells. And this now allows to run stimulation protocols, optical stimulation protocols, and to conveniently adjust frequency, intensity, and pulse length of the pulses, and do all of that in synchrony with the electrophysiological measurements, right? So, and also light stimulation at different wavelengths is possible. So they are available in different wavelengths. At the moment, monochrome, but different wavelengths are available. So this now enables the combination of high throughput or high performance automated patch clamp and optogenetics. And by this really, I think, drastically extends the experimental scope for optogenetic tool developments. My group is in the Institute for Auditory Neurosciences. And one of the major goals of the Institute for Auditory Neurosciences is to develop an optical cochlear implant. There are currently roughly 700,000 otherwise deaf users which are using electrical cochlear implants. And so there's a problem coming with this kind of activation. So the cochlea is organized in a tonotopic way. That means that the place within the cochlea determines the pitch which you hear. But the electrodes of this electrical cochlear implant, the electric field is really not focused, it spreads, right? It's an electrolyte. So therefore, a high number of these spiral ganglion neurons, which are in the auditory system, going to auditory nerve from the cochlea, are activated. So there's the frequency, so there's a broad range of frequency activated at once. Therefore, these cochlear implant users have problems in enjoying music or also to hear, to understand in the presence of background noise. The idea of the optical cochlear implant is now that light can be very precisely confined in space. And therefore you can really, significantly increase the frequency resolution. And this is then, will in turn also lead to closer to natural hearing for the deaf. So that's the goal of this optical cochlear implant. Channel wall absence suitable for use in the future optical cochlear implant should have certain properties. One is that they should have fast channel kinetics. Because these spiral ganglion neurons are fast spiking neurons, and we want to drive them and close to physiological rates. That's, for instance, important for directional hearing, right? So to locate sound. Another thing is that these channel wall absence used there should be red light activated, because red light is far less phototoxic than blue light. So that's a safety issue there. And they also should also generate big photocurrents, because big photocurrents enable the photostimulation of those neurons at moderate expression levels and also at low light intensities. And one channel wall absence which is actually meeting these criteria is at Crimson. But I want to tell you now that quite recently we engineered a new variant, which is based on a channel wall absence called Carmine, which was discovered in the labs of Deuis and also of Spudich. But we improved that channel wall absence by introducing certain mutations. And as you can see, is that the photocurrent density increased significantly. And we are now having photocurrent densities which are, down here you see, let's say, the common really state-of-the-art wall absence, the channel wall absence which are most efficient. But this one, which we have now engineered, is significantly higher in photocurrent. And that now allows really a more efficient way of activation. But there's a problem, or not a problem. So the optimal wavelength for activation is in the green, not in the red. And also the channel kinetics, so this is that mutant, is way slower than what is needed in the optical coherent. So it has really big photocurrent, so it will likely be much more efficient for the activation. So you don't need to boost expression so much. You can apply lower light intensities, but it is a bit slow and it is green light, not red light activated. So that's why we, one of our goals is to take this around and shift it to the red and accelerate closing kinetics. And we want to, for that, also use the SyncroPatch, which of course extends, possibilities to reach the goal. And we are also interested in, of course, finding the reason for these big photocurrents. And I will tell you a bit more about this on the coming slides, also using the SyncroPatch. So what we did here is, first, the channel wall-absence have really, really small photocurrents. So you cannot do single channel conductance measurements in excised patches or something. So you do need to do a stationary noise analysis. And this is usually very time consuming. So I had in my former lab in Frankfurt, there was students working really for a month or almost a year to get value their very repetitive measurements, right? And we are now really with having now this machine, we could, it's not months, it's a day of measurements, and we get enough data meeting the criteria needed to derive those values from stationary noise analysis in this very, very short time, which is of course a very nice thing. But what we found there is that catch, this is one state-of-the-art channel wall-absence, has a lower single channel conductance than carmine wild type as well as the mutant. So you see that the single channel conductance is a bit bigger of carmine variants. So now you can ask the question, why is then the photocurrent of the mutant bigger than that of the wild type, right? So, and what you see here is, this is the stationary photocurrent at different intensities. And the black line is the wild type. You see that this is measured in manual patch clamping. So just to say that, the photocurrent increases and then it decreases to a value which is lower but unequal to 0. This is very, very atypical for channel wall-absence. I have not seen that before, right? So this is indicated for a substrate inhibition of the partial type. So the substrate light activates and it inactivates, but not to a closed state, but to a state which is a bit lower in, let's say, photocurrent, a lower photocurrent state. So this indicates a second photon process leading to a reduction of the photocurrent. So, and our mutants now show here, this is a semi-logarithmic scale, so a sigmoidal dependence, right? So we are classical hyperbolic dependence on light intensity. And that shows that we could, by introducing those mutations, eliminate sort of a light-dependent inactivation process in those mutants. And what is really interesting, that is in the power spectra, which we use for analysis of the single channel conductances. So for this, we observed the following. There was, on the left side, you see the power spectral density of carmine wild type. And on the right side, there's a power spectrum of carmine dull mutant. And there's a high frequency component visible in the wild type, not visible in the mutant. Right? We also quantified that for many cells. That means that there's likely a second short living, because it's in the higher frequency range, open state seen, right, for the wild type, not seen anymore for the mutant, which is really quite interesting, because I think that explains possibly, potentially, why there is a reduction of photocurrent in the wild type, not present in the mutant. So it's a second photon process, likely leading to possibly lower conductance, short living open state, which we, by introducing the mutations, get rid of, right? And so we have a very, very efficient tool in our hands. And this is was again done with the SyncroPatch, very fast, very nice. So, oh, that was the wrong direction, sorry. Okay, but we have now, as I said before, there's really big photocurrents, possibly because of the higher single channel conductance and the cancellation of the inactivation process, but it doesn't have very fast channel kinetics, and it's also not red light activated. And we want to, for achieving that, we want to also use the SyncroPatch, which we received in spring this year. And we have been working on the optimization of the workflow. For that, we plan to screen certain mutant libraries. I will explain that a bit more in detail later. But what we did here is that we performed transient transfections. So this is transiently transfected cells. And out of this, we get, at the moment, 25% success rate, meaning 25% really having perfect seals, low series resistance, and of course, having in mind, let's say, the transaction efficiency. A quarter of those is giving, at the moment, photocurrents, which can be used for further analysis. And that means that we can screen roughly 12 Orions per chip with n equals 8. What we also did is that we performed the FACS pre-sorting. So clear, so we have a certain transaction efficiency. If you only sort those which are transfected, we then, in this case, all patched cells show photocurrent, right? So this is of course beneficial at that point. But you need a larger amount of transfected cells or starting materials to do that. Thinking about screening, so of course you have to think about costs and time for additional step and for additional material, meaning more cells, transfection reagent, and so on. And so I think that the use case for this would be when we really want to look into a variant closer, right? If you, and not the screening approaches. And the other thing which we tried is the 4-hole chip. In this case, we had a success rate of up to 90% or roughly 90%. Why is it moving already? Sorry for that. And I think the use case for this would be if the question is a certain variant functional, yes, no. So because we will have unfunctional variants in there. So I think that this 4-hole chips are very valuable in that regard, right? What we plan to do with that is now that we will generate mutant libraries. So there's certain spots inside the protein which are so hotspots for certain functions, right? And we want to generate mutant libraries at these hotspots for function. So we will at certain spots increase variability, right? And then have mutant libraries with the goal to then have something which is big, red, and fast in the end, right? And we will do that on the one hand by the mutant libraries, right? And variability at hotspots for function, which we know and which we think they are. So there will be new spots, but also, let's say, from homology and what is known from literature, some of the spots known. And the other thing is, of course, we will characterize previously unknown BCCR. BCCR is the family in which also this protein belongs to. They're a bit different than germosins, which were found before. They are trimeric, not dimeric. Have other protonation pathways. Protonation pathways are important there because they determine, these protonation reactions determine the open time of the channel. So they are linked to channel kinetics. And so we will perform natural variant screening and mutant library screening. And in the first step, check for plasma membrane expression using spinning disk confocal based system from Yokogawa, a high throughput system. And then whatever is nicely localized will then subsequently be tested on the SyncroPatch 384. And I think that is looking forward to the, you have already quite first quite promising results coming out of this pipeline. Thank all of the particularly Tobias Moser for the opportunity to work on these projects, and also Alexeyev, who's sitting over there in the audience, who has been very SyncroPatch workflow and has also determined the single channel conductances shown in. Thank you for your thank.
Of other drugs, thank you, of anticonvulsants and antidepressants. And in many cases, patients actually choose to come off their medicine and live with their pain because the drugs are just not that good. So we're now looking at many other pain targets that are non-opioid and non-addictive. This is the main and big issue, especially in the US, of course, where we've got the opioid abuse and addiction crisis. So there's a number of ion channel and receptor targets that people are particularly interested in for analgesia. And I'll touch on some of those today. So those are expressed in the real DRG sensory neurons. The second area where sensory neurons could be quite useful for drug discovery is in respiratory disease and chronic cough. So we've got vagal sensory neurons that innervate the lung. So they can actually respond directly to airborne irritants, and they can also respond to local inflammation. The very popular targets at the moment are the P2X receptors. So there's a number of these in the clinic. The earlier drugs are not very selective for the subtype 2, subtype 3, but it seems that the more selective compounds have fewer side effects. So there's a push on to develop more of these drugs. And there's also a number of companies such as Nanion that are developing traditional sodium channel blockers, but Nanion are taking a rather unusual take on that. So there's some classic receptors and channels in vagal sensory neurons that are relevant for drug discovery, and I'll talk about some of those in the anatomic sensory neurons as well. And finally, there's the other side of drug discovery, which is safety pharmacology. So peripheral neurotoxicity is a really important and debilitating side effect that's seen in the clinic and with many patients. Traditionally, this has been associated with chemotherapy and HIV drugs, but we've also seen a number of recent clinical trials halted gene therapy trials because they started seeing peripheral neurotoxicity in their patients. So this is an ongoing issue. The FDA recognizes it. They've created this new tox initiative, which is similar to the SIPA cardiac safety pharmacology. Its main focus is on CNS seizure liability, but they're also looking at peripheral neurotoxicity. And why this is important with stem cells is that some of the known neurotoxicants are not active in the rodent cell and tissue assays, and that's why we need human tissues for better translation and more predictive drug discovery for efficacy as well as side effects. So the cells I'm going to be talking about today are developed by Anatomic in the US. So they use a cocktail of small molecules and growth factors to direct the differentiation of the cells from the ectoderm through the neural crest and onto the immature sensory neuron phenotype. So we know that these are sensory neurons because they express two of the characteristic transcription factors, so IL-1 and BRIN3, and they're also labeled with the peripheral neurofilament peripherin. And this process is very rapid, 7 days from IPS to immature sensory neurons. After 7 days, the cells are frozen and then they're shipped to the user for additional maturation. And there's a couple of real valuable advantages of the stem cell approach compared to existing approaches. The main one of which, obviously, the main competition here is native human DRG tissue. And it's incredibly hard to get that. It's very expensive to get that. And it's even harder to get viable preparations where you can make functional studies in cells and tissues. So the sample sizes from human DRG are very small, and they're not representative of all the different nociceptor subtypes that we know are expressed in adult DRG. They also can be very expensive. And the other issue, which I think is a bit delicate, but is also very important to mention, which is ethics. So human donor programs allow research work. They may allow not-for-profit, but they very, very rarely allow commercial exploitation. So with the stem cell neurons, such as those made by Anatomic, we have a large supply, so a scalable supply of a large number of cells, which allows the sampling of different nociceptor types. So these cells populations, they're 95% pure, but that's not just one cell type. There are multiple nociceptor phenotypes in that population that are representative of the human DRG. This is a scalable approach, and it's also got commercial freedom to operate. So there's a number of advantages over what is still perceived as the gold standard, which is native human tissue. The main driver for work on human sensory neurons, of course, is species differences. The industry still relies on preclinical animal models and cells and tissues. And there are really big and quite worrying differences in gene expression between rodent DRG and human DRG. And there's just one example here from Ray looking at mouse versus humans. So we've got genes along the x-axis here. This is native human tissue, but in the rectangle, and this is native mouse tissue. And we've got one type of nociceptor here and another nociceptor over here. This one on the right, PEP1, we have a pretty similar pattern of gene expression in the human and the mouse in terms of the relative expression and the levels of expression. But if you look at the PEP2 nociceptor, you see an almost reciprocal expression of genes between the human and the mouse. So only 63% of the genes that you find in the human DRG are actually expressed in the mouse. We don't want to cure pain in mice, we want to cure pain in humans. And if you have these massive differences in the gene expression, then you are going to fail when you get to the clinic. And what's really different is with the Anatomic's real DRG, 90% of the DRG specific genes in the human, adult human, are expressed in their sensory neurons. And that is an amazing point by itself. And that obviously gives tremendous potential for further maturation and looking at different phenotypes of the sensory neurons. So this is just a snapshot of some of the ion channel gene panel that was profiled. So we see the anatomic data over 4 weeks of culture, and this is against native human, adult human DRG tissue. We see some ion channel genes and receptors that are expressed at high levels. Most are expressed at similar levels, and we have a couple at the bottom that are obviously expressed at lower levels because these cells are not yet mature. They haven't been fully differentiated and matured yet. This is just a snapshot after a couple of weeks in culture. So there is still work to be done to increase the maturation. This is an everlasting question about stem cells, but I will address that in a future slide. But this is very promising. We're getting a lot of the genes we expect and hope to see from human DRG expressed in the stem cell sensory neurons. So what we're particularly interested in is seeing if they're functional. So today I'm just going to focus on automated patch clamp. So I think this is actually a really good technique for studying sensory neurons compared to, for example, CNS neurons, because the neuronal architecture, the morphology of nociceptors is very simple. There's no dendrite, there's very little synaptic activity or connection. So you've got a large soma, very simple axon. So when you dissociate these cells, you have a nice big axon. It goes very well with automated patch. You can have nice recordings from the soma. You have good voltage control. You have most of the targets that you're interested in are expressed in the soma. And another thing, especially when you go to high throughput, is we think there's up to 12 different nociceptors present in the human DRG. So you need big samples. So if you're only going to sample 20 or 30 cells, you might only have 1 or 2 each from each of those different nociceptors. So you need much larger samples. So you can then start to break down your data by phenotype. And automated patch certainly gives you that opportunity. Now, some people might think it's going to be really expensive running all those cells, stem cells on an automated patch. You've got to pay for the chips, etc. Actually, it's not. So the other major technique that most of you will be familiar with, of course, is multi-electrode array. And that relies on high levels of spontaneous activity and dense synaptic connections. Neither of those happen in DRG sensory neurons. They pretty much stand alone. So if you want to do an MEA, you've got to have big wells, lots of electrodes, lots of cells, which means you screen fewer compounds per plate. So it's actually more expensive to do it on MEA than it is to do it on automated patch. The other major technique, obviously, is calcium imaging, high throughput, but it's indirect and there's no voltage control. So we think the argument for using stem cell sensory neurons on automated patch is actually quite compelling. Now, a traditional technique that you use to classify or categorize your patch clamp data from sensory neurons is to look at cell size, which you measure with membrane capacitance. And I thought we could certainly extract that data very easily from the automated patch recordings. So we've got a sample here of nearly 500 neurons that have been run on the SyncroPatch. And we see exactly what I was hoping to see, which is a distribution of cell size, which is multimodal. So we have a collection here of small diameter neurons, a larger spread of medium diameter neurons, and maybe a little hint here of some larger diameter neurons. And this is very similar to the cell size distribution you see from the rat DRG. And this is very important for classifying DRG neurons. This is the data from single cell transcrimics. So 12 different nociceptors, each with a different distribution of their size. So this is data that we can extract very quickly on automated patch to help classify and categorize our data. So now let's concentrate a little bit more on the specific nociceptor pain-related drug discovery targets. The usual suspects. We have good expression here of the TTX sensitive and TTX resistant sodium channels. The red bars are the real DRG sensory neurons, and the black bars are native adult human DRG. So we see very similar expression of the sodium channels. We see expression of all 3 of the T-type calcium channels, which are also pain drug targets. And we also see the P2X2 and P2X3 in different relative amounts to adult. So this is quite interesting for those people working on chronic cough. The target that you always get asked about when you're working on sensory neurons is TRPV1. So it's no longer a popular drug discovery, pain drug discovery target, let's say that, but it is still a very important marker of certain types of nociceptors. So yes, it is seen, but at lower levels at the protein and the RNA level compared to the adult DRG. So there is still work to be done to increase the maturation of those specific nociceptor sensory neurons that express TRPV1. But this is a snapshot at 1 week, 2 weeks. If you look at the time course of expression of these important nociceptor markers, you see this rapid induction, and then that is maintained for 3 weeks. So we've got week 1 out to week 4. So these important and relevant pain targets are expressed in a stable way for a long time. This gives you a really nice wide assay window for assay development and screening using the stem cell sensory neurons. So what about functional responses? So some of you might have seen that Anatomic put this heat map of sodium channels up on LinkedIn a couple of weeks ago. Maybe if people were paying close attention, they might have been a bit surprised to see the cardiac nav 1.5. What on earth is that doing in a sensory neuron? Well, that's completely normal and completely expected. And it's been known for a long time, for 20, 30 years, that that channel is normally expressed. It's developmentally regulated in the DRG. It's expressed at high levels in the neonate, and then it's downregulated, but persists into adulthood. And this is exactly what we see from the transcriptome. High levels after 1 or 2 weeks of culture, which then decrease as the cells mature in vitro. And this is seen in transcriptome data from human DRG, adult human DRG. The NAV1.5 here is the last column. Low level, but throughout all the different nociceptor types, except for a hotspot here in the A delta mechanoreceptors. And it's also been seen many times in other stem cell derived nociceptors. So this is work from Angelica Lampart's group, looking at the NAV1.5 RNA expression in ES and IPS derived nociceptors. She also looked at functional responses. So if you use a concentration of 5 micromolar TTX, which largely inhibits the human NAV1.5, but doesn't touch the human NAV1.8, it produces significant inhibition of the inward current in nociceptors. So there is functional NAV1.5 in DRG neurons, and that is perfectly normal and expected. So that is actually a good result. So we've got all 3 of the expected TTX resistant currents in the anatomic sensory neurons, NAV1.5, NAV1.9, NAV1.8. And how do you discriminate between them? Well, the first one is obviously using TTX. And as Angelica did, if you have a moderately high concentration of TTX, you can block 1.5, but you can then leave NAV1.8 and NAV1.9 alone, so you can separate the current that way. A little cleaner, perhaps, is to use NAV1.8 selective antagonists. People are trying to find out the structure and buy the compounds from Vertex that I put into the clinic. That's a bit difficult. So most people are using this older compound from Abbott, 803467. And what was really interesting is a study from Eli Lilly and Michael Gold a couple of years ago in human DRG, is they were surprised how little of the inward current was blocked by the NAV1.8 selective inhibitor. And when they thought, well, maybe there's something wrong with the experiment, so they went and did this in the rat. Much more is blocked in the rat. And again, if you look at the kinetics of the currents in the human and the rat, this is what most people expect NAV1.8 to look like. This is what it looks like in human DRG. There's a lot less of this persistent current. So we need to reset our expectations about what NAV1.8 is going to look like in a human sensory neuron or a human DRG neuron. You just try and remember that. I'll go to some data from the SyncroPatch. These are the TTX resistant currents from the anatomic sensory neurons. They look very similar to what you see from NAV1.8 in the human. They don't look like what you see in the rat. So that's a really excellent result. We have robust TTX sensitive currents here in blue and red, but it's a lot harder to resolve this small component of TTX resistant current. So these are the real DRGs on another automated patch clamp platform. Quite hard to resolve the TTX resistant component. It's much better resolved on the SyncroPatch. We've got very nice voltage control because the activation curve is nice and smooth. And if you look at the TTX resistant current IV curve, that has been shifted to the right, which is more typical of the way you expect NAV1.8 to be, not NAV1.5. So we think that this is largely NAV1.8. As Eli Lilly saw, the amount of TTX resistant NAV1.8 current is actually surprisingly low in human sensory neurons, around 10 to 20%. We see this on SyncroPatch, and we also see similar fraction when we do the same experiments on PatchLiner. Well, what is interesting is that the fraction of current and the number of cells expressing that current seems to increase slowly as they mature in vitro. So this is what we would expect as the neurons mature and become more like adult DRG neurons. The other advantage of automated patch is being able to use the current clamp capabilities to record action potentials and assess excitability. And this has worked very well on the PatchLiner and the SyncroPatch. You do see some spontaneous activity, but thankfully it's very low. And this is only a couple of the % of the cells actually show this. And this is exactly what you expect from healthy DRGs. If they're somehow inflamed or injured, then you have higher rates of spontaneous firing. This is why it's difficult to run DRG on a MEA, but we can get some beautiful recordings from individual neurons on the automated patch. We can evoke firing. You see repetitive firing on the SyncroPatch and the PatchLiner, but you also see rapidly adapting firing. So again, this is confirmation that we've got different phenotypes, different types of nociceptors in the anatomic sensory neurons. So we have a mixture of cell types with different behavior, different repertoire of ion channels and receptors. And I'm particularly impressed by this slide. So we can use these complex steps and ramps, current injections, to assess the excitability of individual sensory neurons. This could be very important if you're developing or trying to develop a pain drug with a particular mechanism of action. Say it's state dependent, say you want to have a drug that is or is not use and frequency dependent, or you can profile it using these kind of protocols on individual cells with a high throughput. And this would also, I think, be very interesting to classify pain and patient-related channelopathies that you could express by genetic engineering in the sensory neurons. So again, this is preliminary data coming out of New Jersey, but I think it looks really impressive, and it shows the potential for phenotypic screening with sensory neurons, stem cell sensory neurons. So the last two slides are now going to switch to looking at ligand-gated responses. So another advantage of the automated patch is you've got much better control of agonist application and washout than you do on plate-based assays like CASM imaging or MEA. So you can control the volume and the speed of application using the puff and the stack tip applications. And I've got two examples here. The first is GABA. So this is not a synaptic transmitter. This is a local neurotransmitter in the DRG, which people might not realize. So this is an expression of GABA receptor subunits in the rat, and this is their expression in the human. So all across different nociceptor types, you've got GABA-A receptors. So they're depolarizing, the inward currents depolarize the membrane potential, and we can look and we can evoke rapidly activating desensitizing currents with a fast application of GABA on the SyncroPatch and the patch liner. So yes, it does desensitize, but you can wash that off. And so we're now working to get repeated applications so we can do a GABA-A screening assay. This is GABA-A because this is data from last week, thanks to Tim and his team. So it's blocked by the classic GABA-A antagonist, bacuculin. And what we see in terms of functional expression is the cells gradually mature. So when you go out to 4 weeks, you see an increased functional response to GABA-A. So it looks like this response is gradually maturing as the cells mature themselves in culture. And the final example here, again, to the P2X23. I don't have time to go into this slide, but this is a target that shows it's another example of very big differences in the expression pattern between rodent and human. So when we did the preliminary experiments here, we could see these very rapidly activating and desensitizing currents in response to ATP, but we could only do it once. They just do not recover from the desensitization. It's quite profound, and this is normal for this receptor. But Tim and his team, they had a good idea to use a ATP inhibiting enzyme, degrading enzyme, hexokinase. So when you put this into the solution, you can get repeated applications to puffs of ATP. So we have the potential here for a P2X3 screening assay, which is obviously of great interest to those people developing selective ligands for chronic cough. And in comparison to what we saw with GABA-A, the expression of P2X functional responses actually decreases maturation. Okay. But Anatomic are working on this. They've got a client who's interested in this particular target. So they've looked at a number of different media and protocols to try and increase and maintain the expression of this particular target. And this is really one of the two final points that I want to make that I think is very important, that these cells are not yet finished. They're not fixed. They're still plastic. They're still malleable. So they're still maturing. And that gives the opportunity for all of the users to develop their own protocols to really create their own assays, bespoke assays for particular pain signaling or neurotoxicity mechanisms for particular targets that they're interested in. And I think that is a real potential. When you get tissue from a human, it's fixed, it's done. But this is still malleable. So it gives tremendous opportunity and potential for assay development and creating really important and useful translational assays. And the other aspect, of course, is simply that these are just a very usable reagent. So they rapidly mature. They're available at scale with affordability and a commercial freedom to operate. So they have great potential as translational tools and reagents for neuroscience drug discovery. So hopefully I'm on time. Thanks for your attention, and I'm happy to take some questions.
Thank you so much, Marc. That was really exciting. Or I have to say, I am very excited about the real DRGs. I think this is, yeah, really way to go. It's well, something we have been trying DRGs over the years here and there. I think mostly from rodents, and that never really went anywhere. So having IPS-based DRGs available, that is a big step. Just to mention that there's also from the company who does that, Anatomic, we have also a participant here, Vincent came over. So any specific questions regarding these real DRGs can also be posed to Vincent.
Vincent Patrick. Vince, there he is. And also thanks to Tim and the team in New Jersey for generating the electrophysiology data.
Right, that's the Nanion team in New Jersey. Right, so Jan.
Thanks very much. Very interesting. I was just wondering, you in the very beginning, you mentioned they have big axons and you can patch those. Can you actually patch the axon? I mean, I know that some DRG cells have really large
they're
axons.
pseudo unipolar, so it's supposedly the same axon that goes to the central terminals as well as the peripheral skin synapses. So,
but
no, they disappeared. They are present in the culture.
Yes, sure.
But I don't know. I'm pretty sure, Vincent, no one's actually tried to do any sort of functional recordings. I mean, calcium imaging, you could get dyes in there and see responses in the axons.
You're basically relying on the fact that, strangely enough, these DRG cells, they express whatever they express on their axons, they are also expressed at the
absolutely, yeah. They're a fairly simple kind of neuron, and the assumption and the data tends to support this is that most of the targets, if not all of the ones that are of interest that may be expressed over here, are actually coming out of the soma.
And
so
do
you can still
do you see the same sort of functional heterogeneity in your stem cell derived DRG cells as you see in the, because not all of them are nociceptive, right?
In this population, yes, we think they are, but they're not fully mature. So that particular phenotype, there's a lot of different ways of classifying them, and you have to do this polypharmacology, which can be very, very difficult. So one of the future hopes is to be able to do this sort of sequential polypharmacology on the automated patch, put on GABA, P2X, sodium channel antagonist, record some action potentials. Quite challenging, but it will give you a definition of which cell type you're making that recording from.
Classically, the A delta and the C fibers are the nociceptors, right? Those are the ones that are the classical nociceptors.
Well, sorry, yes, nociceptors as opposed to proprioceptors, as opposed to, well, you guys think that you might have non-nociceptors in there as well, Vince, right? Yeah.
We've been looking at some of the subtypes. There's another publication out there that breaks down, that does single cell spatial transcriptomics, and we take those gene fingerprints and we run our bulk RNA-seq data to sort of look at these plots here. So everything shown in red has an R log value of 6, which is considered to be expressed. We express a lot of these genes on the bottom right. Those are a lot of the common pan-DRG neuronal. There are some missing, like the TRPA1 nociceptors that we are trying to work on, but we're thinking, we have some A delta mechanoreceptors, some of the pink nociceptor genes that are expressed.
So it's quite a diverse population. So there's a lot of potential here to be really drilling down into these different phenotypes.
Right, there's another one in the back.
I have two small questions. One is the functionality of TRPV1 throughout the 4 weeks. When is it closest to the human DRGs? And how difficult is it to do a single cell suspension after 4 weeks when you get to your primary neurons, when you put them on the automated patch?
I can't really answer the first question myself because I'm not familiar with all the data that Anatomic have themselves and with their collaborators when they are looking at functional responses to TRPV1, probably with calcium imaging rather than maybe with MEA rather than automated patch, but these are on the list of experiments to do. And your second question here is about in terms of dissociation, or we can look at this in terms of success rates. So you can see that the success rates here at 4 weeks are not different from those at 2 weeks. So the cells are still able to be dissociated. They're still healthy. They still have responses that you're interested in.
How challenging is it to make it single cell?
I'm the lucky guy because I haven't actually done any of these experiments, so I have no idea. I don't think we have this much data if it was that difficult, but I don't want to undersell the work that's been done, the assay development that's been done with the collaboration between Anatomic and Nanion in the US.
Yeah, we essentially get capsaicin responses after 4 weeks via calcium imaging. We're actively working on getting it on the automated patch clamp system.
Thank you very much for the excellent talk. I was wondering, because you mentioned several times maturation, I was wondering if you're working on maturation profile. are you going to have like another sensory neurons, like a 2.0 sensory neurons, or are you like working on maturation media, maybe co-culture if it is relevant? Not sure
Yeah,
for
so
these
I
sensory
took that
neurons.
slide out because I didn't think I'd have time, and I get excited. So yes, these are different ways of it further increasing and directing the maturation of the cells. So they've been doing a lot of work on cell culture. The important point to mention here is that they are still viable and functional beyond 4 weeks, which is not the case for some other commercial sensory neurons. Co-culture, obviously, with swan cells and microglia, that's just started. Also looking at media protocols. Anatomic are doing that already, and I showed you that example with P2X3. And finally, there's some groups that have been doing 3D organoids, so DRG organoids. Personally, I'm not a fan of that. I'm not sure what you're going to get out of it, that you wouldn't get out of from single cells, but it is possible to do that. So there are many different ways. And again, this is not all on, sometimes there's an expectation that the provider is going to do all this for you. That I'm speaking independently now. I think the onus should also be on the user. Well, what are your ideas? Well, how about you do some assay development? You go and create some sexy assays, right? And then you share the data. And I think that's where we're at with this at the moment, and trying to generate that enthusiasm rather than a passive, please deliver me an amazing assay, right? Everyone's in this. We're all scientists. We should all be applying our knowledge.
Yeah, same thing with instrumentation, right? Here you have it. Good use of it.
We have a question from Jeff, I think, right? Jeff, online, can you unmute your mic? Yes, you did. Okay, we can hear you.
Yes, I did. Hi, Marc. I was wondering, so on the other additional slide you have shown, there is the success rate, but the percent of expression, is it on your percent of the chip or the percent of the cell you capture, and then what is the success rate of capture? First question.
This, I believe, is based on cells, on the number of cells sampled. Well, viable recordings. So there's an initial QC of whether you've got a decent cell on there or not.
Okay, because I was just wondering if the the capture and going in whole cell process is also like very high, as I'm used to have on the SyncroPatch with heterologous system. The second question is on NAV 1.8. So let's say, not surprising for you, but for some people, NAV 1.8 current shape. Do you think it's because of presence of accessory subunits or something else? What is your idea on this?
To be honest, I have no idea, Jeff. I'm a phenotypic guy. It is what it is. I'm not sure where the differences are. It could simply be, just different kinetics. It's probably more than likely the accessory subunits. I don't think there's going to be a major difference, but there is a talk tomorrow from, what's her first name? Jennifer Deuis, where she's identified a novel accessory subunit or protein that interacts with sodium channels. So there is still stuff to learn about how sodium channels operate. So it may be related to that species difference in subsidiary or accessory subunits.
Okay. Thank you, Marc. Thank you. Bye.
All right. Nice to speak to you again, Jeff. See you, mate.
Bye.
Okay. Thank you again, Marc. Great presentation. And we then move on to the last presentation of this session, which is going to be given by Carlos Vanoye. Carlos is a research associate professor at the Northwestern University in Chicago. And his research projects include interaction between voltage-gated potassium channel, KCNQ1, and the accessory KCNE proteins that modify biophysical properties, surface expression, and pharmacology. In addition, he studies function regulation of the KV potassium channels KCNQ1 and HERG that implicated in cardiac disease, and also is interested in mutations of neuronal sodium channel subunits and their mechanism in epilepsy. And apart from that, Carlos is a real super user of the SyncroPatch. They have the SyncroPatch there since 6-7 years. and have been doing great work with that. So very much looking forward to your presentation. I suppose you're online, Carlos, and you can.
Yeah, I can hear you. Yeah.
Wonderful, great. If you can also turn your, now I can see you, perfect. Then
Good morning, I mean, good afternoon, sorry.
Yeah, that's right. Thanks so much for joining in, and please, you can share your screen and go ahead. Thank you.
Okay, thank you. Can you see my screen?
Not quite yet, but we can nicely see you and your office.
Hey, here we go. Okay. Oops. I skipped to the last slide by accident. So let me get out real quickly. Sorry about that. I don't want to give the punchline before I start.
Fair enough.
Okay. Okay, can you see my screen well now?
No.
Seriously? Man.
No, but not sure if we are doing anything wrong. No, there we go. There's something happening.
You should see a title of my
Yes,
presentation.
yes, now it's fine. Great.
Okay, good. Thank you. Sorry about that. Not enough caffeine in me yet. First of all, thank you very much to Nanion for giving us a chance to present some of our current and latest work, which involves a high throughput analysis of iPS-derived cardiomyocytes. First, I want to acknowledge everybody that was crucial for this work. This work has been done in the Department of Pharmacology in the lab of Al George. And this is a collaboration with Paul Burrish here in the same department. And the work that I'm going to be showing you is the cells, the development of the protocol that we have been using to develop our myocytes is developed by Mallory, a postdoc in Paul's lab. And also I'm going to have some data from Ashley, a graduate student from Paul's lab too. High throughput EP has been successfully been used by us and other groups to do functional analysis of cardiac channel variants. However, now we're very interested in using the same technique to determine the effects of these ion channel variants on not just the currents that we can determine in isolation in an expression system like HEK cells or CHO cells, but what is the effect of these variants on the actual, the integrated response of this mutation that we will be able to discern by any effects on the action potential. So that's our next phase. And our goal, so it's really is, can we do high throughput recording of action potentials, and ion currents from control in patient-derived human iPS cardiomyocytes. And for this, we're using the SyncroPatch 384. And actually, this is a recent photo. Actually, I took it this morning. This is our machine here in the lab. It's currently being upgraded by Matt and Leo. They're currently in the lab, upgrading the system where we can now record.
Carlos, quick comment. Please speak right into the microphone. You sometimes go very low.
Oh, okay.
Thank you.
Can you hear me better now?
Yes, that's better.
Okay.
Close by.
Yeah, so this is the our system, the SyncroPatch 384. It's currently being upgraded here in the lab by Leo and Matt. We're very excited about this because now we will be able to record our action potentials and ionic currents from iPS cardiomyocytes at not just the room temperature, but we'll be able to bring up the temperature to more physiological values. So we're very excited about being able to do that. First, I want to show you how we are generating our cardiomyocytes. This is a work generated by Mallory Blankert at a postdoc in Paul's lab. This is the general schematic of how the cells are being generated. We obtain peripheral blood cells from patients. They're reprogrammed, generated, induced PSLs. Then we do chemical induction and generate cardiomyocytes. So this is the protocol after the chemical induction, differentiation starts. And here is where we're doing the changes. We're modifying this protocol by either changing the type or concentration of the serum present in the media. And this is with the goal of developing the best possible cells that we can record using our high throughput system. And then we're recording anywhere between day 23 to day 31. And the reason we're doing that is we want to be able to nail down what is the best time post-differentiation to get the most complete cardiomyocytes. And by that, I mean, in terms of most complete, do they express the sodium currents, potassium currents, calcium currents, and of course, generate action potentials. So we're trying to optimize the best recording conditions. This next video here actually shows the standard differentiation protocol, two modified differentiation protocols. And here on the standard, you can notice that the cells, you can see beating of the cells, but it's in isolated groups. There are some sections of the cell of the field that you don't see contractions. As we have moved to the modified differentiation protocol, you can see that basically virtually every cell in the field, we can see a very nice global contraction exhibited by all the cells here. And then if we go longer, and this is what I was talking about, if we go longer on the culture mean time, you can see that this particular group of cells that is the same condition as this, but a longer culture time, the cells are contracting so well and so rapidly that it's difficult to get a good video of them, because when they're contracting, they keep coming out of the plane, or the focus plane. So that's why it kind of looks a little bit out of focus, but that's because the cells are contracting so well. So we're trying, we're using the cells at this time to record the best conditions. So first, we wanted to do action potential analysis. This is a screenshot of recording by using the three recording differentiation conditions. Here we have 8 columns for each condition. And by the way, these are recorded at 28 room temperature, so about 22, 23 degrees in the summer in Chicago. It will be much colder now, but hopefully with the temperature control system now we'll be able to always have a constant temperature. These are the action potentials here. This is the selected well for well A4. And this is another differentiation condition and another one. And we can see here the action potentials of group A, group B, and group C. This is the analysis of the action potentials. This is the average of 25 cells. So this is the average action potential of 25 cells, 6 for condition A, for 6 for condition B, and then on 3, which is the worst condition, we were not able to record as many. This is the APD 30s, 70, and 90 that we're recording for each condition, and the change in voltage from baseline to the peak change in the voltage occurs during the action potential. We're also from the same cell, so once we record action, and by the way, I should have cleared that from the beginning. The way we do our analysis, we first record action potentials, and the reason we're doing that is because we're hoping to be able to record spontaneous action potentials. So far, we have not been able to do that. I think only maybe on the first or second attempt that we tried action potential recording, we were able to see spontaneous action potentials. We have not been able to see them since then. We're not sure yet why. Hopefully, by doing it now at warmer temperatures, we should be able to see that. But we do not see spontaneous action potentials, but induced action potentials, we see those all the time, especially with the optimized differentiation conditions. So we do action potentials first, and then we switch from current clamp to voltage clamp, and we record wholesale currents from the same cells. So these are a screenshot of the same group of cells where we're recording sodium currents, and here's the selected wells from each condition, and it's actually the same cells are marked here from the action potential. Now for the sodium current, we do this is the average of all those wholesale currents we were able to obtain that pass all of our criteria that we call a good cell in terms of seals, axis resistance, capacitance, and all of those measurements. These are 74 cells that we are able to record from condition A, 50 from B, and again from the worst condition, only 9 cells. These are the biophysical properties of that sodium current. This is the IV. And then we have the voltage dependence of activation, the voltage dependence of event activation. We see a little bit of difference, but it's not the statistical significant here on the terms of current amplitude. It's just the number of cells that, depending on the differentiation condition, how many cells express the sodium current. And a good validation for our approach that we're not changing the biophysical properties of the cell, we're just changing how many cells are, good for recording, is that when we look at the V1/2 of activation, there, and I apologize for the numbers, they're very small, but you can see that here is -31, -32, and -31 millivolts. So there's no changes in the voltage dependence of activation. And when we look at the voltage dependence of inactivation, -78, -81, and -78 again, there's again no differences. So that we're changing by the differentiation protocol is how many cells express the sodium current. We're not doing anything to the properties of the channel. After recording from sodium currents, we also record from calcium current to attempt to record calcium currents. These are calcium currents recorded in the same cells. So we do is we record in the same cells in the absence of blockers. We just perform a long depolarization protocol to inactivate the sodium channels, and then we change the potential to record the calcium currents. These are the calcium currents on the 3 conditions. These are the individual wells. Here there's showing the wells that were selected. Here is the average of the calcium currents that we were able to record from the 3 conditions, and these currents are normalized to membrane capacitance. This is the condition A, which again, as like we saw for in terms of action potentials and in terms of sodium currents, generates the most cells that are viable or useful for analysis, and then all the way down to condition C that has the least available cells that we can analyze. Excuse me. This is the voltage, the current IV for the IV for the whole cell currents, and the voltage dependence of activation for that sodium, for that calcium current. We noticed that in some cases our calcium currents are very small. Sometimes they're very small to discern well what the properties of these currents are. So what we have done to circumvent that issue is we substitute the external calcium by adding barium. So barium will then give us barium is a better goes through the channel, so we'll get larger currents. We will lose some of the calcium dependent inactivation of the channel, but since we're looking here at wild type channel, we're not concerned about that. I imagine in the future, if we're looking at calcium cells derived from patients that have a mutation in the calcium channel that may affect the inactivation, we may have to be more careful about these experiments. But here are the same cells where if I go back real quickly here, these are the same cells recorded with calcium on the outside in the external solution. So the same cells. Then we substitute the external calcium with barium, and you can see that the currents get much, much larger. So they're much easier to be recorded and to be analyzed. And also to compare, we can see that in this particular condition, condition C, not as many calcium currents are present. So that also shows that maybe the differentiation protocol has an effect on the number of calcium channels present at the plasma membrane, and may explain some of the differences that we see in the number of cells that display measurable action potentials, induced action potentials. This is the average of these whole cell currents. So this is the average of 52 cells, 51 cells, and 9 cells. We have very nice, the kinetics are very nice. They're very clean clean recordings. This is the current voltage relationship for the cells, and also the voltage dependence of activation recorded when barium is the main carrier in the outside for the calcium channel. We also started to do some pharmacology. This is a screenshot of an experiment that we've done with iPS cardiomyocytes exposed to a sodium channel compound of a drug at 0 micromolar, 10 micromolar, and 30 micromolar. And these are the average, not the average, I'm sorry, these are the actual traces from different wells. At 0 micromolar, you can see the length of the action potential, and it's obvious as we increase the concentration when we go to 30 micromolar, And finally, to 30 micromolar, the action potential is getting shorter just by looking at the traces. This is the average of those wells that we were able to record from that met all the criteria, again, seal condition, quality of the capacitance measurements, access resistance, etc. These are 33 cells under control condition, 10 with 10 micromolar and 5 with 30 micromolar. This is the changes that we observe in APDs 30, 70, and 90. And as we increase the amount of drug present, we can see that the action potential is getting shorter. The APD is getting much smaller here, comparing the 30 and the 30 and the 0 micromolar. And also the change in V1 and potential and membrane potential from resting potential to the peak observed during the action potential. It's also severely diminished as we increase the amount of drug. We recorded from the same cells after doing the action potentials. We recorded sodium currents to see if there was any effect on the sodium current. These are the current traces for the cells under 0, 10, and 30 micromolar compound. The currents are here. This is the voltage, the current, the V1, the IV, I'm sorry, the number of cells that we record for each condition, the changes in current amplitude. There's no effect when we look at the voltage dependence of activation. The voltage dependence of activation under all three conditions, 0 and 10 and 30 micromolar, the V1 half has not changed, but we see an effect on the voltage dependence of inactivation, shifting from -72 to -85 as we increase the concentration of the drug. And lastly, these are some cells that are provided by Ashley, the graduate student in Paul's lab. These are cells derived from a longitude patient. We're also able to record sodium currents, calcium currents under controlled condition, and also currents in the presence of external barium. And these are the behavior of these cells recorded, the properties of the sodium and the calcium currents, either in the calcium as the carrier or barium as the carrier. So this is just to give you an idea of the things that we're very excited about being able to do with our system. We're looking forward to now being able to do at higher temperature and see how things compare. So in summary, following some optimization, we have been able to use high throughput EP to successfully analyze action potentials and ionic currents from the same cardiac myocytes. We're also been able to do pharmacology of action potentials and ionic currents from the same cardiomyocytes. And we're also able to record from iPS cells derived from cardiac patients. So we're very excited about all the opportunities and potential experiments that we will be able to do in the future. And currently, we're recording and analyzing iPS cells derived from controlled both male and female subjects that we were able to, we got the blood cells, the blood samples, I'm sorry, iPS cells were generated. Now we are recording and analyzing, and this is with the goal to establish a functional baseline of the behavior of, or the properties, if you would, of the action potential and the ionic currents under control, both female and male cardiomyocytes. And we can use those values for later future assessment of the function of iPS cells derived from patients with several or different clinical phenotypes. Thank you. That's it.
Thank you so much, Carlos. Great presentation and really exciting work. I mean, it's really nice to see this thorough analysis and also the sort of statistical power that you bring to place there. Really neat. Are there any questions that, yeah, Marcos, I pass the mic on.
I want to say hi to Carlos. Hi, Carlos.
Hi.
That's very amazing data. Very nice success rates. Awesome. Really.
Thank you.
I have a question regarding the patients that you want to look at. Would you then also maybe deduce some kind of treatment if you have characterized the patient cells with your approach?
Yes, some of the things that we're, when we're talking about the pharmacology, we're also going to be doing the pharmacology of on the control to get the response of some of these treatments that are currently used to see the effect. And then we want to try the same compounds on the patient's derived cells to see if, what compound is, what is the effect of these compounds on patients, on cells derived from patients. So that's one of the things that we would like to look at, to see if a particular variant in the sodium channel or in the calcium channel or, any of the other cardiac channels that we will be looking at, what's the effect of not just on the isolated ionic current, but what's the effect on the, on the action potential.
Thank you very much.
So, Ellie?
Hi, Carlos. Very nice presentation.
Thank you.
You mentioned that you're now using male and female for male and female patients. Do you see any differences there between male and female cells?
Actually, we haven't finished doing the analysis. We don't have enough, and it's actually right now we're doing from control. So it's not. I used my fault. I used the wrong word. These are not. We're getting from control people, not patients with clinical phenotype, just subjects, control subjects, both male and female. And once we get that data, and we can tell if there's any differences. Want to establish the baseline. What is the baseline of a control, the control data, and then see if there's any differences between both between male and female subjects. And that we are not, we don't have enough data yet to make that conclusion, but
Okay.
we're looking into that.
Thank you.
All right. I think this is it. Carlos, I wish you great luck and good fun going forward, working at elevated temperature and looking at further phenotypes.
It'll be warmer.
Exciting.
It'll be warmer. Yeah.
Thank you so much again. Thanks, Carlos. Thanks, everyone from the session. And we will also do some temperature control now during the break, open the windows, get some oxygen in. And yeah, I think we come back here in about half an hour, starting again at 4 o'clock. Thank you so much.
Okay, so we are going to start again. So anybody out in the coffee area, please come into the room. And anybody at home, please come back to your desk. Okay, good afternoon, everyone, and welcome to the second session of the Nanion User Meeting 2022. A warm welcome to those joining us in Munich on site here, and of course, those also joining us online for the second session entitled Beyond the Norm, Novel Applications of Automated Patch Clamp. Our first speaker in this session is Dr. Helen Waller-Evans from Cardiff University. Helen did her PhD at the University of Oxford before joining Cardiff University as a research associate and now as a neuroscience lecturer at the Medicines Discovery Institute at Cardiff University. Her research focuses on elucidating the functions of lysosomal proteins and their roles in neurological disorders and developing novel drugs that target these proteins for use in both diseases of aging and lysosomal storage disorders. Helen is going to talk to us today about her research in her talk entitled Creating Lysosomal-like Conditions at the Plasma Membrane for Whole Cell Recordings of Lysosomal Channels. Welcome, Helen. Thanks a lot.
for that, Ali. So, Brian, I'd like to say that although I'm giving the talk, then the data I'm going to present is like a collaborative result between the work of my lab and that of Emily Lloyd-Evans, also at Cardiff University. So just to start off and give you a very brief introduction to lysosomes, which are the focus of our research. So lysosomes are very small membrane-bound organelles, so about 0.2 to 0.4 microns in diameter. They're very important, and you can see that by the fact that they're found in all types of cell except for red blood cells. So the main purpose of lysosomes in the cell is in macromolecule recycling. So proteins, carbohydrates, and complex lipids are trafficked into lysosomes, where there are a huge variety of degradative enzymes that chop them up into their constitutive building blocks, which are then transported out to be reused by the cell. And so central to this process is that the lumen of the lysosome is acidic with a pH of about 4.5. It's been found becoming increasingly apparent that lysosomes are not just there to break up macromolecules. They can also act as signaling hubs. So lysosomal calcium signals control the vesicle movements during endocytosis and exocytosis and control fusion and fission events between lysosomes and endosomes. But they can also trigger more global cellular signaling events via lysosomal ER and lysosome mitochondrial contact points. So of interest to most of the audience here today is that the lysosome has several ion channels at various stages of people knowing exactly how they work. So some of the most important of these is the VATPase is a hydrogen transporter which maintains the acidic environment of the lysosomes. Obviously, that's really important functioning of the enzymes. Then there are also calcium release channels. The main calcium release channel lysosome is trypamel-1, but there are also TPC2 and P2X4 and 7 channels. And these channels are the ones which control the movement of vesicles during endocytosis. And then there are other channels and transporters which maintain the ion balance of the lysosome, such as BK channels, and ones which have really not been very well characterized, such as the TMUM175 channel, which you'll be hearing more about tomorrow, which has been reported both as a potassium channel and a hydrogen ion channel. So hopefully, and we'll be able to shed more light on what it actually is. And the lysosome is sort of like the last frontier of new channels. So there are almost certainly more channels and transporters to be discovered. For example, in placental mammals, we still have no idea how calcium gets into lysosomes. So some of the functional consequences of lysosomal ion signaling, I'm going to talk about in the next slide, just to show why we're interested in looking at these channels. So I'm going to focus on trypamel-1, as this is the best characterized of the lysosomal ion channels. So in healthy cells, trypamel-1 signaling is needed after the fusion of lysosomes to late endosomes. Trypamel-1 signaling triggers the separation back into lysosomes and late endosomes. So that is its sort of like constitutive function, which happens all the time in healthy cells. But trypamel-1 also is involved in the responses of cells to stress. So on cellular wounding, so if you make holes in the plasma membrane, this activates the trypamel-1 channel, and this activation of trypamel-1 causes lysosomes to move close to the plasma membrane and fuse with the plasma membrane, which plugs the holes in plasma membrane. So it's sort of like a band-aid for the plasma membrane. And it's also involved in cellular responses to starvation. So trypamel-1 can act as a nutrient sensor. And in starvation conditions, trypamel-1 activates, and the calcium released via trypamel-1 activates calcium urine, which dephosphorylates TFEB, then translocates the nucleus and causes the transcription of autophagy, even related genes, the formation of autophagic vesicles, and So the cell can start using itself as a source of nutrient. So that's the best characterized lysosomal ion channel. There's a lot known about regulation of TRPML1, and TRPML regulation is quite complex. At its most simple thing, TRPML1 is a ligand-gated ion channel. So endogenously, it is activated by the signaling lipid phosphatidylinositol 3,5 bisphosphate, PI3,5P2, and it's inhibited by PI4,5P2. There are also several synthetic activators and inhibitors which have been developed for TRPML1, which have a range of selectivity across TRPML channels and also various other TRP channels. So TRPML1 is also regulated by ions. So you get a lot more conductance through TRPML1 at acidic pH, which makes sense because the lysosome is its native environment, and that's an acidic environment. But it's also regulated by luminal calcium. So at neutral pH, not only do you not get so much signal, but it's actually inhibited by high luminal calcium. And this inhibition is much reduced acidic pH. So it's duly regulated by both hydrogen ion and calcium ion concentration. And then also in its native environment of the lysosome, TRPML1 is inactivated by cleavage, by cathepsin B mediated cleavage, which people think exists to limit the time during which TRPML1 is active in the lysosome. And current via TRPML1 is also reduced if it's phosphorylated by protein kinase A. So if we would like to, if you like, try and develop even further small molecule modulators of TRPML1, we'd like to keep the channel in what is as close to its native environment as possible so that we can think that some of these regulatory factors are still being accounted for. So ideally, we would like to record its activity in the lysosomal membrane. The issue with this is that recording from the native lysosomal membrane is quite difficult. So lysosomes are small. They're too small to patch clamp native lysosomes. So in order to do this, the lysosome cells are treated with a compound called vacuolin, which causes lysosomes to swell to a size at which they can be patch clamped. But even then, they still need to be extracted from the cells first. And the effects of vacuolin on the lysosomal lipid composition and luminal ion balance really have not been characterized at all. So you're not entirely sure whether what you see in vacuolin swollen lysosomes is exactly the same as what you see in native lysosomes. So what we've been doing mostly in Cardiff is using fluorescent calcium dyes based in the cytosol so that we can look at the effects originating from the lysosome. But the conditions that you need to make sure you only see lysosomal calcium signaling are actually quite complex. You get small releases, which mean that none of these approaches are suitable for any sort of higher throughput application. So what if we expressed the channel at the plasma membrane and looked at it there? Obviously, that's much easier and much more amenable to high throughput applications, but you can never be entirely sure looking at that whether your findings are going to be physiologically relevant if you can get your channel to activate at the plasma membrane at all. So we're thinking about what are the differences between the lysosomal membrane and the plasma membrane. So there are differences in lipid composition. So compared to the plasma membrane, the lysosomal limiting membrane has less cholesterol and sphingomyelin, but it does contain the lipids LBPA and PI3,5P2, which are not found in the plasma membrane at all. It's also much lower in PI4,5P2, and the luminal side contains a glycocalyx, which is like a thick sugar coat, which protects the membrane from degradation by the lysosomal enzyme, which is not present at the plasma membrane. In terms of the ion balance between the lumen and the cytosol, obviously the lysosome is acidic, so you have a higher hydrogen ion content. And the absolute concentrations of the ions are different between the two. But other than that, whether the ions are higher or lower between the lumen and the cytosol is pretty similar. But what is different also is the membrane potential, which is more positive in lysosomes than it is at the plasma membrane. And obviously, the two different membranes contain completely different sets of proteins which may interact with and control the activity of the channels. So what we've been trying to do is try and make the plasma membrane as much like the lysosomal membrane as possible. And we've been doing most of this with TRPML1. So we are expressing TRPML1, which contains the variant Baudelaire V432P activating mutation at the plasma membrane. And we know that this one can be activated at physiological pH, but we wanted to see whether there was any sort of pH regulation of the channel. And we find that there is. So if you mildly acidify the extracellular solution, you can make the extracellular solution more like the lysosomal lumen and increase TRPML1 activity. So as you can hopefully see in these bits, in the purple trace, which is at a pH 5.5 outside, this is via FLIPR-based calcium imaging, not by EFIS. We get a higher level of activation, and we also get a leftward shift in the concentration curve compared to neutral pH. And we know that this is mediated by TRPML1 because we can inhibit it using the TRPML1 inhibitor MLSI1. So we then went on to look at this using patch clamping, and we find essentially the same thing. If you try and record TRPML1 mediated current at neutral pH, you see very little. But once you drop the pH to below pH 6, you get activation of TRPML1. And again, we know that this is TRPML1 dependent because we can inhibit it with MLSI1, and we can get a concentration dependent effect, and we get the same sort of membrane potential dependent effect that other people see when patching TRPML1. So we're reasonably happy that acidifying the extracellular solution makes the plasma membrane more like the lysosomal membrane. So as I said at the beginning, that TRPML1 is activated by PI35P2, which is an endosomal specific signaling lipid, which is not present at the plasma membrane. So that we should be able to add this lipid and therefore increase the activation of TRPML1. However, we've only just started doing these experiments, and when you try and add the lipid in your patch clamp, possibly it sticks to the pipette, it forms micelles, and that at the moment is really not working very well by patch clamping. So we need to work on various methods to solubilize lipids to get it in. But what we have looked at is whether reducing the PI45 content of the plasma membrane can also remove some of the inhibitory effects from TRPML1. So we've treated cells with adelphysine, which is a PLC gamma inhibitor, which causes the relocalization of PI45P2 from the plasma membrane to intracellular vesicles, which is what you can see in these pictures here. You see that it's around the outside in the wild type and in the minus adelphysine cells, and then becomes much more clustered in the middle of the cells in the PLC gamma mutant and adelphysine treated cells. So we looked at this using our FLIPR calcium assay, and we do see that there is a slight increase in signal using PI45P2. We asked the question of why are we not seeing an enormously big effect of changing the lipids as we might expect. With addition of the lipids, it's possible that there's just technical difficulties we're currently experiencing with that. It could also be because we're using this activating mutation in the channel, that it might be less affected by phosphatidylnostols than the native channel. It could also be that removal of the PI45P2 in plasma membrane is not sufficient to increase activity, but what you actually really need is to add the PI35P2. But it could also be an effect of the activator we're using. So in these experiments, we've been using MLSA1, which forces the TRPML1 channel into an open conformation. And it might be that this mechanism of action is not dependent on PIPs. Whereas if we use a more subtle activator, we might see a greater effect. So it's recently been shown that some rapalogs can directly activate TRPML1. And this effect is synergistic with PI35P2. So that's in this graph with the blue boxes round. So the left one is the rapalog alone, and the right one is the rapalog with the PI35P2. So you see there's a huge effect of adding the PI35P2, whereas what we've been doing is looking at MLSA1, which is the second from the right in this graph by itself, and then affecting the PIPs. So the level of increase in activation that we see with adelphysine is pretty consistent with what this group found with the MLSA1 and PI35P2. So next on our list of things to do is to try the adelphysine with some of these rapalogs. So the third thing that we can change to make the plasma membrane more like the lysosomal membrane is membrane potential. So at this point, we're moving away from TRPML1 because TRPML1 is already active at plasma membrane potential. But we don't know whether this is necessarily the case for other lysosomal channels. So I'm going to introduce you to a new channel now called CLN3. CLN3 is a lysosomal membrane protein. If you look at the predicted structure, it has a channel-like topology. So you can see it's got a pore through the middle. It's got a nice cluster of transmembrane domains. So this is work done in Emir's lab. We didn't know what sort of channel CLN3 was. So we've used our patch liner to look at whether we can see any current in cells which overexpress CLN3 at the plasma membrane. And we found that if you need to decrease the pH of the extracellular solution down to lysosomal conditions, and then as you make the membrane potential more positive, you get this inwardly rectifying potassium current, which you can see in the red box here. So thinking, okay, so CLN3 is inwardly rectifying lysosomal potassium channel. Obviously, you can't say just from that data that that's entirely what's happening. So a colleague of ours, Sam Pitt, up in the University of St Andrews, has done some single channel recordings with purified CLN3 in lipid bilayers. And she's found that the purified CLN3 is also permeable to potassium ions and less so to sodium ions, and that the permeability to these ions is voltage dependent. So the single channel data and our patch clamping data seem to back each other up, that CLN3 is a voltage gated internally rectifying lysosomal potassium channel. So in conclusions of the studies we've been doing on this so far is that we can adjust pH and membrane potential to make the plasma membrane more like the lysosomal membrane, and so gain more physiologically relevant responses in lysosomal ion channels expressed at the plasma membrane. We're still working on adjusting the lipids, but we think that's a technical difficulty more than anything else. But what we also know is that it will never be possible to fully replicate the lysosomal membrane at the plasma membrane. So you will still need to do some experiments within the lysosomal environment, but we hope that by making these changes at the plasma membrane, then this will make any higher throughput screening studies more physiologically relevant, and so give a higher hit success rate when it comes to doing the native lysosomal experiments. So, I'd like to thank all these people. I'd like to particularly thank the Cardiff University Research Infrastructure Fund, which paid for our patch liner and also has now paid for both the porta patch and the surfer. So we're looking to complete our set of Nanion gear. and to thank the people in the Waller Evans lab and Lloyd Evans lab who completed all this work. Obviously, big thanks to Sam, thanks to our funders, and thank you for your attention.
Thanks very much, Helen. Very nice talk. I think I've just identified your next Nanion instrument. If Connie's in the audience somewhere, I think Helen needs an orbit. Are there any questions? Oh, yeah.
Very interested by this last channel, the potassium channel, CLN3.
Yes.
And I'm wondering whether there's anything known about possible blockers of that channel. For instance,
no small modulation.
So would it be blocked? Is it known whether
So
it's
we
blocked
have,
by
with the
cesium, for instance?
single-channel recordings, we have blocked the current using an anti-CLN3 antibody.
Yeah, but cesium, for instance, would it block it like it does other potassium channels? Is it known?
We haven't tried.
Okay. Or barium, yeah.
Yeah, or barium.
Very nice talk. I didn't know that CLN3 actually can be a channel. Following your work, do you think there might be like a new, there's a chance to develop new drugs for Batten disease now?
Yeah, this is much more Emma's, who's sat in the audience just over there, work than mine. I'm sure he'd be very happy to talk to you about it after the session.
Thank you.
That was fascinating. So what caught my attention was one of your slides with the 3 different ways that the lysosomal membranes can be introduced as the wound healing.
Yeah.
Then you do get lysosomal membrane being put into the plasma membrane, but I
Yes,
imagine that's a very small amount compared to the whole cell membrane.
Yeah. Yes. So the lysosome, in comparison to the volume of the plasma membrane, then obviously once you patch from the lysosome, you can make lysosomal exocytosis, but you still will not. The spread of lysosomal channels then across the whole of the plasma membrane still means there's far too few of them to actually be able to
So
use
just micro
that.
domains.
Yeah. Is something to look at? Yeah. So something which is difficult to control is the amount of lysosomal exocytosis you'll get at any one point, and so the amount of channel present.
Any other questions? No. Okay. Thanks very much, Helen. Okay, moving on now to our second speaker, who I can see is online, thank goodness. Thank you very much for joining us, Francesco. We have Associate Professor Francesco Moccia from the University of Pavia. Francesco obtained his PhD at the University of Turin, and after completing postdoctoral positions at KU Leuven and the University of Naples, he was appointed Professor of Biology and Physiology at the University of Molise. In 2008, he started as assistant professor and is now associate professor of physiology at the University of Pavia. Francesco's primary areas of research interest are in vascular endothelium and calcium signaling, and he was the first one to describe a calcium current activated by NAADP to trigger the embryonic development of echinoderms. Francesco is going to talk to us today about membrane currents and resting membrane potential of the human cerebrovascular endothelial cell line, HCMEM-D3. Okay, thanks, Francesco.
Thank you very much, Ali, for this very, very kind introduction, also for this very kind invitation. I'm just sorry that I cannot attend the meeting in presence, but yet I am very happy to still have the opportunity to present our very preliminary data on the membrane currents and resting membrane potential of the human cerebrovascular endothelial cell line HCMEC3. First of all, I'd like to explain the reason why we are very much interested in studying the mechanism regulating the membrane potential in human cerebrovascular endothelial cells. The reason is related to the mechanism of neuromuscular coupling. Neuromuscular coupling is the mechanism by which an increase in neuronal activity can lead to an elevation in cerebral blood flow. Neurovascular coupling can be initiated either by the synaptic activation in cortical neurons or by the stimulation of cortical neurons which project to the neocortex. Glutamate and acetylcholine represent the two major neurotransmitters responsible for the vascular response to neuronal activity. And indeed, neurovascular coupling is tightly regulated at the level of the so-called neurovascular unit, which encompasses neurons, astrocytes, and of course, vascular cells, including vascular smooth muscle cells and pericytes in respectively arterioles and capillaries, and of course, microvascular endothelial cells. Now, the meeting started with a wonderful lecture by David Beech, who introduced the crucial role played by vascular endothelial cells in the maintenance of cardiovascular homeostasis. And in line with this, a series of recent investigations demonstrated that cerebrovascular endothelial cells express a blend of ionotropic and metabotropic receptors, as well as several trip channel isoforms, which are able to perceive neuronal activity and thereby generate a number of vasorelaxing mediators. For instance, in brain arterioles, endothelial cells express NMDA receptors that can be activated by synaptically released glutamate. Extracellular calcium entry through the endothelial NMDA receptors is able to stimulate the endothelial nitric oxide synthase and to induce the release of nitric oxide, which is one of the most important vasorelaxing mediators in the brain. Moreover, Mark Nelson and co-workers demonstrated that in the mouse somatosensory cortex, capillary endothelial cells express the inward rectifier KIR2.1 potassium channel that can be activated by a modest increase in extracellular potassium concentration, which occurs during neuronal activity. The consequent efflux of potassium is able to induce endothelial hyperpolarization, which is electrotonically propagated to upstream arterioles and thereby causes the vasorelaxation of vascular smooth muscle cells and contracting pericytes and causes an increase in cerebral blood flow to downstream capillaries. This is an example of the inward rectifier potassium current recorded in mouse cerebrovascular endothelial cells. This current presents a strong inward rectification and is inhibited by the extracellular application of barium, which is a blocker of the inward rectifier potassium channels, and is also abolished by the genetic deletion of KIR2.1 channel protein. So this data clearly indicates that the endothelial membrane potential can also be a crucial regulator of cerebral blood flow. An additional mechanism by which the endothelial membrane potential can finally control the production of vasorelaxing mediator is through the modulation of extracellular calcium entry. And indeed, it has long been known that the endothelial calcium response to extracellular agonist, bradykinin example, presents a long-lasting plateau phase, which is due to extracellular calcium entry through store-operated calcium channels. And it has long been known that membrane depolarization that can be achieved, for instance, by replacing extracellular sodium with an equimolar amount of potassium, is able to abolish this calcium-dependent plateau phase by zeroing the membrane potential. And this is very relevant in brain microvasculature. Indeed, it has been demonstrated that neurotransmitters such as glutamate and acetylcholine or neuromodulators such as histamine can generate a long-lasting influx of calcium in cerebrovascular endothelial cells, which is due to the activation of store-operated calcium channels, and this proteophase is able to stimulate the production of nitric oxide by activating the endothelial nitric oxide synthase. From this table, you can appreciate that the average value of the resting membrane potential in vascular endothelial cells is rather depolarized. However, a landmark study carried out by Virgili and co-workers demonstrated that even in endothelial cells presenting a rather depolarized membrane potential, these are calf pulmonary artery endothelial cells, the endothelial membrane potential can scatter from very negative up to very positive values, such as plus 5 millivolts. There are three main conductances which are responsible for maintaining the endothelial membrane potential. In hyperpolarized endothelial cells, the inhibition of the inward rectifier potassium channel with barium causes a rapid and reversible membrane depolarization. On the other hand, in rather depolarized endothelial cells, the removal of extracellular sodium induces a rapid and reversible membrane hyperpolarization. Moreover, it has been demonstrated that vascular endothelial cells can express a volume-sensitive chloride channel that is activated even under isotonic condition. And the inhibition of this volume-sensitive chloride channel by adding 100 millimoles to the extracellular solution is able to induce a rapid and reversible membrane depolarization. What do we know regarding the mechanism regulating the resting membrane potential in human cerebrovascular endothelial cells? Actually, we do not know much, and to try to fill this gap in knowledge, we have recently focused our attention on an immortalized human brain microvascular endothelial cell line that has been developed by Pierre Courreau and that has been termed HCMACD3. As you can see, a growing number of studies over the last 17 years have used HCMACD3 cells to investigate a number of processes that are regulated by human cerebrovascular endothelium. For instance, HCMACD3 cells have been used to study the mechanism regulating the blood-brain barrier or the mechanism regulating neurometabolic and neurovascular coupling. We and others have used HCMACD3 cells to demonstrate that extracellular neurotransmitters such as glutamate and acetylcholine or neuromodulators such as ATP and histamine are able to induce the production of nitric oxide in HCMACD3 cells by inducing a long-lasting energy of potassium to store operated calcium channels. So to try to understand the role of the membrane potential in controlling calciumetry, we had to, of course, to understand the mechanism regulating the resting membrane potential in this cell line. And therefore, we adopted the SyncroPatch system to start these experiments. And our first current clamp measurements demonstrated that in the presence of a physiological potassium-based intracellular solution, HCMACD3 cells present on average a rather depolarized membrane potential. However, we could observe some heterogeneity in the single-cell membrane potential values. When intracellular potassium was replaced with intracellular cesium to block potassium channels, we could observe a small but not significant membrane depolarization. And again, we could observe the scattering of membrane potential that we have observed in the presence of a potassium-based intracellular solution. Interestingly, the inhibition of the inward rectifier with barium did not affect the membrane potential. Conversely, removal of extracellular sodium and reduction of extracellular chloride concentration caused a reversible membrane hyperpolarization. So this data seems to suggest that the inward rectifier does not play a major role in the control of the resting membrane potential in HCMACD3 cells, whereas sodium and chloride conductance seem to play a major role. And indeed, when we switched to voltage clamp, we found that even at very hyperpolarized membrane potentials, such as minus 140 millivolt, we could not detect any evident inward rectifier potassium current. Conversely, HCMACD3 cells displayed a voltage-dependent outward current at positive potential. Normally, this current appeared at membrane potential more positive than minus 30, minus 20 millivolt. To further investigate the ionic mechanism responsible for this outward current, we replaced intracellular potassium with cesium. And again, we found that even in the presence of cesium, the outward current at positive potentials is still present and is even larger as compared to the outward current recorded in the presence of intracellular potassium. So the next step was to reduce the extracellular chloride concentration, and we found that this strategy caused a reversible decrease in the outward current, thereby suggesting that the voltage-dependent outward current in H-symmetric 3 cells is contributed by chloride currents. However, we know from early literature that vascular endothelial cells can express a number of voltage-dependent potassium currents. And indeed, when the outward current was recorded in the presence of 20 millimolar thiachloride to block a number of voltage-dependent potassium currents, the outward current was again inhibited. So this preliminary characterization strongly suggests that the voltage-dependent outward current recorded in H-symmetric 3 cells is contributed by chloride and potassium channels. To further confirm this evidence, we repeated some of these experiments in the presence of a cesium-based intracellular solution, and current Klemper measurements confirm that removal of extracellular sodium and reduction of extracellular chloride concentration caused a reversible membrane hyperpolarization. In the presence of a cesium-based intracellular solution, we investigated the effect of a hypertonic extracellular solution to block the volume-sensitive chloride channels. But as you can see from these measurements, the membrane potential was not affected in the presence of a hypertonic extracellular solution. In agreement with these observations, when we switched back to voltage clamp, we found that the removal of extracellular sodium reduced the inward current, whereas the presence of a hypertonic solution did not affect the ion currents. So this data confirmed that H-symmetric 3 cells express a non-selective cation current, which is responsible for the resting sodium permeability, and express a chloride current, which is not dependent on volume. A feature of non-selective cation currents that have been recorded in both macro and microvascular endothelial cells is their sensitivity to extracellular calcium. And in agreement with previous work, we found that the removal of extracellular calcium caused a dramatic and reversible increase in both the inward and the outward current. In the absence of extracellular calcium, you can see that H-symmetric 3 cells present a non-selective cation current, which presents a linear IV relationship and reverses at quite positive membrane potentials. In our opinion, this feature is very much interesting and deserves future investigation. And the reason is that it has recently been demonstrated that the resting sodium permeability in neurons is mediated by a sodium leak channel that has been termed NAC. And NAC channels present a linear IV relationship. Moreover, this current is increased by the removal of extracellular calcium. Therefore, we are going to use the SyncroPatch system to assess whether a NAC line conductance is responsible for the resting sodium permeability also in H-symmetric 3 cells. Always related to sodium currents, we have then focused our attention in this preliminary characterization on voltage-gated sodium channels. And the reason is that we knew that voltage-gated sodium currents can be recorded in vascular endothelial cells. Voltage-gated sodium currents have been recorded in cardiac microvascular endothelial cells and even in UVEL, which represent the most widely employed endothelial cell line. A feature of voltage-gated sodium currents in endothelial cells is their low sensitivity to TTX. So we moved again to voltage clamp by using a cesium-based intracellular solution, and we had the cells at very negative potential at -120 milliliters to prevent channel inactivation, as it has been described in other types of endothelial cells. And with this protocol, we found that membrane depolarization elicited rapid inward current, which inactivated, which disappeared in the absence of extracellular sodium, and which displayed a bell-shaped IV relationship, which is a feature of voltage-gated sodium currents. And the very recent QRT-PCR assay demonstrated that HC-MECD3 cells express many NAV1 subunits. In particular, they express NAV1.5, NAV1.6, and NAV1.7. And in agreement with the previous data on endothelial cells, these voltage-gated sodium currents were rather resistant to TTX. Only at 3 micromolar, we observed a partial inhibition of these currents. So the conclusion of this preliminary characterization of the biophysical properties of HC-MECD3 cells are that these cells present a resting membrane potential, which is mainly contributed by sodium and chloride conductances. We could not find any evidence yet in favor of KIR 2.1 channel expression, but of course this issue requires a more careful biophysical and molecular evidence. We found that HC-MECD3 cells present voltage-dependent, outwardly rectifying currents that are carried by chloride and by potassium. The chloride current are seemingly volume insensitive, and we think that these currents could be mediated by calcium-dependent chloride channel, and maybe we could discuss about this in a short while. Interestingly, potassium currents are sensitive to TIA, and now we are trying to understand which is their molecular nature. In addition, HC-MECD3 cells present a non-selective cation current that maintains the resting sodium permeability and is sensitive to extracellular calcium, thereby suggesting the involvement of an electrolyte channel. As demonstrated in other types of vascular endothelial cells, HC-MECD3 cells also express voltage-gated sodium channels, which are likely to be largely inactivated at the resting membrane potential of the more depolarized cells. Therefore, their physiological roles and their molecular characterization require a deep investigation. So, as a general conclusion, our future work by using PortaPatch will have to confirm all of this preliminary evidence, and we'll have to assess whether and how these sodium, potassium, and chloride channels contribute to the endothelial decoding of synaptically released neurotransmitters and to the control of neurovascular coupling. In conclusion, I'd like to thank the PhD student of my lab, especially Valentina Brunetti. These experiments were carried out just in 3 months by these 2 students. We did not have any experience with patch clamp, so I am very glad about their work. I really thank Cecilio Serra, who is the research manager of our department. And in particular, I thank, as the previous speaker, the sponsor who allowed us to buy the PortaPatch, which was a grant by Regione Lombardia. And of course, I thank you very much for your attention.
Thank you very much, Francesco. That was very nice. Questions from the audience? Anybody? Maybe I have a question then. Francesco, did you do all of your recordings at room temperature, or do you do anything at physiological temperature?
No, we did all our experiments at the room temperature in our infrastructure. We observed that an increase in temperature beyond 40 degrees with the temperature control of the PortaPatch was able to induce a membrane current that we are also very, very willing to investigate. But all these recordings were carried out at room temperature.
Anybody else? Marcus.
Hello, Francesco. Very nice talk. Thank you. Have you considered also ligand-gated ion channels in the endothelial cells?
Thank you very much for this. Thank you very much for this question. Yes. Well, in the past, by using conventional patch clamping, we demonstrated that these cells express NMDA receptors, although this current is very difficult to be activated. And we published a paper demonstrating that indeed NMDA receptors in HC-MECD3 cells induce calcium release. They work in a metabotropic manner. We also checked for the expression of GABA-A receptors. We have a paper which is going to be submitted soon. We tried to record GABA-A mediated current with the PortaPatch, but we could not detect any current. We repeated this experiment with conventional patch clamp, but again, we could not detect any current. But again, we have this preliminary evidence, which is quite exciting for us, that GABA receptors can induce calcium release. We have also tried 3B1. We tried to activate 3B1 with capsaicin. Actually, again, we were a little bit upset because capsaicin did not induce the activation of any current, but then we found in the literature that another group was unable to find 3B1 mediated ion currents in HCMF3 cells, and the reason is that it is very poorly expressed. So there is the expression level of this protein on the plasma membrane are very, very poor. So it seems that it is not able to mediate any more current. After this preliminary characterization, we would like to check for the expression of other glutamate ionotropic receptors. We would like to check for the expression of AMPA receptors that have been described in mouse cerebrovascular endothelial cells. We would like to see whether they're also expressed in this cell line.
Okay, thanks. Any other questions? Anybody online have any questions? Just raise your hand or write it in the chat if anyone does. No? Okay, then thank you very much, Francesco.
Thank you very much. Thank you very much.
Okay, so next up we have Dr. Edward Humphries from AstraZeneca. Ed is currently a senior scientist at AstraZeneca in Cambridge in the UK. He did his PhD in cellular physiology at the University of Liverpool, where he was investigating cyclic AMP signaling and ion channels. Ed started working then at Metrion in 2017, where he led automated electrophysiology capabilities before moving to AstraZeneca, where he's been instrumental in building a new electrophysiology capability, developing cellular models to support ion channel research in early stage drug discovery. Ed is going to talk to us today about how they've been putting their SyncroPatch through its paces, and it's going to show us the many sides of SyncroPatch beyond traditional screening in drug discovery. Thanks, Ed.
Okay. Will it come up on here? Or? Thank you for the opportunity to talk here today. So yeah, like we introduced, I'm going to talk today about our SyncroPatch usage at AstraZeneca. Some of it's going to be our traditional screening, which many of you might know, but also some of the not so traditional, how we've been pushing the SyncroPatch over the last year or so. So traditional screening. So what do we do and why do we do it? So some of you, and hopefully most of you, will know that ion channels are responsible for action potentials. And especially in AstraZeneca, we do a lot of safety screening when it comes to cardiac ion channels and screening. We primarily look at 4 different channels. So we're looking at the sodium channel, 2 potassium channels, and then also the third, obviously, HERG we look at. And we do that because cardiac safety in AstraZeneca is very driven on ion channel screening, which allows prediction of cardiac risk. And internally, we actually have a QT risk Bayesian model, which allows our IC50 data from our SyncroPatch, along with Cmax data, to predict a QT risk. And it's used on all our drug candidates within the AstraZeneca pipeline. So obviously, to get the risk, we need to know our HERG IC50s. AZ, we run a number of different HERG assays to kind of get the most accurate IC50 we can. But our standard, we call it frontline HERG assay, is a 6-point cumulative assay. For those who don't know what cumulative is, that's where we apply different concentrations to a cell. So we ascend So we start off at low nanomolar and work our way up to extremely high micromolar. We do that because it allows us to get a high throughput and capacity in through the SyncroPatch, and also in a very timely manner as well. So we can do approximately 85 compounds per plate. We're using the Beech cell line, which a lot of people in the industry will be using. Last year, we did over 4,000 IC50s, which based on post-COVID and everything slowed down a tiny bit. We were quite busy there. And a major thing for us, and something that I'll talk a little bit later, is we really do optimize the gene data integration with the SyncroPatch. At AZ, it's quality, but it's also speed. Our DMTA cycle, so design, make, test, and analyze cycle. HERG has included that, so it's metrics. We are time dependent. So anywhere in AstraZeneca in the world, so it could be in Gaithersburg in America, it could be in Gothenburg in Sweden, or it could be Oldley Park or Cambridge. From the time that a chemist requests that IC50, we have less than 10 days to get that IC50 back to the chemist, and that includes shipping. And post-Brexit, some of you may know more about shipping than others, or you might know more about shipping than you ever wanted to know. So speed is of the essence. Also, assay development, assay performance is really crucial to us. I won't go into too much what NDR is, minimum discrepancy ratio, but effectively we are saying if we have a different compound that's within is more than half a log different, it would be a different compound. We are always within 2.5-fold if we retest a compound, which is exactly where we want to be. We want to be producing accurate, reliable, and quick data within AstraZeneca. And you can see for one of our positive controls over the last year, we've never strayed out of that half a log. We have made some improvements. This might make Marcus quite happy in the audience. We used to use the old plates. They were good, not to say they were not good, but they were good. We've also gone to the newer thin glass plates. And this is a user meeting, so I can show you this quite nicely. The thin glass plates for us were magically different, very nice, to the point where we are sorry to do a little exchange as well afterwards. So you can see our seal majorly increases. We get over double our seal, and also interestingly, our current size also increases. These two experiments were done almost the same day on the same cell line that was in culture, same solutions. So for us, this was a really big step forward. And I think what was quite nice for us is our SyncroPatch is 2016. So it's not the newest SyncroPatch, but still, we can still improve our assays going forward now. So we really enjoyed that. And I'm sure some of you in the audience or online have used the thin glass plates. I kind of mentioned the slow onset flag, and there's been best practice and there's been SIPA, and making sure that you get to actual plateau. And we do need to make sure that we provide an accurate IC50 for our risk models. Along with gene data, we've developed a slow onset flag. So it predicts when you're going to reach plateau, and then it'll flag to us in our initial assay if we're not reaching the plateau or close to the plateau. And that now triages us to go into a new assay called extended HERG, lots of flavors of HERG. That doesn't take away from our frontline assay. We've actually done concordance between 78 AZ compounds that went for GLP and our frontline assay, and only 5 out of 78 of those were within a large difference in IC50. But we are seeing our chemistry at the moment is flagging quite a lot with this slow onset, and so we've brought this in. And you can see here that normally we do 3 minutes on our frontline assay, and for these compounds, we've extended it out to single concentration per cell, 15 minutes, and we've seen huge differences in some of the kind of standard slow onset compounds that you'd expect to see, not being great in a cumulative assay. We don't just do HERG. We do 3 other ion channels at the moment, and more are ongoing in line with SIPA. We have IKS, ITO, and NAV 1.5. Again, these are all run on the SyncroPatch. We're running HERG at least once a week, maybe even twice a week, and these are run every 2 to 3 weeks. So we're really pushing our SyncroPatch here. Lots of different assays, lots of different cell lines. But again, you can see over there with the slow onset on the red, that's working out the tau fit for the on rate. And that's really important, especially when it comes to HERG. So some of you might know all of that already. The SyncroPatch at AZ is used for frontline safety screening. It aligns very nicely with our compound management, facilities, so we get assay-ready plates. Then the SyncroPatch will do its work, does it well. So we get efficient screening, which is important. There aren't that many of us in the ion channel department, AZ. We get quality results, as you can see, based on Z primes we use, based on the GLP correlation. And also one thing that us is really important is a reliable machine. Rarely do we have to phone up Ken and say, we need a fix, please. And in some cases, if it's an oncology drug and we go down through ICH S9, this could be the only HERG IC50 we'll actually use for regulatory. It doesn't go via GLP. And therefore, we really do need that accurate quality data to allow our QT prediction. So not so traditional. That was all very based for small molecules or toxins. What about chronic assessment? Some of you in the audience may have heard of PROTACs or ASOs, antisense oligonucleotides. These aren't going to inhibit HERG straight away. Some PROTACs might pharmacologically acutely interact, but generally their mechanism is long-term. We're looking at days, 24, 48 hours for degradation. And you can see a target protein, this wasn't HERG, but you can see you can get a vast array of onsets as well when it comes to PROTACs. It could be 4 hours, it could be 16 hours, who knows? Obviously, if we put this through our standard assay, we're not going to be looking at degradation at all. We're going to be looking at that acute pharmacology. And we know that because the HERG turnover, shock horror, is greater than 30 minutes, which is our longest HERG assay we do. That's been done partly, didn't put it on there, but it's partly by Metrion. They've released some stuff recently with RNA silencing, and that showed greater than 8 hours, but also there's been chase experiments done for a long time now, showing that the HERG turnover is a good few hours. So if we are thinking that we're degrading, trafficking, or any kind of interfering in that mechanism, we're going to need to do a longer assessment. You could do it by imaging, but that was defined in AZ as not being a very good functional readout. We wanted functional HERG assessment chronically. So how do we do this? Functionally assess it. Our current method, how many flasks is too many? We're using, some of you might have seen on the SyncroPatch, where you have your multi-well Teflon. We are doing, T25, T75, lots of treatments for 2 days. That's high plastic usage. AZ wants to be carbon negative by 2025. We've got a high FTE rate. Someone's going to be doing a lot of TC. High cell numbers, scaling up cells, not using assay-ready cells. It's not where we want to be. Where do we want to be? We want to be using a 384 well plate as the cell store. That means that we can dose using our Echo liquid dispenser, and we can incubate the cells for 2 or 3 days in culture, and then we can straight onto the SyncroPatch. That's where we want to be going. And we have had a go. It was quite, a bit different. Basically, cell store doesn't become cell store anymore. If you try moving it too much, you get a crashed robot with a 384 well plate, but you can call cells somewhere else on the plate. So we did that. Initial testing, so was capture. So we seeded our cells at about 5,000 cells per well, and then culture them for a day in the presence of compound or DMSO for this, for example. Capture worked pretty well. You'll have seen in some of the previous assays, we get really good capture, but we do heavily push the cell number for our assays to make sure we get good capture. But from a 384 well plate, it's not swirling. It's pretty much everything an electrophysiologist said we shouldn't do on automated patch clamp. You should have nice swirling suspension. It worked pretty well. So we got over 70% capture on our first test. And since then, it's never been as good because we tried to lower the volume to increase the density of the cells in the plate, and that led to a lot of cell wicking. We're doing this in very standard glass bottomed or tissue culture plates. So going forward, we are going to try and optimize this and try and make sure we can get an accutase treatment, a resuspension, and all of that on the SyncroPatch to allow us to, hopefully in more high throughput as well, do this. It's never just one compound that needs testing. It's always multiple. Moving on to intracellular perfusion. Some compounds, most of the compounds in our routine screening will apply externally. They'll either bind externally or across the membrane. There's some compounds that you need to apply internally. A lot of people do some manual patch clamp, which is, gold standard. It's single cell, but if you apply it internally, so you put it in your patch electrode and then patch and go whole cell, you actually never really have any pre-compound baseline to do a proper normalization to, but it does have limited plastic and no fluoride. Good points. SyncroPatch is 384 well, means I don't have to sit there patching a cell for days and days and days and days. But you also get a pre-exchange baseline. So that's where using the intracellular perfusion, you can do 5 minutes of baseline, then do your intracellular solution exchange during your experiment. So you've got an in-cell normalization, which is great. There is more plastic involvement if you're using the intracellular exchange and you've got compound, you never know about sticking, and you do require fluoride. Tiny little recap in case people didn't know about SyncroPatch and planar patch clamp, but I'm guessing most people do. But what we're doing here is we're exchanging this here, the red intracellular solution during the experiment. So we want to test compounds externally, multiple concentrations. We want to do as much on one plate as we can because it minimalizes our FTE time and our consumables. So on our first test, we actually managed to do a massive multiplex using different externals, different internals. It was really great. So it worked very nicely. We did all of this experiment in 30 minutes. If anyone's ever done internal solutions on a manual patch clamp, I wouldn't know how many days this would have taken us to do. And you always have the question of, has it come down the pipette? Do I need to backfill? Do I want to put positive pressure and hope it's gone in? I remember during the PhD using a CAM kinase peptide inhibitor. Who knew if it got in? We can show it definitely got in here. And we can show that quite nicely here because we didn't even just want to do one concentration. No, we obviously need an IC50. So we had an internal application IC50 of 1.8 micromolar. Externally applied, that was about 80 micromolar. It's exactly what we expected to see based on manual patch clamp, some initial studies. We did multiple voltage protocols as well to align it. And, cost versus manual patch clamp, if we want to outsource this, the FTE usage would have been incredibly high. Whereas for us, it was, again, we did 4 different channels in 2 days. IC50 against more. Fantastic, really great. But we didn't actually just get an IC50 as well. We saw some mechanistic stuff as well. So lidocaine at the top, for example, P1, P25, very standard use dependence. Hopefully everyone knows about that. But you can see here, first on the left, the tonic block, quite minimal, becoming activated with use dependence. P25, you get good block. Then on the SyncroPatch, we had to do a second set of that pulse train. And the inhibition had relieved, and then you re-inhibit. For compound X, that doesn't happen. You do get that use dependence at the beginning, but after doing a few different sweeps sets, you have no use dependence. You've just reached full block, and it's never coming up. And that's really important for certain ion channels. That's all I can say. So, there's a paper. Fluoride in APC. Regulators don't like it. They want us to try and use as close as possible to gold standard manual patch clamp. So I was in touch with Nanion and they said, do you want to give it a go? And I said, yeah, let's give it a go for sure. It was really interesting, but you can do it. Yep, we got gigaseal quality on the SyncroPatch, our 7-year-old SyncroPatch. It was good. We've only done HERG on it at the moment, but it is available. Yeah, it worked really nicely. There are some learnings. Sorry, no. There are a few learnings that I have from this, and Marcus knows them already. Wholesale access was challenging. Compared to going wholesale on our thin glass plates, we found it extremely challenging. We do most things Essen, perforated patch, and even then the Essen concentration we needed to increase. We also saw huge HERG currents. Our average is kind of 1.5 to 2 nanoamps in a 4 times thin glass plate. Sorry, it's not COVID. And they were huge. We use KCl solutions to try and align as possible to standard kind of manual patch clamp. And you can see here On the right-hand side, hERG channel, hERG current, 4 nanoamps, single cell. And look, we run with leak correction. You don't need it. You no longer need it. So yeah, there's a lot of learnings. And I think going forward, this is something that we will start using more for our kind of later stage drugs to try and align it nicer to the regulators. What's next for us? Obviously, there's efficacy projects ongoing, just can't talk about them. We want to assess the pharmacology on CYPA compounds in all our hERG assays, including fluoride-free plates. We're looking at SURFA as well, looking forward to that and trying to see if that technology is amenable to AstraZeneca. And also we want to start using some of our SyncroPatch data to build a Bayesian hERG IC50 prediction. We've got so much good quality SyncroPatch data now. Along with the AI, we think we can really push forward in that area. Just to acknowledge my 2 other Ion Channel colleagues, and then there's all the other AZ colleagues as well, and Mark will be laughing at us. Thank you very much.
Thank you so much, Ed. That was amazing. I think I was going to say, I think you've made Marcus's day and Ken's day today, but I think with that talk, you've probably made the entire SyncroPatch team's day. Does anyone have any questions? I maybe have one then. With your hERG extended time experiments, do you reapply the compound several times or do you just put it on once?
No, we reapply it 3 times. So the first time is like a 2-time stock, and then we have a second plate, which is applied 1 time. We did try 1 times, but we did see a few compounds where maybe it's plastic involvement in some of our chemistry.
Do they then always reach steady state at some point, or do you have ones that just...
There's 1 or 2, even then, don't quite reach steady state. So then they go to either manual patch clamp, or normally by then we've got a pretty good IC50. And if they're going for QT studies, they'll go in vivo QT studies.
Yeah, great talk, really impressive. I was intrigued by the sort of cell culturing in 384 well plate and then echoing them into the module.
Not into the module. Sorry. So we echo into our cells. We plate our cells in a 384, then we'll echo them. Then they'll go into culture for 2 days. And then we take that exact cell culture plate and we now put that straight on the SyncroPatch and use the SyncroPatch to do all our accutase treatments, all our resuspensions, basically automate our cell preparation from a 384 well plate.
I see. Yeah, still intriguing. Have you worked with like frozen stocks at all? Is that something you...
So our frontline assay works from frozen or from fresh. It all depends on...
both.
Yeah, we work from both. It all depends on flexibility. Yeah. But with the chronic, we do find that we need to use the live cells. hERG seems to like to recover for a passage or 2. Yeah.
Fair enough. Thanks.
Oh, of course.
Outstanding, young man.
I know. Talk well.
Some of that looks familiar. No CAV in your SYPA panel, no CAV 1.2. Do you do that elsewhere, or you're not too fussed about liability against the calcium channel?
Binding data is outsourced at the moment. There is an AstraZeneca paper from about 5 years ago looking at binding versus in vivo versus some older SyncroPatch data. The binding, it's a competition with Verapamil binding. Actually, it's Verapamil and diltiazem. They run 2 different. But yes, we have actually a certain Saeed El Hal is currently working up a CAV 1.2 assay. It's looking really good. And then also late sodium is on the list of ones to bring up. Yeah. Once it goes live, it goes live. So it has to be pre-looked at a long time.
Okay. Oh, Sonja has a question.
Great talk. Thank you so much. So I was just wondering, the Bayesian model prediction that you do, are you doing this in collaboration with some of those SIPA colleagues?
So the Bayesian QT is
Yeah.
in-house. So it's based on 150 compounds
Mhm.
that have QT studies from AZ Chemistry. And then the Bayesian model was built in-house, looking at Cmax and Herc IC50s.
Great, cool, okay, thanks.
Okay, thanks very much, Ed. Okay, well, thanks very much to all of our speakers in this session. These were incredibly inspiring talks with many aspects of using APC in scientific research and drug discovery. So thanks to all three speakers. And that does conclude the talks for today. For those here in Munich, we now have some instrument demonstrations down on the ground floor. So please join those to see any of the instruments that you're interested in. If you're not sure where to go, then please ask one of us from Nanion, probably not me, because I'll send you all to my favorite instrument, the patch liner. For SyncroPatch users, we have in this room, we have a SyncroPatch workshop. This can also be joined by the people online. Any SyncroPatch users, just stay in this Zoom meeting or stay in this room if you're in Munich. This is a great opportunity to find out the latest hardware and software features of the SyncroPatch and some tips and tricks. Later on, we are going to have a poster session up here on the ground floor. This is, of course, only for people joining us in Munich. So please, we do encourage you to go around the posters and talk to people about their work. We also have, of course, our famous beer mugs. So grab a mug and grab a beer. And later on, at about 7 o'clock, we're going to be treated to a great meal cooked and organized by Patrick Mum. So please do stick around for that. Talk to us. Let's do some networking. For those that are joining us online and who are not staying for the SyncroPatch workshop, thanks so much for being with us today. I hope you enjoyed the talks. Please join us again tomorrow morning from 9:15, where we're going to hear some more talks about automated patch clamp. Later on, we'll have some membrane biophysics talks involving surfer and orbit instruments, and cell monitoring talks involving the cardiac site 96 instrument. So I hope you join us tomorrow. Bye for now. And oh, and Søren has promised me that he's going to do a dance. So definitely stay for the SyncroPatch workshop.
Yeah, okay. All right. I think we're good to go. Yeah. Yeah, you can hear it? Super. All right. Hello, everybody. I think we are almost, well, we are ready to start this workshop here. We might run into some pauses when we are switching to another computer, but for now, everything seems to be fine. This workshop here is as Ali said, for SyncroPatch development and new features. What I'm going to talk about is more small hardware features that we have made. That are, some of them are already available and will be shipped around to you in the near future, and some are very far from being developed. There is Tom will talk about the Opto module, which is also a new feature that you can acquire already. It's still not a complete product, but it's very close. And then there will be some descriptions of some of the new features that we have in the analysis of the SyncroPatch data. One new thing that we have made over the year here is a device for bleaching your external electrodes. It's a very simple device. It's just a plate where you put your a compound plate with bleach. You place it here on the deck here, and then you simply put your electrode on it. You only use the robot here for filling up the compound plate. The good thing about that is that you don't have to use, it doesn't have to be idling at the same time. For now, normally as you do now, you have your electrodes on the module, and if they're bleached, then that simply takes time. Now you can just do it on the desk. There has also been a few cases where these plates have broken and bleach has spilled into the system. That's also something that you can avoid now. So those we will send around to everybody who has a service plan here when we have enough for that. Another little nifty thing is this holder here for internal solutions. There are two versions, one for Falcon tubes, 50 ml tubes, so that they don't fall around. Usually I have mine stacked on top here on this deck here with a little rack, and that's also kind of dangerous. They can also tilt over and you can spill. There's also one here for the Nanion solutions, the bottles that we provide, that fits those just like a glove here. You can see here from the other side that the liquid sensor will sense the liquid here on the. Oh, you cannot see my mouse. I thought you could see that. Oh, never mind. Well, I don't have a pointer then. Okay, so it will sense the liquid level on the internal solution here. For the Falcon tubes, it will not. That simply we've disabled the liquid sensing there. So since there's basically enough for one internal or one experiment or two. So those are also something that we will send around to people, especially this Falcon tube holder here, and the other ones will come for people who uses the solutions that we provide. So these things, they do already exist. The next thing that will talk about is a device that we have tried to develop for recycling internal solutions. As you know, the internal solution consumption of the SyncroPatch is quite large. It uses between 20 to 50 ml per plate. And some of these can be quite expensive. It depends, of course, on your composition. But if you use, for example, BAPTA or IP3, then it adds up, and it becomes difficult to actually justify the experiments, you can say. So we devised a little, conceptualized at least, a way of doing this by adding an extra. Oh, I can move around now. A little bottle here. So this is a schematic of how it looks now. So you have the waste bottle here, you have the module here, and then what we did was placing a little bottle here in the middle, and then a clever valve here to turn it on and off. And this is how it then ended up looking as a really rough prototype. So just a bottle here, and then you have that little valve there, and the waste is sitting down there. So this is our recycling bottle. And that we tried to see if we could use it by simply doing some simple experiments. So the idea is that you can then turn on the valve to your collector and then simply collect it and reuse it. That's the basic concept. Of course, you need to then switch the valve at the right time and for the right time, the right amount of time. And that was what we first try to determine how long that would be by using some colored solutions to see when it would pass through the tubing that you were looking at and the valve, and also then measure the osmolarity of what the collected solution that you were using. And we found that that was a lot harder than we thought. We couldn't really time it in a way where it actually made sense. So we ended up simply using only one internal solution for the whole experiment. So simply removing the remove chip steps and all the stuff where we were using other liquids than the one we were using. So basically what we had in the system was only the solution that we were using, the internal solution, but we could then recycle it from our recycling bottle. So that was completely clean. So that was a bit of a a hindrance for some use cases. But we went ahead and of course tried it out. So what we did was we tested it with the cell line we had at hand at that moment, which was a CHO cell line expressing KCNQ1. And we simply just ran 3 plates using a double protocol. We were looking at rundown at this time, so we looked at rundown from the 2 different step protocols here, and then looked at the success rate. So we were using 4x plates with first a new fresh internal solution. Then we simply took the bottle and ran it again with the next plate, and then we ran the same bottle 3 times. So we ran the same internal solution 3 times, and we found that the success rate was barely decreased. We started with a 85% success rate and then ended up at 79, sorry. You can see that it turns a little bit less green here in the schematic, but it's actually, since it's a 4x, it's of course depending on where the sliders are. When we looked at the resistances, there is maybe a little bit of a decrease, but they're still above 400 megaohms in average here, and which is very nice for a 4x chip. And the currents are also fairly stable here along the 3 plates. The only thing that I found a little bit concerning was this part down here where we were looking at the catch. It's a little bit left shifted. There are some sites where we don't really catch too much, and I was a little bit concerned with if that would make any difference for fluoride-free, where the catch is quite important. So we did then same experiment here, but with fluoride-free internals using the same approach as is it described in that paper with the internal solutions. What we have here is then the catch resistances. It's histograms. No, this is the seal histograms of the final. Again, the 3 we did again 3 runs, 1, 2, 3. Last one we added a little bit of SN, which I will explain why later. And we saw that the seal distribution was completely similar between them. Maybe a slightly better distribution here in the last one, where we have a bit of a spike here above 100 megaohms, and you can see it a little bit here. And these are the what I do here is these we use these accumulated plots here where we can see how many sites are above 10 megaohm in this case here. Where if you look here, it'll be 300. Above 100, it's what is it here? 250 and so on. So you can see the distribution of the plate in this way. Oh. Yeah, when you draw it like a line, it's easier to see that there is a little bit of a shift here with the third one with SN. We actually have a little bit better seals under those conditions, which is quite interesting. The only thing was that we ran the same solution 3 times, but then with 20 micromolar SN. The reason for that is that, as Ed also mentioned, it's very difficult to form host cells, at least in our hands. When we looked at the currents here, we can see we have a QC criteria here, which is set to half a nanoamp of current. Everything below here, or you can see here, then the green part is then the number of cells that actually has that meets that criteria, and it's not more than 50 cells or so 10% in the first 2 runs. But when we go with SN, we actually end up on a very decent 70% success rate with that expresses current and has a seal of more than 300 megaohm. So we could also then use it simply for fluoride-free experiments, which I was a little bit surprised to see, but the problem that we couldn't really use more than one solution was a little bit of a problem, because you do need sometimes this SN here for your fluoride-free experiments. So we couldn't really do more experiments. We wanted to do some where we could exchange in SN later, but since we only could have one solution, we had to stop a little bit here. But the proof of concept was that we could actually recycle the internal solution. It still needs a lot of refinement. It's not easy to use at all. You have to sit and fiddle with that valve there. It's only if there is a request for recycling these internals that we will continue working on it. But then it can work, but it needs a lot of refinement. And that's it for that. So then Tom is the next. Do you have this?
And then I am already in the meeting. Request. Would like to share my screen. Share screen. Yes. Screen. Share. Okay.
Yeah, it's here now. Oh, yeah.
It's not there.
I need to. I will do that. Maybe here.
Are you sure? Because you can see it here, then why is it not on the beamer then? It might be a different if you move it over. I think it's just two screens. If you take your Zoom window and put it there and make it big.
Yeah.
That's me. Too many mice in this screen.
You have one, I did not have one.
Okay. You can see my screen. I hope everyone online can see my screen as well.
I can.
Otherwise shout, maybe we can hear it. So my name is Tom Götze. I'm part of the SyncroPatch team. And as Søren already said, I would like to introduce some features we recently developed with the latest data control software, which might be helpful for you guys to do some analysis and adjustments with your data that were tedious before. So one new feature I very much like is the external QC. And so what is that actually? It's a new command in the BioMac project since version 2 point something. So we are currently at 2.2. And I think it was already available at 2.1 and 2.0. And so it looks exactly like the regular QC commands, and it works in the same way. So you would just add it into your method. I would suggest to put it pretty much at the front of the method, because then this external QC is available for all the recordings, the rec files, the recorded data later on, done in that method. And it is nothing else but just a placeholder for quality filtering, and you can choose whatever you like. So that works in the way that you can later on during the analysis put in any parameter as a QC parameter, or any online analysis result directly derived from data control. Or you can even have some more complex calculations done in an Excel template or so. So you would export your standard table from data control, paste it into your Excel template, let some calculation be done on those data. And then with the outcome of this calculation, you go back into data control, feed this result back into the data, and then can use it for quality filtering. I will show you an example live. So just to make it obvious that it's quick and easy to use. So all it needs is basically a spreadsheet table with tab delimited columns to be readable by data control. And I always go for the text file format, because that has this tab delimited and everything you need. There is more explanations on this in the new quick start guide quality control of a recording. And that is available on the cloud in the packages of all the QCs. So how does this look? Maybe you can see more than I. Okay, so as I said, in our standard methods from project 2.2 of the BioMac software, we already put it into the standard methods, like here in the section. I think you can see my mouse. It's very small. It's just an instance of a QC. And also here from the graphical user interface, same thing as a normal QC, except for, you can give it a name, but you don't have to give it a scope like in the old days with global or statistics only. I mean, the users among you, you know what this means. And again, same like any other QC, it appears in data control in the QC selection tab and as another instance here in this list. And if it is not used yet, then it is it has this gray font, but it is available for the analysis. So, and now I wanted to quickly shift into data control, like here. Right, this is the new data control. Some of you might have that already on your computer. Otherwise, also for users of an SyncroPatch 384 PE or 384i, I suggest to get the latest data control version, because it's always compatible with old data and it just has more features, has the latest technology basically. So this is a data set we recorded a while ago. It's a mediocre success NAF 1.5 data set, so really everyday quality there. And I now jump into the analysis as normal. Like here. And all I was doing so far in my analysis was activating these two QCs, regular QCs on seal resistance, sometime during the reference period. So we start here with a peak current in reference solutions. That's the gray background. Then the blue background is our compound liquid period, and the dark gray is the full block liquid period. So I was choosing a QC somewhere here, sweep 59, which is by the end of the reference period. And I was filtering everything out that doesn't fulfill 500 mega ohms of seal resistance. And also the current had to be bigger than minus 500 picoamps. So pretty standard. And now I would like to use this external QC, which is over here. Well, I played around with these data already. That's why it's not gray yet. So, but I go through the process as if I hadn't used it yet. So you all are familiar with this last tab where you get your results out in different standard table formats. In this case, it is the online analysis export table, and it just has the well ID. And then in every column, you see the peak current from sweep to sweep over time. And my idea was to use this external QC now for examples like this, for example, here, where we have some rundown. So it would be otherwise hard to filter out wells like this, because you would have to look at the time course of every well one by one and then use the manual QC. And that's, of course, tedious. I mean, almost 400 wells, that just takes too long. So external QC should help here. And I was thinking, I take the sweeps here in, let's say, sweep 13, 14, and 15, take the average out of those, and then I take sweep here, which is 57, 58, 59, again, taking the average. So at the beginning and at the end of my reference period. And I take the ratio out of these two mean values. And then if there is no rundown, then it should be around 1 because both currents are the same. If there's rundown, then it is below 1. Let's say 20% of rundown or even 30 in this case. If there's run up, then the ratio out of these two mean values is of course above 1. So, and that's exactly what I'm doing now. So I go to my results tables. Here I have the peak current over time for all the different sweeps. So I just copy it to the clipboard and then start access. So this is really live. I hope it's not getting too boring watching me working with Excel, but I thought showing you some real life application. So here's my table basically. And what I wanted to do is taking these three sweeps, which is sweep 13, 14, 15. So I highlight them. That makes it easier to see. And then what was it? 57, 58, 59. Let me quickly go there. It's these three here. Okay. And then to make it a little bit easier to follow here, I make those also very narrow. Okay. And now I just create one more column where I do this calculation of the mean of the beginning of the reference period divided, or other way around, the mean of the end of the reference period divided by the mean of the beginning of the reference period. That means I say average of these three values divided by average of these three values. Okay, so here we are, and I give this a name. I call it, let's say, run up, run down or so. Run up, down, just for to label this column, which I later use as an input value for my external QC. And to make it a little bit easier to see these values, so they are clustered around 1. And for example, this one had almost 20% rundown. This had 40% run up. So it's really now a good measure to be used. So next step would be I save this as, let's say, on the desktop. And I'm using, I like to use this text, tab delimited text format. And then give it a name, run, up, down. I might have that already on my desktop. Let's see. Oh yeah, okay, I overwrite that. Good, that's all I need to do now. And so I go back to my QC selection tab, find my external QC and say, and right click that one and say load external QC data from CSV or text file. And then I only need to find my file, which I just generated. Is it this one? Yeah, I just created it. Okay. And it now lets me decide which column do you really want to read now, or the 384 values. And I want to go for my run up down column. Okay. So now those results out of my calculation are the basis of my QC. And now it works like any other QC. Yeah, I already put in my limit values. 80% to 120%, let's say I accept 20% of rundown. So I mean, this can be flexible. We can play with this value. And as soon as I activate this QC here, then we can see which of the, here, for example, this is a good example, which was filtered out only on the basis of my run down, run up calculation. And it's obvious why, because there was, yeah, pretty much quite some run up. Or this one, I think, also failed. And I mean, this is really some nice and specific way without me going through 400 wells to filter out. And this is just one example. People are asking about rundown correction or at least detection of rundown and so on. I think with the external QC, you can easily do that. If I was, for example, saving my Excel sheet here as a template, so I only have to paste my table for the next data set then and even have it automatized, saving it as a text file or so, then it's really quick. But of course, external QC is allows flexible application beyond this. You can do any kind of analysis in your tables, and you know that you can extract pretty much every value into a table. So it's up to the user to define what criterion you want to be then for feedback into data control and as a basis for quality control filtering. Okay.
You can have there's an unlimited amount of those, right?
Yeah. Yeah, I mean, the only thing is you have to integrate it into your method. So there must be one instance of this external QC in your method. Then it's available in data control. And I'm pushing for having this, let's say 3 instances in one of the standard commands, let's say fill ship or so. Just simply have in the subroutine, so it's not really needed that the user remembers having them. So there are always 3 external QC available for all the following rec files, and then they can be used for whatever you want. So in the future, we will work on the quality control concept generally and make retrospective insertion of QCs or something. I don't want to promise too much at this stage, but this is something I wanted to have since years. But I think with the external QC, we at least have full flexibility for whatever. And yeah, it's fun that it works. So things go gray when they fail. It's a situation where you like the gray wells. Okay. I think that was it for the external QC. Any questions on this?
When you're making taking a table from the online analysis, taking it, you must take it from all 380 not just
yeah, that's right. Yeah, but this is an option in the data control table tab here. If this checkbox is not ticked, then you always get the 384. Right. Any other questions? It is because you can or you just have to install the latest data control onto your office computer, because the 2.x version of data control requires a different LabVIEW runtime environment, which is the 2019, also on the cloud. You can find the full package on the cloud. And then you would just uninstall your old data control 197 or whatever that is. And
you cannot use that.
That's right. That's right. because the command is missing. Yeah, that's right. Okay, but there is, I'll advertise Martin's talk, which will cover the fitting of IV curves and usage of fitting results, like let's say the V-half value, and then doing pharmacology on the V-half or so. So that you could use with the latest data control version. It's only necessary that you don't use the data control 2 point something on the machine itself, because if you're running their patch control 1.x, then you need to, but on any office computer, you can use all the features of data control 2.x. Okay, and then there's not really, I mean, we can, we have any more questions on this later on. Now, because I only have 10 minutes, I think that was more than 10 minutes. I wanted to quickly show you some prototype development, which we developed together with Thomas Mager and Alexei from the University of Göttingen. I mean, you saw those data and its application in research. I would just wanted to show you the hardware and how to use it, basically. And because it blinks and it's cool to see. So this is how it looks. This is the actual LED module. If you can see here in the center, there is the PCB board where we have 96 LEDs on, and then there is a fan unit at the cooling unit at the top, because they can become pretty warm. And then in the bottom here you have the connectors to mount it onto your external electrode of the module. You need an extra frame here, which would be usually screwed on. Today I just used double-sided sticky tape, which also works. But yeah, if you have that, then it's a normal external electrode plus this frame. And then you can put on this module. And as it is 96 LEDs at the moment, it covers a quarter of the chip. So you would illuminate the entire chip successively, so one after the other. And here you can see a scheme where the light guides reach into the well of the chip. And the current version, the light guides go up to this height. I mean, the cell sits somewhere here. We're working on another prototype now to bring the light source even closer to the cell. But I think light intensity wise, we already managed to saturate quite a good number of variants of the channel rhodopsins. And the nice thing, these LEDs are very powerful and you can get them in various colors. So in the visual spectrum, there's lots of colors there. And these are even 4 different UV LEDs. I mean, with those, I tend to be a little bit more careful because you don't want to get a sunburn. And they, I mean, 96 of those, they are pretty bright. So these are power LEDs. Okay, together with this hardware, you would get another tab in your Biomax software, and that has 5 more commands which you wouldn't need in a normal application. The first and most important one is the LED opto settings command, where you set your stimulation protocol, which has these 3 parameters: frequency in hertz, and then the pulse width, how long is my flash in milliseconds, so 100 milliseconds, for example, and what is the current size that drives the LED. So in this case, 100 milliamps or so. There is a limitation with all these 3 parameters together to make the LEDs not overheat. So we have a safety measure there that it's not possible to add here, let's say, 10 hertz with for the full second, and then 400 milliamps or so. That would make them become too hot. But yeah, so there's this total safety calculation going on. At the bottom, you have 6 checkboxes because it's a quarter of the chip is 6 columns. And if you, for example, only want to use a limited number of columns, then you can switch these tick boxes off, obviously. Then the next command is LED mode, and that's very easy. You can switch the LEDs on or off, or you can start your stimulation, which then instantly starts the protocol you defined in this command. So at 1 hertz for 100 milliseconds or so. And then there are 2 more options, which are very useful, I think. Stimulation gated and stimulation feed through. That's prototype names at the moment. And what it does, these use the voltage protocol for your recording for starting the stimulation. So, and it is, let's say you start with your holding potential and you finish with your holding potential. Usually you wouldn't change really the voltage. You would keep it at a constant voltage and then just let the channel open and see the current. But you can introduce multiple steps of minus 80 millivolts or so. And every 3rd step or episode would then trigger your stimulation. So in stimulation gated, it would follow this settings here. So basically 1 hertz within, blinking with 1 hertz at the 3rd and at the 6th episode of your voltage protocol. And stimulation feed through is even simpler. So it's again every 3rd, but the length of the episode, it simply switches the LEDs on. So by this, you can even have different lengths here. So here you can blink it as you like, and here you can really have different saturating lengths or something like this. Then if you want to use, for example, the stimulation command, you also have another biomech timer which works in the millisecond range. So it's a more accurate timer like the pause command or that you are familiar with. And then 2 commands just to move the pod away, because obviously you can see it here, and maybe for the online users, once the module is sitting on the external electrode, you don't want the pod to reach into the module and attempt to withdraw liquid or so. So that would crash. But we also have this safety measure. As soon as there's this LED module sitting there, it is sensed by the software, and the pod wouldn't attempt to go there. But yeah, you first want to move it away, move to Z max and somewhere to the right side of the deck, and then you can put it on manually. This is a quick example how you would integrate it into your experiment. And what I was using here is, for example, the work list command. I grew quite a fan of this because a work list is nothing else but a loop. And the number of cycles of the loop is defined. So you need a text file somewhere, which is again tab delimited columns. You simply introduce variables like variable names like current, length, and frequency. And you call, like in any other biomech command, you call a variable by having equal signs, and then you put in the name of the variable, which, yeah, I chose freely by myself. For example, in this already a little bit more sophisticated application, I use these 3 variables, and then in my work list, I can run through multiple cycles of this part here, where it starts with a trigger, where it starts a recording, and then switches on the stimulation protocol, but with different values. For example, I keep the frequency at 1 hertz, but then first I have very short and then a little bit longer pulse widths. And I wanted to change the intensity of the LED light, going from lower to higher currents. So this would generate 6 recordings of my voltage protocol, and then with different illumination parameters. And yeah, so I would trigger the recording, then I switch it on with simply the stimulation command, and then I switch it off and it starts over again. And later on, it would add external solution. There are 2 pauses to remind me, put the module on, and then not to forget to take it off again before the robot comes and perfuses the external solution. So, and this again sits in a loop of 4 because I want to use it in all the 4 quarters of the chip. So, but there's easier applications also available. I just wanted to advertise this work list thing. And here is, as a last slide, where I used something like this with a work list that looked like this. So I started with a frequency of 1 and then 2 Hertz and then 4 Hertz. I kept the length of the light pulse the same, except for the last sweep. And then I increased at these different frequencies. I always increased the current, 100, 200, 400, and so on and so forth. And this is the total recording. I mean, I wanted to look at a fairly long stretch of recording time, so 10 seconds. And I started with a little bit of a delay here because this is my biomac timer in the millisecond range, because I want to see my baseline first. And then the illumination starts, and you can see already that at 1 Hertz, which is the black trace here, there's a little bit of desensitization of the channel rhodopsin until it plateaus out. So recovery from desensitization is stabilizing. And then I zoom in now in the next picture. So this is the same thing. First sweep, 1 Hertz at the low current. So I get these kind of shapes of the channel rhodopsin current. And then I increase the current and even more. And you can see it becomes a little bit steeper. And also this inactivation here becomes a little bit steeper. And it's only 50 milliseconds. So after I switch off the light, current really goes down to 0. And then I increase the frequency to 2 Hertz, low, medium, high light intensity, and then even the 4 Hertz, again, low, medium, high. And then the last sweep was doubling the length of the light flash to 100 milliseconds and then looked like this. So this is just an example. I would like to thank Jeff from Axam, who provided the cells, which were very useful. It's a cell line, hex cell line, expressing channel rhodopsin 2 together with sodium 1.5. So this is a very nice cell line because you can even use it, for example, in current clamp, and then use light to depolarize. So you open the channel rhodopsin, you depolarize by the opening of the channel rhodopsin. At some point you cross the threshold and then the NAF 1.5 starts to shoot. So it starts to open up. We also have done that in current clamp. I just didn't have the time to find these data, but I wanted to. Now I would like to show you a little bit of the light show. Maybe later on when the music is on, we can use it. And for this, I only need to, well, this is the basic version of the controlling this, the LED modules. So this maximum duty cycle is the value which we can set as a safety measure so that they don't turn too hot and so on. These are the 6 columns. And then currently I would let the lights flash or light up every second, 1 Hertz with 10 milliseconds of a pulse length. Let's make it 20 and maybe a little bit brighter here, 20 milliamps of LED current to drive the light. And as soon as I start now here, stimulation on, Then everyone here in the room, sorry for those coming, joining online. You can see that, and you can see that. Maybe like this, more obvious. And then if I increase the frequency to 2, it's becoming faster, and 4 or 10. So then it becomes stroboscopic, yeah, like disco. I'm not sure if Stefan likes me to do that later on, because I mean, it's not hot at all. So it should be fine. Yeah, this is a prototype yet. We try to convince management to make this a proper product. For that, at some point, yeah, we are still collaborating and optimizing things. But I think already now it's very usable, very easy to control it through the Biomax software, where you don't need this little window because you use these commands. And yeah, that's about it. Any questions? By using a different module. So we have these with different PCB boards where different LEDs, yeah, change it, yeah, physically. So I think also there is the idea of having multiple colors feeding into the same light guide. I mean, these are very small LEDs already, so we have to find a concept how to miniature this and make it all compact. But
theoretically, you could do the same thing and have all the LEDs on a board, and then do the whole thing with light guides.
with, yeah, like this, or with a revolver or something. Yeah, light guides, definitely. I think we will end up doing something in that. Yeah. Yeah. Mm-hmm. Yeah, that's a good idea.
You could have a separate cooling unit, and you don't have an issue of electrical noise from the unit.
Mm-hmm. Yeah, we looked into this electrical noise issue. We had some in the first attempts, but then it was just a grounding issue, and we got rid of that. But yeah, that it's exciting, definitely, and we appreciate input from the users in this case, definitely. Okay, then Martin, where are you going? I would hand over after. Okay, then I would hand over to Martin and stop sharing my screen. So bear with me if it takes a little for switching the laptops. Yeah. Sorry, I haven't stopped sharing. Sharing. No.
Okay, you can hear me. I share my screen.
Yes.
Sorry. Okay, hey, it's late now, I think, but I'll try to make it in less than 15 minutes, so you can grab a beer and go for the dinner. Okay, hey, I'm Martin. I'm also application scientist on the SyncroPatch, and yes, I want to show you today the IV analysis in Data Control 2.2. So it's a new feature, or yeah, a new type of analysis we have. So I show already now here a small picture which we then can get. It's a nice shift of inactivation by applying lidocaine in different concentrations. Yeah, so first of all, yeah, to show that feature, I wanted to use a compound which really has an effect on activation or inactivation of a sodium channel. And I found here this paper from Van, where they used lidocaine and mexiletine on the NAV 1.5 channel. And they saw this nice shift in negative direction of inactivation. So I want to go quickly through the method which I used. It's a standard thing. You fill the chip, add the cells, go whole cell, and then the patch experiment looks like this. I first a farm protocol to see nice stable currents, a short pause, then I add again a solution, then I stop the protocol, and then I run a IV. I picked this special IV, where I have first a reference peak, where I can always check, okay, how much channels are available, how much current do I have? Then I have a big pause at the holding potential, where I get all the channels back so that they can recover. Then I have a pulses, where I get the activation peaks, but I leave this pulse also for 750 milliseconds, like they had in the paper, to get the inactivation. And then after that, different pulses, different steps, voltage steps, I again activate at minus 20 millivolt to see how many channels are not yet inactivated. And after that, I run again the NAFAM protocol. So you can see here, it's a append protocol. Can I have a laser pointer? Yeah, so here I don't use the start protocol command, but the append protocol command. It's also a new step which we have in the 2.x or 2.2 version. So this just adds the following sweeps to the recording which was already done. The only thing you need to consider, the protocol must have the same name like you used in the start protocol command before. So here I appended the protocol, the NAFARM one. Then I added here my compounds. You can see on the right side. So I have two controls and two columns of a 12-well Teflon boat. I have two compounds, mexiletine and lidocaine. And these were the final concentrations in my module then. And yes, so I applied the compound during the NAFARM protocol, which was running. Then I stopped the protocol. And then I recorded again this IV protocol to get the inactivation and activation. And on this, I will show you the analysis, IV analysis and data control. So I now switch to that one. So here we go. This is the recording. So you can see now, is it big enough? No, it's. So this one is the NAFARM protocol. It's only one recording, and this is the IV protocol. So what you can see now here, NAFARM, this is now, yes, you can see here in the beginning, this is my reference, which I applied here once. Then here is a big pause. This is the time period where I recorded my IV. And then I appended the protocol to the recording. So the sweeps go here on when I have the application or addition of the compounds. And then I see here the block. You can see here already in the 384, this was the highest concentration of mexilatin. And here in the back, it was the highest concentration of lidocaine. Here I see also the block. So when I go now to, as usual, QC selection, I start with dialogue. So I want to show you how it looks now here. So also for other recordings, which are not with SIV recorded, you can see here in the top, the IV analysis part, but it's grayed out because we don't have here any IV data. But you can do this blocker analysis like you did before. And when you get, I just don't do any further QCing. You can get here the control results and okay. You can see that it's not that nice. For lidocaine, it's already now nice, but mexilatin, I need to do some QCing. Let's quickly open the analyzer file, which I used already. Or I don't have one, okay. But never mind, I want to show you the IV analysis. So I go here into this data set. Already now the view, it's the new thing is here the x-axis. You can now decide if you want to have it as a voltage or as a time option. When you use the time, you can see here, of course, the OA parameters. First in the reference, then here's the pause where I did the append protocol of a farm recording, and here the second IV, which I appended. And I go quickly again into my protocol, which you can see now. Here is the big gap where I recover my channels. I can hide it quickly so that we have a better view. And my OA parameters are here in the first one. It's the peak 1 reference. I named it because it just checks for the current I always get. Then peak 2 activation. This is really the activation current I get. And then peak 3 inact. It's the inactivation current I have. So you can see here is just the reference peak, which of course gets blocked when I have compounds. And when we go for the peak 2 activation, you can see here the IVs from the activation, and peak 3 is the inactivation. And now you of course also can look at them not in the time option as x-axis, but also as the voltage. And you can already here see the blocking effect and also the shifting of the IVs, which is, I will tell now already, which is due to the time. So in general, this recording was made with fluoride in the internal solution, and it is well known that over time the IVs shift to negative potentials. Okay, now let's go through the next steps. Start with dialogue. So now you can see I can now select if I anyway want to do no IV fit analysis, or I have now the option to do IV fitting. So here we named it like it is now here. We have the option for inactivation curves. You have a standard sigmoidal fitting. And for the IVs, where you record your current at different potentials, you select the Boltzmann inclusive conductance. That's the reason, because especially for the sodium channels, then we go again back and look at the activation. You need to take into account this linear part where all the channels are already open, but the driving force is different. So you need to use another fitting formula. So now here, when you go to the analysis, you need to know what you want to analyze, of course. So as we saw in the paper, it was the inactivation, and this is just a standard normal sigmoidal fit. And I want to analyze the V half and slope. This I already select here because depending on what I want to analyze, the VHAF or the Imax, I have here now the option if it makes sense to do a normalization or not. On VHAF, it does not make sense to normalize on a VHAF value, which is a voltage. Actually, if I use the Imin or Imax, I can do also here a blocker analysis of the maximum current I had in the inactivation. Okay, here, so here I have my OA parameters, the inactivation, it was with peak 3, and I select the VHAF value. When I go to the next step, it's the QC selection. Already here, I would just quickly do, for example, the RS QC. So if my R series value is too big and my current, for example, after compound, it's of course smaller, but my current peak reference current, then it must be more than 200 picoamps in watt current. So I get this. So I would start with this QCing. Of course, it can be also made a bit better, but let's see now. Okay, this is my result for the control. This is for all the wells which only got reference. I can go now only into one well to show what data control now can analyze or show in the end. So what you know from the past, from the just concentration response analysis, you always need to have a compound connected to your data so that you get some results. So it's also here. This is marked as a compound, but with concentration 0, and it's just a reference addition. So these points are in the end used now for the analysis. Here in the bottom, you can see again the traces like it was before, but now when you have AV data, you can also look into OA values and the IV fit. So now here from well A1, these were the OA data, the inactivation. When you mouse over the fit curve, you can get the data. I don't know if it's visible. Not nicely, right? But you can get the R square value, then the Imax, Imin, the slope, and the VHAF values all already here displayed. This you can get for every single well. When you click on the group, you will get here in the top, because we selected we want to check for VHAF, you get the mean VHAF value with error bars. But of course you can, as in the past, you can now select also the pooled option. So now you get for all the wells which are in this group, you get the single VHAF results. Now let's go to the group maxillitin. Now it's the same. You get for every well one VHAF result, but now of course we have different concentrations of this compound. So it's, yeah, depending on that, it's on a different position on the y-axis, on the x-axis. Then we go for the averaged view. So now we can get the average VHAF values for all the compound additions, and you can see here nicely that it's shifted to negative values. Now for the group, we have here in the bottom, we have now also the traces option as before, but now you have further on, otherwise you will get too many traces and you can't distinguish between the different concentrations. You have to select here which group is interesting for you. You can mouse or you can select one concentration. And now when you go here on the right, so I picked here now the highest concentration. And when you now mouse here over in the OA graph, you will get for every point, you will get highlighted the trace below. Now for our IV, here we get further information. So here, for example, we have some info if the IV fit was, let's say, good. We have many or a few criteria. For example, if a VHAF result is in the range of the x-axis, if it's If it's more negative or more positive, it doesn't make sense. So it will tell you here, okay, something is wrong. Then you can check every single well, or you do like Tom already has shown external QC, for example, on this value. The other thing is, for example, also R square. If this is below 0.95, it will also show this information. And yeah, it's hints like this we have here. But what is shown, maybe here it's okay. Of course, the higher the concentration is, the more current is blocked, and the more complicated it is also to make a fit when you have really low current. So I will show you another example here with a lower concentration. You will get here this view. So we selected here average. What gives us here a fit curve, which is generated out of a averaged, no, out of all the fit parameters you got from all wells. And what we now decided to show is also here the well which had the maximum VHAF and also the well which had the minimal VHAF. This, of course, now doesn't look very nice because it seems it's really spread, but it's really the maximum and minimum. Maybe in future it would be nice to have some quantile or I don't know, but now it's like that. If you go for the pooled, you will get now here the black curve here is only one well, and it's the well with a median VHAF value. And like this, it is for every concentration. But what you can also select is here all LPs. That means all liquid periods. So now you get for all the concentrations, the median well, the well with the median VHAF value. Maybe better, it is look at the averaged. So now you get really for every concentration, it's the fit curve which is generated out of all the parameters from one concentration you got. And now you have also here for display the normalization options. So you can normalize against the reference period. So it's really now normalized against the maximum current from the reference period. Or you can normalize with a liquid period, what does maybe make more sense because you want to compare really the different concentrations of the compounds. And now you can get here, yeah, see the shift of inactivation. As we had in the past, the option here in the CRC graph to do a fit to get the IC50, it's still in there, the option. So in the settings, I selected here just raw values. It's also for compound analysis, you have it there. And if I select now the Hill equation, it tries to fit these values. I'm sorry, so it's not possible for the software now to do here a fit through that. Let's see if my ANA file, which I used, I can't. Yeah, I'm sorry, I can't find it now. Yeah, so for example, I could fix it. In the bottom, or what I like to do is I use the control as really the control, let's say. So I would go into the compound file and now say my control is maxillitin with concentration 0. Maxillitin was number 3. Number 3. Why can't I apply it now? Interesting. I know what's the problem. Yeah, I think I need first to generate an analyzer file, right? Maybe I discard and okay, here I have it. Let's see if it's now possible. Analysis settings, and I select here now the Hill equation. And now here it's now possible to do a fit through that, and it will deliver you a IC50 value, what tells you, okay, with this concentration, you get the half maximal shift in V half. I don't know if you need it or not. Normally you just calculate the difference in regard to the control. So now I try again to go into the compound file. I hope it works now. And yes, it looks fine. Apply. And now we have here maxillotine, and it added here the control value. So what is now possible here, you can easily, per right click, you can open the context menu and copy data to clipboard, for example, and then you paste it to Excel or wherever, and you can simply just calculate the difference between the V half values which you have here, and you will get the shift in activation. Here are already some questions. Yes.
Just one question about all the technologies that you get to put your mouse on the different that like those ones. Can you display them in the results table?
Yes, you can export for every well, every fitting parameter. Yes, so I would show you also the analysis of activation. Let's go into this. I again start with a dialog. So I want to do the activation analysis. It's the activation here, and I select the raw values. And yeah, QC selection. I would load the ones which I already used. So, all right, don't do that. Sorry. So let's go here, and now we get the IV fit for the activation. So here another fitting formula is used, which delivers you an additional parameter, which is the reversal potential. This is of course needed to fit that, and you get also the G maximum and G minimum. Yeah. You can as well here. It's the same. You will get for every compound concentration, you will get here these results, and you can also show for every concentration the mean IV curve. And of course, as it is the activation, and we fitted the curve and calculated out of that the conductance, you can display also the activation as a conductance curve. So calculate it back into a real sigmoidal curve. And when you normalize it here within the liquid period, you can also check if there is a shift in the IV activation. But you can see here, or it seems so that there is no shift with maxillotine. Interestingly, okay, for lidocaine, there's also no shift visible, but you can see I did not proper QCing. So here is one well. It's slightly off. It's really off. It has no appropriate results. Maybe this also. And you can here, by pressing Control and mousing over it, you can also disregard this result. And then you, now it looks a little bit better than taking out these two wells. And yeah, here it seems that maybe the highest concentration of lidocaine does some shifting of the IV. Yeah. I think it's 10 before dinner and you are all hungry. This is the main feature of the IV analysis setting. We have there also a really big document It's called Quick Start Guide, but it's more or less a long start guide. But yes, so if you have questions here regarding IV analysis, SyncroPatch support, email, you know it. Okay, if there are no further questions, thank you.
Yeah.
Okay, what do we say to the people outside there? Thank you for your attention and see you tomorrow.
All right, it's time to start, everybody. So welcome to the second day of the Nanion user meeting. And the first session this morning is looking at ion channel modulators from the natural world. And we've got two speakers this morning from Australia, where there is lots of natural products that are from very scary animals. And the first speaker today is Jennifer Deuis, and she's a postdoctoral researcher in our own Vetter's lab at the Institute of Molecular Biosciences in the University of Queensland. So she's been looking at ion channel modulating toxins from spiders, scorpions, ants, and plants, and probably some other species as well. So today she's going to be talking about a new venom that's from a stinging nettle tree, and possibly working through a novel and unknown accessory protein for the sodium channel. So Jennifer, are you there with us?
I am. Can you hear me?
Good day. Yes, we can hear you.
All right, I'll share my screen. Can you see the right view?
Yeah, Jennifer, we can see it. Thank
All right. All right, so thank you for that introduction. So yeah, Australia doesn't only have venomous animals, but apparently we've also got venomous plants. And I'll be talking to you today about one species that we've been studying. So we've been looking at stinging nettles, and these are a large family of plants that cause pain when you come in contact with them. One that most of you may be more familiar with is the common stinging nettle. That is native to Europe, but it can now nearly be found worldwide. But the two species that we've been studying lately are the species that are from Australia, which is commonly known as Gympie gympie, and a species from New Zealand that's commonly known as Unga unga. But for today's presentation, I'll just be focusing on our work from the Australian stinging tree known as Gympie gympie. So how do stinging nettles cause pain? The leaves and stems are covered with trichomes, which may look like harmless hairs, but they are in fact hollow needle-like structures that inject a bioactive fluid, which we would argue is a venom, upon contact with skin. This sting elicits pain that can be both very severe and very long-lasting. Historically, it has been thought that this pain is due to the injection of small molecule neurotransmitters, such as acetylcholine, serotonin, or histamine. However, these, when they are injected, are unable to fully explain the symptoms that's experienced after a sting. So we've hypothesized that there must be some other bioactive molecule responsible for the pain. So there's two species of Australian stinging nettles that we've looked at. One is the giant Australian stinging tree, which can grow up to 40 meters high. And that's found in northern New South Wales and southeast Queensland. And another is called the Dendronide moroides, which is the mulberry leaf stinger. And that one's found in far northern Queensland. And this one's much smaller, so it's more like a shrub or a bush. So if you have a look online, there's lots of interesting anecdotal reports from people who have been stung by Gympie. This one is one of my favorites. So they describe it as being stung is the worst kind of pain that you can imagine. It's like being burnt with hot acid and being electrocuted at the same time. And the pain from these plants has prompted local councils to put up warning signs in rainforest tracts where the plants can be found to warn visitors to avoid contact with them. So what happens when you get stung by Gympie? Immediately upon contact with the needle-like structures, you get pricking and stinging pain, as well as itching and piloerection. After a few minutes, you get rapidly developing wheals. More slowly, so after about an hour, you get a axon reflex flare and axon reflex sweating. And you also get local vasodilation. And then over hours to often days, and some people have told us that it can happen weeks or even months after being stung, you get waves which can be more or less painful and can be described again as pricking or burning, crawling or shooting. So it was because of this intensity, as well as this long duration, that we were really interested in studying the venom of the Australian Gympie stinging tree. So what causes the pain? To work this out, we fractionated the stinging hair extract by HPLC, which is shown in the black trace there. And we injected each of these fractions into the hind paw of mice, and we assessed spontaneous pain behaviors in the hind paw. So for example, licking or flinching of that hind paw. And that's shown by the red graph there. And you can see that there was just one single late-eluting fraction that caused nociceptive or painful responses in mice. And MALDI-MS showed that this contained 4 kilodalton molecules, which we hypothesized would most likely be peptides. And indeed, when we did triptych digest followed by MSMS and transcriptomic analysis, we identified a family of 36 amino acid peptides with 3 disulfide bonds, which we've called the Gympie-tides. We found 3 closely related sequences from Dendronite excelsa, which was the giant Australian stinging tree, and 1 from Dendronite moroides, which was that shrub-sized stinging nettle. And these do not have a very high similarity with any other known peptide sequences. The most closely related are albumin-derived plant peptides, and the cysteine spacing is similar to animal-derived cysteine knot venom peptides. And when we had a look at the 3D structure, we found that indeed they do show an inhibitor cysteine knot structure, also known as ICK or knottin. And this is a very stable fold that's similar to neurotoxins found in venomous animals such as spiders and cone snails. So to confirm that we had indeed isolated the pain-causing component, we synthetically made these Gympie-tides and tested them again in the hind paw of mice, and they caused a concentration-dependent pain behaviors. And they also cause local vasodilation as quantified by an increase in paw temperature. And this is very similar to the symptoms in humans following sting with the whole crude venom. So we are pretty confident that we've isolated the main pain-causing component from the venom. So we next wanted to assess what the pharmacology was of these Gympie-tides. So when applied to dissociated dorsal root ganglion neurons from mice, we get an increase in calcium influx, which shows that they directly activate neurons. This partially overlaps with neurons that respond to capsaicin, so they express TRPV1. So presumably they are acting on neurons that are nociceptive or pain sensing. They don't act on non-neuronal cells, which is suggesting that they are selective for an ion channel or receptor that is expressed on neurons. So we then wanted to assess what they did to sodium currents in whole cell DRG recordings. So excelzotoxin inhibits inactivation of the voltage-gated inward current. So the control there is in black, and the green is after exposure to excelzotoxin, and it causes a significant increase in persistent current. And we quantify this by the late current divided by the peak current. And this persistent current is abolished when you replace the external sodium with choline, and it's also inhibited by TDX. So this suggests that the gimpetides are acting on a TDX sensitive voltage-gated sodium channel, which is most likely going to be NaV 1.7 given the expression in DRG neurons. So to confirm this, we tested excelzotoxin on endogenous NaV 1.7 current in TE671 cells, which are a neuroblastoma cell line. And they endogenously express NaV 1.7 as the main sodium current, and the effect of excelzotoxin could be blocked by the NaV1.7 selective inhibitor PN3A. In addition, excelzotoxin-induced pain behaviors are also abolished in NaV 1.7 knockout mice. So we're pretty confident that the gimpetides are able to act on NaV 1.7. What was surprising to us, though, is that when we tested excelzotoxin on NaV 1.7, which is heterologously expressed in HEC293 cells, it had no effect. And this is that trace here shown in HEC cells. We also had the same result when NaV 1.7 was expressed in CHO cells and oocytes, but haven't shown that here. So this was quite unexpected for us. We also tested it on NaV 1.1 to NaV 1.8 in HEC cells, and again, it had no effect. And we also tested it on some other neuronal cell lines that endogenously express NaV 1.7, so SHSY5Ys, F11s, and ND7s, and again, it had no effect. So this led us to believe that there's some differences in the cell background, some factor that is present in neuronal cells, or some neuronal cell lines, but not in HEC cells, that's needed for the toxin to have its pharmacological effect. So to try and find what this missing factor is, or this target, we used a genome-wide lenti-CRISPR screen in TE671 cells where the toxin had an effect. So what we did is we had TE671 cells, we infected them with the lenti-CRISPR library. We selected cells with puromycin, and then we either had control cells or exposed the cells to excelzotoxin, and we assessed cell death, and then we sequenced cells that have been that had survived, and to see which genes are enriched in cells that had resistance to excelzotoxin-induced death. And this is the results of that screen. So we had 3 genes which actually conferred toxin sensitivity, and we had 11 genes that conferred toxin resistance. What gave us confidence in the assay is removing SCN9A, which is the gene for NaV 1.7, conferred toxin resistance. So we're pretty confident that the assay, the screen has worked, identifying the genes that we're interested in. And it also identified some genes that are already known to have a role in sodium channel surface expression and trafficking, including NED4L and RNF121. So what we did next was we overexpressed the 11 genes that conferred toxin resistant in HEK cells with NAV1.7 to see if any could then, co-expression with any of these would then cause them to be sensitive to the pharmacological effect of excelsa toxin. And that's the results here. This is a membrane potential assay. And when we overexpressed all of those genes with NAV1.7 in HEK cells, only co-expression with a gene called TMEM233 was able to cause the excelsa toxin to have an effect on membrane potential. And then we also confirmed that with patch clamp in HEK cells expressing NAV1.7 with NAV1.7 alone, the toxin has absolutely no effect. But when you co-express NAV1.7 with TMEM233, you get this really remarkable effect on persistent current. It basically is inhibiting fast inactivation of NAV1.7. So what is TMEM233? It's a transmembrane protein. It's related, it's part of a dyspanin family. It's only small, it's 109 amino acids. And this is the alpha fold structure here. And it's predicted to have one transmembrane domain and one intramembrane domain. And so far, there's been no reports on what its function is. So we next wanted to know whether excelsa toxin is binding to NAV1.7 and that TMEM is just required for its effect, or whether it's binding to TMEM. So we looked at flow cytometry with a biotin labeled excelsa toxin. In HEK cells, there's no non-specific binding. When you look at HEK cells expressing TMEM233, you get a binding of excelsa toxin. You get nothing when NAV1.7 is expressed alone, and you also get binding when you express NAV1.7 with TMEM233. So this suggests that the toxin is directly binding to TMEM233 and not NAV1.7. So in summary, the GIMPides are a new class of plant-derived neurotoxic peptides. They impair the inactivation of voltage-gated sodium channels to cause pain, and we have not been able to find any sequences related to these in any of the other stinging nettles so far, so it appears to be unique to the Australian nettle branch. Excelsa toxin binds directly to TMEM233 to modulate the function of NAV1.7, and we're still investigating exactly how it's doing this. And the GIMPides are a new class of novel dyspanin targeting peptides, and hopefully they'll be useful tools for us to understand the function of TMEM233 and NAV1.7. So that's it. Thank you.
Great, thanks a lot, Jennifer. That was fascinating. So I'll open up the floor to questions. No? Oh, okay, Sonja.
Very nice talk, Jennifer. Thank you so much. Great study. I was wondering the GIMPitide inhibition effect on the sodium channel, is that reversible? Like, can you wash out?
No, it's not reversible. So we've tried washing it out over half an hour and it doesn't come off at all. So we're assuming that explains why when you're stung by the gimpy that you've got the long-lasting effects of lasting, days.
Yeah, this was my guess. This would have been my second question then, if this is correlated maybe. Great. Thank you.
Anybody else? Man from Alamone? No, nothing? Right, I do have a few questions and we do have a little bit of time. So I'm intrigued by, with toxins, especially with co-evolution. So the fact that you've got this novel protein, accessory protein that seems to be present in vertebrates. I guess you've looked at mouse, rat, and human. how does that co-evolve with the plant? I mean, that single protein presumably is expressed in those species.
Yeah, so it's expressed in mammals. We are not sure yet how it has evolved, because it's like lots of venoms have evolved to directly target sodium channels. So, you know, spiders, scorpions, cone snails, they all directly target sodium channels. This is the first venom that we've found that indirectly targets sodium channels. So we're not sure whether it evolved to do that initially, or whether it had some other functions such as being insecticidal. But we haven't found any insecticidal activity with this toxin. So that's something we're still investigating as to why it evolved to indirectly target sodium channels by targeting this transmembrane protein.
Yeah, it's a really cool story. So what about turning that around? Do any of your other ICK animal-derived peptides bind to TMEM233?
Not that we haven't actually tested anything directly on TMEM233, but all of our other peptides that we've isolated always act, you know, on sodium channels in hex cells. They don't require TMEM233 to be there, so we haven't found anything else that bind directly to it. Whether TMEM233 alters the pharmacology of other peptides, that's possible. That's something that we're going to assess.
Okay, and what's special about this TE671? Is that a human? That was like a rhabdosarcoma cell line, I think.
Yeah, the original reason that we chose it was because when we injected the toxin in mice, we did notice some muscle fasciculations. So we originally thought it may be acting on a target in muscle. So that's why we ended up using them. But then we found out they're actually more neuronal than a rhabdosarcoma cell line. So it's kind of a bit of a by chance that we found that they worked in the TE671s, and they proved to be a really useful tool for us to be able to find the target, because I don't think we would have been able to find it otherwise.
Yeah, you could have really missed that. So you showed the delayed inactivation, but I don't recall you showing any biophysical curves. So is it specific for shifting the activation of the NaV1.7, or does it also have effects on activation or other biophysical parameters?
Yeah, so yeah, I didn't show that, but yeah, it affects other parameters as well. So when you express, when you look at the effect of the toxin with NaV1.7 and TMEM, you also get a shift in the voltage dependence of activation and a hyperpolarizing shift. And you also get shifts in the voltage dependence of steady state fast inactivation. And it also affects recovery from fast inactivation. So it affects a lot of parameters.
That sounds pretty complicated. So this is close to publication, and we're going to be able to see this data in the public domain soon? No pressure.
Hopefully, close to submission.
Excellent. All right, thanks very much. It's been a pleasure to e-meet you. I'm a big fan of the toxin work that you guys are doing at UQ. So thank you very much for your presentation today.
Thank you for the invitation.
Right, I have no control over the technology, so I'm really hoping this is going to work. Oh, okay. So next talk is from Carol Milligan. So is she live at all, or are we just going to play? Hey, Carol, come on now, don't be shy.
Hang on one sec. Yeah, I am live, but I'm quite happy for you to play my recorded presentation. I'm very happy to be here. Thank you very much for the invitation.
Great. It's nice to see you again. It's been a long time. So
It has been forever.
yeah. So Carol's trained at the University of Leeds in pharmacology, and as we heard from David Beech yesterday, they were a very early adopter of the patch liner. So not only was she an early adopter, I think she maybe was one of the first users of the patch liner in anger, as we like to say. And she's also the lead author of a seminal Nature Protocols paper in 2009, which really gave a lot of people an introduction to the capabilities and the flexibility of automated patch clamp. So this is a very important collection paper in the collection of APC papers. So Carol immigrated to Australia in 2012, and she's now a senior research officer in the Melbourne Brain Centre, where she works in the Florey Institute as the ion channel analysis facility. So today she's going to be talking about natural products, specifically natural products from the cannabis plant and the potential for additional effects of other components less well known in the cannabis sativa. So please play the video.
Good morning, everybody. Thank you to Nanion and the team for the very kind invitation to come and talk to you today. Unfortunately, I can't be there, which is a shame because I know how beautiful Munich is this time of year, and it's been an age since I've seen anybody from the Nanion team. Anyway, such is life. So today I'm going to talk to you about this nutraceutical product, which is extracted from cannabis sativa. And so we know that the product modulates voltage-gated sodium channels, and that's what I'm going to talk to you about today. So with this project, we set out to determine the mechanism of action of a set of the 10 less well characterized phytocannabinoids. And also as part of the project, we examined the effects of this product. It's commercially available. Anecdotally, it's been reported to reduce seizure frequency in patients suffering with Dravet syndrome, which is a very debilitating epilepsy. So today I'm going to share what we found out about this product. Let me just give you a little bit of background. Medicinal use of cannabis in the treatment of human disorders extends back nearly 5 millennia. Cannabis therapy specifically for epilepsy was recorded as far back as 4,000 years ago. So what we know today, we know through clinical trials, preclinical trials, case reports, and anecdotal accounts from epilepsy patients, or indeed their parents, because a lot of these patients are children, that cannabidiol, the major non-psychoactive phytocannabinoid in cannabis, exhibits therapeutic potential for intractable epilepsy. So those are the epilepsies that don't respond to the regular anti-epileptic drugs. Despite this promise, there's only scant information available concerning how these phytocannabinoids affect neuronal networks. So research shows that CBD acts on multiple molecular targets, and I've summarized them here in this schematic. So these are very broad. However, despite the research, there's very little known about how the anticonvulsant properties of phytocannabinoids come about. It's still really in its infancy. So I'm particularly interested in the role of ion channels in epilepsy. So today I'm going to focus on this voltage-gated sodium channels. So in the primary sodium channel isoforms of the central nervous system, both gain of function and loss of function mutations cause epilepsy. And these can range from relatively mild epilepsies right through to the really severe syndromes such as Dravet syndrome and Lennox-Gastaut syndrome. And this piece of work that came out of our lab 2 or 3 years ago illustrates very nicely how mutations in the SCN1A gene can affect the biophysical properties of the NaV1.1 sodium channel. The mutations in red in the domain 1 causes a gain of function for the channel, and this is indicated very nicely by a hyperpolarizing shift in the voltage dependence of activation, which is also accompanied by an enhancement of the action potential firing that you can see over here in the right-hand panel. However, the mutation in blue in domain 3 causes a loss of function, and this is indicated very nicely by the rightward shift, the depolarizing shift in the voltage dependence of activation. And this is consistent with loss of channel function, and you can see when over in the right-hand panel, this channel in blue does not fire action potentials. So it's interesting to see how mutations in a single gene can cause either loss of function or gain of function, two completely different phenotypes. So this work was funded by the Lambert Initiative over in Sydney. The overall aim was to evaluate 10 different phytocannabinoids as potential targeted therapy. So there were 4 phases to the project, and we used 3 different technologies. In phase 1, we wanted to determine the mechanism of action of these 10 phytocannabinoids against sodium, calcium and potassium channels expressed stably in different cell lines using the patch liner. In the second phase, we were looking at cultured primary neurons from a mouse model of Dravet syndrome. So this is an SCN1A mutated mouse. And we used multi-electrode array to examine the excitability of these primary neurons. Phase 3, we're looking at using manual patch clamp and brain slices from the same mouse model. And in the final step, we managed to get hold of patient-derived pluripotent stem cells from a patient with Dravet syndrome. And I'm going to tell you today about the broad spectrum CBD-rich extract and recordings I made using the patch liner. So you can see that from the table at the bottom that the product contains hemp seed oil, 4 phytocannabinoids and 4 terpenes. So it's quite a mixed compound, but the highest concentration with the phytocannabinoids is CBD. This is an unregulated compound. So we evaluated the compound using sodium channels either transiently or stably expressed in cell lines, and we looked at the whole family, NAV 1.1 through to NAV 1.8. We examined the effects using the patch liner, and this figure comes out of the Beech lab paper from 2009. And we looked at the biophysical properties of activation, inactivation, recovery from fast inactivation, and also pharmacology. So as a control, we used hemp seed oil that was devoid of phytocannabinoids. However, the terpenes were still present. We observed variable effects of the product. The left-hand panel shows representative raw current traces in the presence of DMSO vehicle, and they're depicted in black. The color traces, the colors for the different channels, are in the presence of 3 micrograms of nutraceutical product per mil. The right-hand panel shows the mean normalized peak current amplitude, and this clearly shows that there's a lack of cell activity. There's an overall block of peak current amplitude for all the channels, except at NAV 1.3, where we're actually observing an increase in peak current amplitude, and no obvious effect on NAV 1.8. Interestingly, we saw this increase in peak current amplitude at NAV 1.3 in the presence of hemp seed oil. You'll remember that this hemp seed oil control is devoid of phytocannabinoids. So this would suggest that the effect of the preparation on 1.3 is due to hemp seed oil itself or the terpenes in the oil, and not the phytocannabinoids. The activity of 1.3 is likely to have minimal effect on neuronal function because its expression is highest just before birth, with a very rapid postnatal decline. Next, we examined the effect of the product on the biophysical properties of channel activation and inactivation and compared them to what we saw in the presence of hemp seed oil control. We observed variable changes in the voltage dependence of activation by both the product and hemp seed oil. Hyperpolarizing shifts in the V-half are indicative of an enhancement of channel function, and depolarizing shifts reduce channel function, but clearly both the nutraceutical product and the hemp seed oil are contributing to the effects. Looking at the voltage dependence of fast inactivation, although not all the channels are affecting it to the same degree, the V-half for all channel subtypes was shifted in the hyperpolarizing direction, suggesting that channels move into the inactivated state much more readily, and this is consistent with inhibition. The effects of the hemp seed oil on these channel properties, although less pronounced, are contributing to the overall effect nonetheless. Neither the product nor the control hemp seed oil affected time constants of steady state fast inactivation, and I'm just showing the effects of the product here, but we didn't see any effects with the hemp seed oil either. When we looked at the recovery from steady state fast inactivation, we saw that there was a slowing of recovery for all of the channels except for NAV 1.3 and NAV 1.8. Hemp seed oil was without any effect on recovery from steady state fast inactivation. So slowing of recovery suggests that binding of the compound stabilizes the channel in the inactivated state. This CBD-rich extract inhibits NAV 1.1 through to 1.7 in a concentration-dependent manner to produce IC50 values that range between 1.6 and 4.2 micrograms of product per mil. And when we calculated the concentration of CBD in the product, the IC50 values are the equivalent of nanomolar concentrations of CBD, and that's interesting. The bottom charts show that the effects of purified CBD. So we looked at the concentration response curves of CBD against this subset of channels, NAV 1.1, 1.2 and 1.6, with an interest as epilepsy targets, and NAV 1.7 as a target for pain. And the IC50 values we got for purified CBD are in the micromolar range. So what that suggests to us is that a nutraceutical product is an order of magnitude more potent than purified CBD at these sodium channels. So I'll just conclude by summing up what I've shared with you today. So this nutraceutical product, which is a CBD-rich extract of cannabis, modifies peak current amplitude of the voltage-gated sodium channels NAV 1.1 through to NAV 1.7. The compound had variable effects on the biophysical properties of all of the channels we tested. When we looked at the IC50 values for the product against the different channels, they ranged from 1.6 to 4.2 micrograms of product per mil. And then when we calculated the CBD content of the product and looked at the IC50 values, these are actually in the nanomolar range. And this is very different to the values we got for purified CBD against the subset of voltage-gated sodium channels, which are in the micromolar range. So that suggests to us that this CBD-rich product is an order of magnitude more potent than purified CBD. And so this implies that either another component of the product potently inhibits the sodium channels, or that various components are working cooperatively to inhibit channel function in what's known as an entourage effect. So that really summarizes what I've shared with you today. Moving on, we're now looking at individual components. So we're examining the different phytocannabinoids that we know are in the product, and that work's ongoing. I'd like to wrap up by acknowledging the people that were involved in the project. Steve Petru, my immediate boss here at the Florey, and he's affiliated with the University of Melbourne as well. And you can see the Petru group down at the bottom. And we're actually much bigger than this now. There are more than 50 of us. We work very closely with this team over in the University of Sydney, and I worked particularly closely with Jonathan Arnold and Lindsay Anderson on this project. You see the picture at the top, right hand is the Florey, and I can't believe that I've worked here for nearly 11 years now. And finally, I'd like to thank the Lambert Initiative for their very generous funding. Joy and Barry Lambert, back in 2015, donated $34 million to the University of Sydney to do research into cannabis products for human disorders. And they did this because their granddaughter has Dravet syndrome. And so, yeah, it's been great being able to work on this very interesting project with these really great people. So thank you very much for the opportunity to speak today, and thank you for your attention, and I'm happy to take any questions.
Super, thanks very much, Carol. Lovely to see you again, if it's only virtual. So do we have any questions from the audience? They're still warming up, Carol. So I've obviously got a few, and I have an opportunity now to ask them. So there's been a lot of discussion about, the specificity of cannabinoids. So clearly they have an effect in the brain. Clearly they have an effect on sodium channels, but there's been this disconnect between the pure drug effects in vitro assays and the in vivo efficacy and exposures. So what do you think we're missing here? You think it's because we're using these purified CBDs that we're missing out on these other cofactors?
I think that's probably the case. And with the clinical trials, it's actually quite challenging, because when you're trying to make comparisons using animal models, it's quite difficult, because what we're looking at with human trials is very small patient numbers and a very broad age ranges, and they're all on very different medication regimes. So, and also the clinical trials they're not for just like the Epidiolex, it's not just a monotherapy that they're looking at. These patients are still using the anti-epileptic drugs that they've been maintained on. So that's quite difficult. But I think, to answer your question, yeah, we're missing out on something, because clearly this product that we've tested is very much more potent than purified CBD. So there's definitely some synergism going on here.
Related to that as well is this idea of promiscuity. So whether we shouldn't be taking this reductionist approach to drug discovery, we shouldn't just be looking at one target. Yes, epilepsies, quite a number of the epilepsies are driven by mutations in single sodium channels, but there are other forms of epilepsy that may have multiple targets. So maybe this promiscuity, the fact that it affects most of the sodium channels except for NaV1.8, is a good thing, but doesn't this also bring risk? I mean, I noticed that on your tau from recovery, the most significant effect is actually on the cardiac NaV1.5 channel. So is there some side effect risk from these natural product usage in the clinic?
Yeah, so that's a great question, and that's very true. So I think if you were doing proper double-blind clinical trials with this compound, you would definitely be looking at the safety aspect and monitoring cardiac effects. However, cannabis is actually well tolerated in humans, and there's very little evidence to show
cardiac effects and the concentrations that have been recorded in plasma from cannabis users is much, much higher than what the patients are receiving in the clinical trials for purified CBD. However, yeah, I think this caution needs to be used, certainly from the cardiac side. But they're definitely cannabis is well tolerated by humans.
Okay, I'm not going there. We have a similar.
According to the literature.
We have a similar debate in the UK. So there's a similar thing with these young families, parents who are getting involved with buying cannabis because it's actually medically legal in the UK, but doctors are not prescribing it. So there are families and young families and children who are just basically paving the way and driving the drug discovery themselves. So it's interesting what you said about the Lambert Initiative as well. It's good to see that that's also happening in Australia, but it's also a little bit sad that that has to be the way. But I think this is an important part of drug discovery, is getting the patients involved.
Yeah, what's really ironic is that we spent 2 years trying to get these individual cannabis products that we're testing for this new publication we're just preparing. It took us 2 years to get past the red tape to move them over to Victoria. However, I believe these days you can smoke a joint in Canberra.
We've always been able to do stuff like that in Canberra. That's why I quite enjoyed my PhD time, and that's why it took me a couple of extra years to finish up. But anyway, that's another story. Thank you very much, Carol. Lovely to see you, and good luck with continuing to chase this story.
Thanks very much. Thank you.
Right, we're on to the final speaker today. So Professor Francesc Sureda. So he gained his PhD in pharmacy at the University of Barcelona, and then he went to postdoc in Italy, returning to Spain, first again at the University of Barcelona, and now, I'm going to mispronounce this, but the University Rovira e Virgili, which has 2 sites in Reus and Tarragona. And Niels, you will be interested to know that his university has a department of oenology, so the study of wine. So I think, are you already aware of that? Is that perhaps you're collaborating on the side with Francesc? So today, Francesc his interest is in toxicology and neuroscience. So he's going to take us back to an old story of marine toxins and ion channels, such as tetrodotoxin, ciguatoxin, these food poisoning toxins. So he's going to be using some of the electrophysiology as a form of a cell-based bioassay for seafood safety surveillance. Well, that's my take on his abstract. So I'm looking forward to his talk today. Please take it away, Francesc.
Okay, thank you. Good morning to everybody. Is the sound okay?
Perfect.
Okay. So I hope that you are seeing my presentation right now. I think that everything is fine. Well, good morning to everybody. Thanks for this introduction. First of all, of course, I have to apologize for not being here in Munich. It would be a pleasure, but these days are crazy for me, so it has been impossible this time. Maybe next year, I promise. Okay, thanks especially to Ali for inviting me, because I'm really new to Pathfinder, and what I'm going to present here is only the result of 3 or 4 months of work with the Pathfinder. But I hope that I will give you an idea how it has been to start working with it without having any idea of patch clamping. Of course, we had some experience with other in vitro methods in fluorimetry, for instance, working with Fura2 and SBFI and so on. But we have never worked with patch clamp. And in fact, when we came here in April, I took an electrophysiologist with me, David, only to be sure that the patch liner really worked for our purposes, because I had no idea if it would fit or not. Well, this work is part of a joint venture between the University of Barcelona, University of Virgili, and the IRTA, the Institute of Agri-Food Research and Technology, which is located in La Rapida. What we intend here is to find a new method of marine toxin detection in food samples. I am sure you are all familiar with this image. As many of us, we are reorienting our research projects to fit the United Nations Sustainable Development Goals agenda. We are all very concerned about how climate change is affecting the marine ecosystem and how we can get resources from the sea, and at the same time make sure that what we eat is safe and in a sustainable and efficient way. Of course, maybe some of you are familiar with marine toxins. If not, you should know that these compounds can be a real threat for humans and also other organisms, since they may interact with enzymes, channels, or other proteins, causing the death of some organisms. For instance, these are dead fish by harmful algal blooms. But also humans can be affected by these harmful algal blooms. For instance, presenting different diseases like amnesic shellfish poisoning that is caused by domoic acid, or ciguatera, which is caused by ciguatoxins. And they cause other symptoms like temperature reversals, or for instance, changes in sensory alterations. And it is interesting to note that many of these toxins develop these symptoms by inhibiting or potentiating sodium currents through voltage dependent sodium channels. Of course, apart from these health issues, there are other issues like economical losses due to the closing of facilities, aquaculture facilities, and this is also a matter of concern. Of course, algal blooms are typical of warm waters and are quite frequent in the Caribbean Sea, but for some time now also in the Mediterranean Sea and other non-tropical waters like Canary Islands or Madeira in Portugal. As you all know, there's a thing that is called global warming, and of course this affects everybody. And this is a picture of the trend of sea surface temperature to increase, especially in secluded areas like the Adriatic Sea or the Aegean Sea, but also the northern western part. We are here in Reus, and this is also an area that is has warming temperature in the latest years. These are data from the 1993 to 2020, indicating the increase in temperature in sea surface temperature in the Mediterranean. And this has been correlated with more and more frequent algal blooms in the Mediterranean Sea, in this area, and in this area. And the question is if these algal blooms are harmful or not. They are always harmful because when we want to have some days in Mallorca, you expect to have clear and limpid waters. Okay, so at least economical issues are involved during algal blooms. Okay, because the first category is water discoloration, which can be important sometimes, but this is not harmful really to health. Category 2 is reserved for blooms that are harmful to humans. The category 3 is for blooms that are harmful to wildlife, and there's another category that is harmful by inhalation. Some algal toxins can contaminate the air that we breathe, and this can cause some symptoms, or maybe algae are forming aerosols that can be inhaled, and this can cause also health problems. But algal toxins can contaminate seafood that later will be eaten by other fish or humans. So there's a need to control the presence of these toxins before this food is eaten. And there is another concern caused by not algal blooms, but by fish that they are not, they are quite strange in the Mediterranean, but as you will see, it is more and more frequent to see pufferfish species in the Mediterranean Sea. It seems that they are invading the Mediterranean Sea through the Suez Canal, okay, here. They have adapted very well to the Mediterranean Sea. They have strong teeth, can damage fishing nets and bite people, and it's currently estimated that they make up for 4% of our snort catches. So this can be a threat too, because some people are contaminated or are poisoned by eating this fish. As everyone knows, some species of pufferfish contain tetrodotoxin, and which is a channel blocker, and sodium channel blocker, and this can be fatal if injured. Okay. You might think that it is easy to discard a pufferfish from captures, as we tend to think that a pufferfish always look like a balloon, but it is not always. This is not always the case. This is a Lagocephalus scleratus specimen that was captured near Tel Aviv and in Israel, and can be mistook by an edible specimen. In fact, it was responsible for poisoning that was treated with cholinesterase inhibitor. So the European Agency for Food Safety issued scientific opinion regulating the maximum concentration that a fish or seafood can contain before being eaten by the public, and has stated that the concentration below 44 micrograms of the tetrodotoxin equivalents per kilogram of meat or shellfish meat should be safe. To detect levels of marine toxins, one can use different methods, and mainly or biological assays, or also analytical methods. Okay, and that at the IRTA, they are using both methods, but they were mainly interested in developing new cell-based assays that could detect toxins like tetrodotoxin or cigotoxins or saxitoxins. So we started to think that the automated patch clamp analyzer could do the job to detect these marine toxins in extracts from fish, because we knew that APC, that automated patch clamp, is useful in preclinical safety pharmacology, for instance. So our main aim was if automated patch clamp could be useful to assess TTX toxicity as a routine assay from seafood samples, in a way that it should be sensitive enough, fast, reliable, reproducible, and if possible, cost efficient. So first we thought on a small portable system like the PortaPatch, and I asked my longtime friend from our PhD days, Marisa Vinyals, who is working at Almirall, which is a medium-sized pharmaceutical company in Spain, if she had any idea how these systems work, if they could fit in our project. Well, we met during the days after the pandemic in Spain. She told me, well, you know, we are no longer using our patch liner system, and as we are outsourcing this all the clinical and regulatory stuff to companies, and maybe I can rent some papers for a donation to your lab. And of course, this was music to my ears. And after a couple of months, I had the patch liner in my lab. As you can see in my face, I was very, very happy. And okay, we did some actualization of the system and started to prepare to work with it. An important issue to solve before starting working with the patch liner was which cell line to use. Do we have to spend 8,000 or 10,000 euros in a validated cell line that expresses a particular NAV channel, or can we use our well-known cell line that we were using for other cell-based assays at the IRTA, the Neuro2A cell line? This cell line is being used typically to assess the presence of CTX, STX, or TTX, ciguatoxins, saxitoxins, or tetrodotoxins in what is called a Neuro2A cell-based assay using the viability assay, the MTT assay. So at the IRTA, they had thousands of data that correlated the toxicity of these compounds with analytical data using liquid chromatography and other analytical methods. This figure is taken from an article from Antos Tudor, which was a PhD student at my laboratory collaborating with the IRTA. And as I mentioned before, both methods are complementary and have pros and cons. And one of the advantages of the Neuro2A CBA assay was that it doesn't need internal standards. Sometimes you have analogs that you don't have. You couldn't got it as a standard. Okay. Well, we thought that we like to evaluate samples with patch liner, as this would shorten the assay to minutes, because the MTT assay is quite time consuming. You need 2 or 3 days to get the data, and with patch liner you could get some results in minutes. So we have plenty of data with Neuro2A cells, and we started with this cell line, and Ali from Nanion told me that they have some experience with this cell line and that it should work. So we thought that it was a good strategy. And in fact, it has been described that both NAV 1.3 and 1.7 are expressed in these cells, and they were used before in patch clamp, in classic patch clamp. And so we thought that we could use these cells in automated patch clamp. And this was after we started to use these cells using other methods like FURA2 for calcium mobilization studies and SBFI for sodium mobilization studies that were a little bit disappointing. So we were intrigued, we were very interested to know if this could work. It took some months to have the patch liner ready to work because some software actualization was needed, and the most important thing is that we did not have any idea of electrophysiology. But in April, we had everything ready, and I was eager to start playing with a new toy. So I ordered some tetracaine and tetrodotoxin to start the experiments. But since this would took 7 or 8 days to arrive, these products at our lab, I ran to the pharmacy and bought some lidocaine. And we started with lidocaine, and I was surprised to see that the patch worked at our first attempts. And well, the potency was not very high, but it was consistent with what we found in some other reports. And then we did some other concentration response cures with tetracaine and tetrodotoxin. You can see here the blocking effect of tetrodotoxin. These were preliminary results. Then we did other experiments to complete these concentration response curves. And we shared these results with Ali, who had the idea to merge our results with theirs, and this resulted in a communication in the neurophysiology meeting in Copenhagen a month ago. So thanks, Ali, and the rest of the Nanion team for this collaboration. So we had the patch liner ready, and with no idea of patch clamping and only a 1.5 day of training in Munich, we started for experiments, always with the very valuable support from Ali, Christian, and Andras from Nanion. First, we wanted to assess if our extracts could alter sodium currents by itself. This is what we call the matrix effect. Okay, so in acetic acid aqueous extract from fish, you can have other substances that are present in the extract that are not specific toxins, but may affect response. So you have to characterize first that if a negative extract could affect sodium currents in our system. We always use the external solution as a solvent in our concentration response curves. But if you want to use fish extracts that are in an aqueous solution containing 0.1% acetic acid, you have to carry some additional experiments, dissolving TTX in the solvent and looking at differences. We did not find any difference between our first experiments and then with using a 1 part on 10 with acetic acid solution. The EC50s were very similar, circa 3 nanomolar. So we moved on and assessed the effect of negative samples at different dilutions. Here you can see the measurements of 6 different cells at the same time. The efficiency was quite good, having gigaseals in 60–80% of the cells. So this was quite consistent, and there were good news. Here you can see this is the mean of the same results. Okay, so here you can see that different dilutions of the matrix of the negative solvent was not affecting sodium currents. Maybe here, but only one of the cells. And when we mixed tetrodotoxin with matrix with the negative extract, then you can see that neither the TTX effect was inhibited or altered. It was very similar to tetrodotoxin 100 nanomolar in external solution. So results were not affected by the negative extract. Here you will find some useful tips that we used. We used for suspension stem proacutase from Thermo Fisher for cell detachment, and very careful suspension using blue tips and diluting 1 milliliter of this acutase stem pro suspension in 3 milliliters error PMI, non-supplemented, and 3 milliliters external, and quite low cell density. So another typical experiment that we made is what we call a spiking experiment. It consists of pairing the tetrodotoxin concentrations, like if you were going to do a concentration response curve, but using a negative aqueous extract as a solvent to assess and measure any matrix effect. In this case, we used 1 part of extract and 19 parts of external solution. Here you can see that this is the spiking experiment curve. As you can see, this differs very little with our reference curve or calibration curve, which is the green one. This is the mean of 3 different experiments using tetrodotoxin. And here you can see the EC50 values that were very similar in the middle nanomolar range. And sometimes we use, from time to time, we do some tetrodotoxin concentration response curve. Here it is shown the one performed the 15th September. And we always have very consistent results. The previous results were from muscle, and we also performed fish muscle, and we also performed experiments with a skin sample. And we are waiting for results. Well, we are waiting for a sample of liver. We have used liver before, but we want to repeat the experiments with using the same protocol as shown in the last slide. And then we wanted to test positive samples. This is our positive samples from Lagocephalus scleratus. So this is a specimen that of real pufferfish, toxic pufferfish, poisonous pufferfish. And remember that this was our main objective or our main aim to be able to assess toxicity in real samples. So we used different dilutions of positive samples on summing conductance in Neuro 2A, and using different dilutions of this positive sample, 1 to 1000, 1 to 100, and 110 of muscle. These are different dilutions with skin samples, and these are liver samples. We want to test also gonad samples. I'm going to look for this sample tomorrow at the IRTA. And we could quantify the quantity of tetrodotoxin from nanomolar concentrations to micromolar concentrations. We use always tetrodotoxin as an internal standard. Here we use tetrodotoxin 5 nanomolar, and we interpolate these values of a normalized block to this green curve, this calibration curve. So in this way, we can quantify more or less the quantity of tetrodotoxin in a sample. We always look for the dilution that more or less give us 40 or 50 or 60% inhibition of sodium current. So in this way, we can have an idea of what concentration of tetrodotoxin we have in samples. Using 1 to 20 dilutions, we can reach a limit of detection that would be near to 100 micrograms tetrodotoxin per kilogram of tissue. We think that using 110 dilutions or improving the extraction method, we can reach the 44 microgram tetrodotoxin quantity per kilogram of tissue that EFSA recommended. And maybe optimizing the method, we could reach up to 10 micrograms of tetrodotoxin per kilogram of tissue. So we are quite happy to demonstrate that the method is useful for us. But more or less our aim has been met, but Stölzle is also offering other possibilities, since it's a fast way to evaluate methods that can bind TTX. This is useful to us as a method to validate new non-cellular detection methods that we are testing at the IRTA by Jaume Robertet, for instance. And here you can see how a neutralizing antibody is able to inhibit the effect of tetrodotoxin. Okay, so this can be routinely used to assess manipulations or newly designed antibodies or PAMers or silicodextrins that we may test for detection issues. Okay, up to now we are not having success here, but at last we have a fast method to know that we are not in the right way, so we can look for another method. So as a conclusion, the Neuro2A cell line is, we think that is a suitable, non-expensive biological substrate to test drugs or toxins acting at nerve channels using the patch liner. And Nanion patch liner is in our hands, a reliable, robust, and easy of use equipment for scientists with very little training. I think that we can say right now that the TX blocking activity is easily detected in samples from fish or seafood with a neglectable matrix effect, and that reaching detection limits of 20, maybe 10, 40 micrograms of tetrodotoxin per kilogram tissue is feasible. And now we are working with CWA toxins and other toxins to see if this method can be used also for other toxins. And we think that the patch line is a very valuable instrument in any lab interested in pharmacotoxicology. And of course, it is supported by a committed team of people that are always ready to help. And this is our team, or at least the team that visited Nanion facilities in April. Monica Campas is the chemist and team leader at the IRTA. David Soto was the invited scientist that, as an artisan electrophysiologist, would help with telling us that the patch liner was okay for us. And this is me. This is Francesco Sureda. I used to be a pharmacology that always thought that patch clamp was for geek scientists, but until now, okay. And of course, thank you very much to all the people at Nanion, especially Alison, Ali Obergrusberger, Christian Grad, and Andras Horvath, who were very helpful starting our first experiments. Of course, we are indebted to Marisa Viñals for the donation of the patch liner, and the Ministerio of Ciencia and Innovación in Spain has provided the funds for doing this project. So thanks, Ali. I'm ready for questions.
All right, thanks, Francesco. So unfortunately, we only have time for one question. Anybody brave enough from the audience? No, the Alamo man, he's still keeping his hands in his pockets. Right, so I have some questions. One question, the TTX, the samples you're having from pufferfish, these are real samples from the fish from the Mediterranean?
Yes, we have several samples. This was our sample, and we have only one fish, but we have collaborations with Greek scientists, and they have many. But for testing the system, we used our own samples, and these are pufferfish species that you can find in the Mediterranean right now. Yes,
It's a real thing, then. This is a real issue. All right. Well, we loved your selfie with the patch liner. Everyone was smiling here. So welcome to the world of automated patch clamps, sir. And may you have good luck with your research. Thank you very much.
Thank you very much to all of you at Nanion.
All right, thank you, everybody. So that's the conclusion of today's first morning session. Thank you to all of the speakers. And for those of you here in the flesh, we have a 30-minute coffee break. So we'll see you all back at 11 o'clock Central European time. Thank you very much.
Hello, everyone. I hope you can hear me. I welcome you to the second session of today's user meeting. And now we are switching to something a little bit different. We will focus on disease modeling using stem cell-derived cardiomyocytes. And I'm very happy to welcome two people who I think share the furthest traveled award to arrive to our user meeting, one coming from Australia and one coming from United States. The first one is Nathan Palpant, and he is currently lab head and associate professor at University of Queensland. He, in his lab, is using various methods to study cardiovascular development and disease, merging bioinformatics, cell biology, genomics, and physiology. He also received numerous awards for his work. Those include International Society for Heart Research Young Investigator Award and Lorne Genome Millennium Science Award, very long awards. And I'm very happy to welcome him here. Apart from his work in academia, he's also co-founder of Infensa Bioscience, which aims to develop new therapeutics for cardiac and cardiovascular ischemic injuries. And as you can see here from the title, today he will talk about maturation of iPSC-derived cardiomyocytes for disease modeling and drug discovery. Thank you very much for coming, Nathan.
Pleasure.
And please, the floor is yours.
Thanks. Well, it is a long ways from Australia. It takes about 28 hours to get to Europe. And I decided to just make it part of a blitz through Switzerland, Germany, and UK. So it's great to actually meet the Nanion team as well for the first time, since we've just been building in the CardioX and the FlexSite system into my research lab over the last 6 months. So what I'm going to talk about today is a bit more of a longer backstory on some work that we've been doing, and then how we're incorporating some of the technologies around cardiac contractility and disease modeling with the CardioX system within the research program. So let's see if this is going to go. So just a quick disclosure slide. The research I'm going to present today has to do with some of the work we're doing with Infensa Bioscience. And it focuses on, again, looking at Jennifer's talk this morning around dangerous sort of flora and fauna and animals in Australia, is that this is really around an interesting study that we've been developing around the venom of the funnel web spider, and is a close collaboration with some colleagues of mine in the IMB and at the Victor Chang. So the underlying problem that we're really interested in is this issue that the heart is ultimately very sensitive to injuries like heart attacks. And the fact that the heart is one of the organs that really has no regenerative capacity, which is why we're really faced with a significant disease burden around ischemic heart disease. It's the leading cause of death in the world, and has held this for many decades, really. And I just wanted to give you a bit of a background in terms of like, why has this failed, and what is the problem with the research in terms of being able to find drugs that actually make a difference in ischemic heart disease. And wanted to highlight an interesting study that maybe some of you might not be aware of, but it's my former postdoc advisor, Chuck Murray, back in the mid-1980s, published this study where they found that if you introduce short episodes of transient ischemia to the heart can actually tolerate a much more substantial heart attack. And they developed this observation about preconditioning, which means that the heart does have the ability to tolerate a heart attack, but what are the mechanisms that actually are associated with that protection? And this was really an exciting area that really ballooned out in terms of giving us sort of excitement about the opportunity to solve this disease. But interestingly, over the course of the last basically 4 decades or more since that observation, we have really only had one drug that has made it at all close to the clinic, and it is the sodium hydrogen exchange inhibitors that are targeting, cariparide, which targets and blocks the sodium hydrogen exchange channel. Now, the interesting thing about this channel is that it is really a central part of both the ischemia and the reperfusion injury response in the heart. And the clinical trials were really accelerated around these drugs from around the early 1990s. They discovered these drugs as inhibitors of this channel, and then moved them into clinical trials by about 1995. And over this window of time, they were able to demonstrate that these are potent and very effective cardioprotective molecules. But they reached phase 3 clinical trials, at which point they had now sampled and tested this in heart attack patients and in patients that had had coronary artery bypass grafting to show that it is a bona fide cardioprotective molecule, but it was discontinued because basically patients had stroke-like symptoms when they received this drug. And so really since the early 2000s, no drug has made it into clinical trials to protect the heart in the context of a heart attack. And so I serendipitously moved to Australia with no anticipation that I would be studying spider venoms in my research program, but this has been an exciting story that works with Glenn King's lab at the IMB and involves working with spiders and their venoms and understanding the molecules that are associated with venom. And we have an insectarium down in the IMB that houses these spiders and gives us a chance to actually dissect out the underlying biology. Now, importantly, some of you may not appreciate this, but captopril was actually one of the first drugs that was ever derived from venoms for cardiovascular sort of diseases, especially related to blood pressure management. And so this is something that I think is important for people to realize, is that venoms are really potent sources of evolutionarily sort of incredible molecules that have remarkable effects on ion channels and really give us a chance to repurpose some of these molecules, not necessarily, of course, because they derive from venom, which is, of course, a bit of a scary topic, but actually because they could be new sources of drugs. And so now this is really a blockbuster drug that has made incredible impacts for our patients who have cardiovascular disease. So this is, coming back to the work I'm doing in the IMB, is that about, say, 20 or more years ago, there was this discovery that these channels in the heart, or in cells, are called acid-sensing ion channels, are associated with the sort of activation of pH-sensitive processes that are related to ischemia. And a colleague of mine at the IMB, Glenn King, discovered this particular molecule in the venom of the funnel web spider that is off the coast of Queensland, the Kagari, which some people know as the Fraser Island funnel web spider. And it's a double knot spider venom peptide that has close relation to another molecule called PCTX1. And I'm going to refer to both of these molecules in the talk. But basically, PCTX1 is basically a monomer of HI1A, which you can see along the bottom here is basically a duplicate of that same sort of motif. And very quickly, because I want to get to some of the key aspects around what we're doing with the Nanion technology, I'm just going to give you the absolute sort of abridged version of this, which is that PCTX1 and HI1A bind to a channel called the acid-sensing ion channel 1A. And this ion channel is known to be associated with sort of ischemic responses, as I've mentioned. And HI1A and PCTX1 are known to block the activation of this channel. And this is work that has from Glenn's lab, which basically shows the specificity of the HI1A in terms of its ability to block just the 1A isoform and not any of the other isoforms of these acid-sensing ion channels. Now, the important thing is that historically, this channel has been implicated in stroke. And that was really where, over the last 20 years, a lot of the work has really focused around how the channel is mediating stress responses in stroke. And this is work from nearly 20 years ago, where they did a genetic knockout and showed that protection of the brain in the context of an acute stroke, and really was the point at which Glenn's lab really developed the rationale for being able to test these ASIC-1A inhibitors within the context of stroke, which was published in PNAS about 5 years ago or so. So you can see that a lot of the biology around these ASIC-1A channels has been focused on stroke. So, there's not a lot to sort of bridge the segue here, aside from the fact that when I came to the IMB, Glenn's lab was really developing the therapeutics around stroke. And the notion was that the heart also, in addition to the brain, have common sensitivities to ischemic stresses. And really, it's highlighted here in the sense that the heart is one of the least tolerant organs when it comes to transplanting them in terms of the window of time when they can be managed outside of the body. And so I worked with Glenn to sort of set up this rationale that, why don't we at least sort of look into this possibility as to whether or not ASIC channels play common roles in terms of stress responses to ischemia, not just in the brain. And so one of the first things we did was to look at statistical genetic data, which I think is an important sort of step in terms of being able to justify drug targets now that we have this level of information. And so we looked at the UK Biobank to look at genetic associations in terms of the variation within the human population as to whether or not they were associated with sensitivities to ischemia and such as stroke and heart attack. And so you can see here the significance of sort of the association of genetic variation at the ASIC1A locus is associated with myocardial infarction and stroke, and gave us some motivation to continue to build that link between the brain and the heart. And so my lab has expertise in stem cell biology, which is part of the sort of ground work that we do in terms of modeling diseases, and of course, deriving these cardiomyocytes from pluripotent stem cells. And so we undertook a study to look at whether or not ASIC1A and its underlying biology have any role in terms of cardiac cell death. And so what you can see here is that the expression level of ASIC1A is not changed depending on what cells are exposed to pH, but you can see that, of course, as you expose cells to lower pH, you can get a significant induction of cell death. And so we created a genetic knockout of ASIC1A in human iPS cells and performed an in vitro ischemia reperfusion assay, which is basically exposing the cells to low pH in addition to a hypoxic stress overnight, and then placing them back into normoxia for a few hours and assaying cell death using LDH release. And so what you can see here is that the knockout cells have significantly lower cell death compared to the wild type cells during this ischemia reperfusion injury. We also looked at using, this is just acute addition of these ASIC1A inhibitors on cells, where you're looking at any potential effects on electromechanical coupling. And what you can appreciate is on these in vitro derived cardiomyocytes, when you acutely add these molecules, they have no impact on the normal physiology of cardiomyocytes. However, when we expose these, again, coming back to the observation with the knockout cells, when we expose these cells to an ischemia reperfusion assay in vitro with these pharmacological inhibitors of ASIC1A, we see a similar finding in terms of the protective effect of these channels on the viability of cardiomyocytes in that assay. So this is published work, and I'm going to just show you one of the sort of final assays which we did was to take this in vivo and actually test out whether or not these have protective effects in a myocardial infarct model. And so this is work where we delivered HI1A by intravenous injection in the context of an in vivo myocardial infarct, and assayed the recovery of those organs in those animals in a 4-week follow-up using echocardiography and histology. So if you look at the histological slices of these hearts, the sham, of course, on the far left, where uninjured myocardium, the vehicle-treated animals have significant scar formation using this Mason's trichrome analysis. And then on the far right are animals who have treated with HI1A, a single bolus of HI1A during ischemia. And what you can find is there's a reduction in the overall infarct size, which you can see quantified there on the left, about a 50% reduction of the infarct in animals with treatment with HI1A. And we see a significant protection in terms of the overall function of the hearts over the course of a 4-week follow-up post myocardial infarct. And importantly, it's also looking at these parameters of diastolic and end systolic volume that are associated with the adverse remodeling that happens during a heart attack, and sort of demonstrates the fact that this drug is helping to protect the overall structure and function of the heart in the context of a heart attack. So there is overall, I'm just going to give you a summary. We used both pharmacological activators and inhibitors, genetic knockouts in iPS cells and mice, in addition to assaying this in 3 different species and I think 5 injury models in the circulation paper that we published last year. But it really provided a demonstration that these ASIC1A inhibitors are potent potential therapeutics for ischemic heart disease. So the important aspect I think that we are struggling with is to say, well, this is not the first story that has demonstrated therapeutic benefits of drugs that are associated with heart attacks. So what is the problem with the fact that there hasn't been effective translation? And this is why we've just been looking at this and wanting to highlight some of the key areas where we want to try to reinforce and improve the success rates associated with translating drugs into the clinic for heart disease. So this is really looking at a stepwise process where we're looking at trying to really create a bit more rigorous a portfolio around this. Not only looking at genetic data, which I've already shown you, but also in terms of iPS cell modeling using both genetic as well as pharmacological, sort of get that target, a drug target interaction really well worked out to be able to sort of develop that case. In addition to being able to do more complex comorbidity models, which is where I'm going to be heading to in terms of the last part of my talk. And then, of course, there's a need to have not only rodent models where you're looking at reproducibility across multiple sites, and then going into also comorbid in vivo models, where we're looking at the fact that most patients who hit the clinic with a heart attack are not just simply 20-year-olds, which is what we assay them on normal mice who are very young and may not really be properly recapitulating the complexities of heart attack patients. So this is what I want to focus on in the last part of my talk, which is the fact that diabetic patients have a much higher risk of cardiovascular disease and are one of the key aspects by which we sort of want to address the therapeutic benefits in models that are associated with diabetic cardiomyopathy. And so in particular, patients who have diabetes have not only a higher chance of cardiovascular disease, but it's actually associated with their susceptibility to myocardial ischemic injuries. And so in my lab, we've been developing models of diabetic cardiomyopathy. There was one that was published a few years ago, I believe it was in Cell Reports, that in which they were looking at the ability to model diabetic cardiomyopathy in vitro. But we took on a particular study where we're doing our standard cardiac differentiation in vitro and shifting to this diabetic model where they're in differentiated cardiomyocytes and testing out two different models of diabetes. So the cells go through this sort of maintenance mode where they're put into fatty acid media for a few days to really get them sort of acclimated or sort of adjusted to a fatty acid metabolic substrate. And then we shift them back into a glucose stress for a couple of days to model that sort of diabetic sort of phenotype. And we're using one model where we're looking at exposure to a constant state or sort of concentration of glucose, and another one where we toggle between low and high glucose every 12 hours. And so this is more modeling what patients who are going through these swings of glucose would sort of experience in terms of the management of this in regard to the diabetic phenotype. And so this is where you can see on the far left, the cells are exposed to this diabetic stress and assayed for oil red O, which is a lipid sort of indicator. And you can see that both the constant versus variable diabetic models have an increase in lipid uptake compared to the maturation media alone, but it's not distinguishable between the two of those. And similar, BNP, which is a known biomarker of cardiac stress, is also up in both of these diabetic models, but also not distinguishable between the two of them. We also did a fairly sort of routine RNA sequencing analysis of the maturation media versus the two diabetic medias, and found that there's not really anything that substantially distinguishes them in terms of the diabetic phenotype. Transcriptionally, they're basically identical. And so we were also not at that level able to sort of pull apart the biological differences between these two model systems. So this is where we began to work with Nanion and sort of assaying these models in terms of using them in the CardioExcite system. And this was, again, some of our initial assays that we've just been performing over the last few months, where we looked at the contractility between the maturation media, the constant and variable glucose diabetic models. And what's quite clear is that while there is a deficit in contractility with the constant glycemic diabetic model, there is a much more dramatic functional deficit with the variable glycemia in regard to the fact that both the relaxation, the contractility kinetics, and beat rate, all of these indicators really substantially separate out that there is something more substantially pathological about the variable diabetic model. We went on to look at this in the context of other sort of indicators, such as glucose and lactate. So the glucose consumption and their lactate release into the media, where the variable glycemic model really has a much more substantial sort of consumption rate of the glucose, and also similarly an increase in the lactate secretion. And on the ATP utilization, we see, again, separation of these two models, where we're getting a reduction in the overall ATP production with the variable glycemic model. So this brings me back to the fact that we're sort of setting up the assay and wanting to come back again to whether or not AZK1A inhibitors have a therapeutic benefit in a more complex comorbidity model, which was what we were setting up here in regard to the diabetic cardiomyopathy model. And so this was where we came back and looked at whether or not the addition of this comorbid model also recapitulates the known cardiac sort of complexities of a diabetic cardiomyopathy, which is susceptibility to ischemia. And you can see that compared to just maturation media alone, which is on that black line, you're getting, we see a significant increase both with the constant and variable, but the variable has a much more significant increase in that susceptibility to ischemia or perfusion injury. And that's with the LDH release. And we performed the same assay using resazurin, which is more of a cell viability assay. And we see that really it's the variable glycemic model that has a much more consistent phenotype in regard to the sort of susceptibility to ischemic stresses. And so just to wrap up, we've been able to show, this is preliminary data that we've just been pulling together, but HI1A and PCTX1, when added into this variable glycemic model, are significantly reducing the susceptibility to ischemic stresses in the context of this ischemia reperfusion and comorbidity model. So with that, I just want to wrap up and give you the take-home points. We've been excited about identifying a new target for therapeutic development for ischemic heart disease. We believe that this has exciting opportunities for us to be able to develop not only for myocardial infarct, but actually for many other conditions where cardiac ischemia is one of these sort of key vulnerability points for the heart. Just want to wrap up by acknowledging my team. Meredith Red has been the primary driver around the work on HI1A and these AZK1A inhibitors, and has done a handoff to Yuan Zhao Kao, who is a new postdoc in the lab, who's been doing the diabetic modeling and been primarily working with the Nanion team. So I hope I haven't run too long, but thanks for your attention.
Very much, Nathan. Not too long. Do we have any questions for Nathan here from the audience? And also, please, everyone online, if you have any questions, raise a hand or just write in the chat. We have people here reading them. Any questions? You? No? Okay, then I'll start first. As I understood, the cells that you've used, the iPSC-derived ones, are they patient-derived or?
No, we are just, all of the work that we have done has been using a standard iPS cell line just to, do the genetic knockouts. We have a range of other patient-derived lines, but not doing necessarily the diabetic modeling with a diabetic sort of background. But it's a good question, but we haven't moved in that direction just yet.
And looking at the data that you've also shown on the cardiac side, you've shown the contractility data, but have you also looked at any electrical properties of these cells, maybe the EFP change, or have you maybe even tried to patch them, maybe in an automated patch?
I believe we have that. I'm not sure, but I think we've got access to these, but we haven't done any electrophysiology. And I think those are areas where we could definitely expand. Now that we've got the model, I think reasonably well developed, there's a lot that we can do to further characterize these at a physiological level. But again, this is where the physiology has been really important for us to help distinguish those two models in terms of how they're behaving in terms of the disease modeling side of things.
Thank you.
Great.
Anyone? No? Okay, I think also we are a couple of minutes over time, so we will continue with our second speaker. And after thanks, maybe a little bit of an applause for Nathan. Thank you. So our second speaker is Professor Boyd Norman, and he's coming to us from Nashville, Tennessee, from Vanderbilt University. After his training in Georgetown University, he is now professor of medicine and pharmacology at Vanderbilt. And he also has a couple more roles. He holds the William Stokes Chair of Experimental Therapeutics, and he also directs Clinical Pharmacology Fellowship and the Vanderbilt Center for Arrhythmia Research and Therapeutics at the Vanderbilt University Medical Center. Bjorn is a longtime CardioExcite user. We also learned last night that he's also loving hiking as well as wine and skiing. So he's the right place here in Munich. Thank you very much for coming so far away. And today we will hear about how he was using CardioExcite for deciphering ion channel variant pathogenicity in human iPSC models. Thank you very much for coming, Norman.
I'm mute. So now so my laboratory is working on arrhythmia disorders primarily, and we have been using the iPS-derived cardiomyocyte system as a model to study mechanisms of arrhythmogenic disorders since 2014-15 or so. And so if in this talk today, I would like to summarize our studies with hypertrophic cardiomyopathy, and then validate the iPS models against the mouse models, which we had traditionally used, and then share with you some of our work with CardioExcite in 4 different arrhythmogenic disorders using iPS-derived models of hypertrophic cardiomyopathy, dilated cardiomyopathy, long QT syndrome, Brugada syndrome, and then finally end up with some studies using the optogenetic pacing feature of CardioExcite. So, and I've looked at the year, Thomas Eschenhagen, Christine Mummery, and I, we published this review already in 2015, kind of summarizing some of the challenges of this modeling, where we're working on the first principles that differentiating induced pluripotent stem cells in 15 or 21 to 30 days in a dish, we can recapitulate a phenotype that takes decades in human to develop. And so there's a big question mark for, at least for me and for a lot of us in the field, if and to what extent this is a valid model system. And some of our early work actually was focused on that. We started out by actually working a lot on maturation, it was mentioned before. And so a number of our early publications were trying to make cardiomyocytes in a dish look at least a little bit more like a human cardiomyocyte. After we had completed that, then we went on and looked at hypertrophic cardiomyopathy, which is the disease of the sarcomere. And in particular, we focused on a mutation in the troponin T molecule, TNT-I79N. And why is that? Because I spent about 2 decades studying this model in the mouse and working out underlying disease mechanism, how the mutation in the troponin molecule causes disease in vivo. And through a number of studies, we worked out that these mutations cause like key features of hypertrophic cardiomyopathy, which is increased systolic function, decreased diastolic function. We showed that they cause energetic deficits. And for the purpose of this talk here, we could link it to an increase in myofilament calcium sensitivity, and importantly, an increase in cytosolic calcium binding or buffering. So those were key papers that we worked out. And again, it took us a couple of decades to do this. And I'm just going to run through those real quick. This is studies from adult mouse cardiomyocytes showing that there is an enhanced calcium buffering in those cells. that results in changes in the calcium transit kinetics, which can and had shown also lead to a proarrhythmic phenotype, especially at fast pacing rates. So calcium just triggered arrhythmias. Now the question was, what took us about a decade to work out the mouse, can we show that also in humans? And so Lily in my lab used CRISPR-Cas9 to generate a human TNT I79N IPS models. With using our maturation media and the soft substrates, we were able to generate pretty nice looking myocytes. And to our surprise, we actually could very easily show that this mutation causes sarcomere disarray, as illustrated here, and also sarcomere shortening, key features that we had seen before. In the functional assays on a single cell level, we could show that there is the typical changes in calcium handling with an increased contractility and diastolic dysfunctions, impaired relaxation, and enhanced sensitivity to calcium. And so in this IPS cells, we actually could reproduce what we'd done and was much more difficult in the mouse myocytes in a couple months. So this study actually didn't take very long, and to summarize this approach. So here we have a human myocyte, and by using a CRISPR-Cas9 to knock in the variant, we can very quickly generate a disease line and already have an isogenic control. So in my laboratory, when we want to understand mechanisms of a genetic variant, we don't use patient-derived lines. We use a control line that makes really good myocytes, use CRISPR-Cas9, bring in the mutation, we have our isogenic control line, and then we can study the phenotype. And in the case of the hypertrophic cardiomyopathy mutation, it actually really recapitulated what we had shown before. And so I was very enthusiastic. So the next step was, we need to get a higher throughput way of assessing contractile and electrical function. Because again, as I told you, we're interested not only in hypertrophic cardiomyopathy, but also dilated cardiomyopathy, and then the various electrogenic disorders, long QT syndrome, Brugada syndrome, and CPVT. And so I reached out to Nanion. I saw them at the biophysics and said, you have a system here, a 96-well assay, that gives me both contractile function as well as an electrical readout and in the cardiac side, which is a 96-well system. So I was very excited about it. We got one of the first systems in the US, and then we rolled it out onto our various cell lines. And so for the I79N model, actually it worked really well. Here we could show that very quickly, using the impedance feature, that the response to calcium, to extracellular calcium, to the increased contractility, as well as the slow relaxation, could be shown rather quickly. So for HCM, using the impedance feature, it works really well. Then we studied a dilated cardiomyopathy IPS line. Now, this is actually from a patient at Vanderbilt, LMNA variant, which is the second most common one. We generated also isogenic controls. This is not published, but just to show you, compared to population control, the DCM has essentially also slower kinetics, which is illustrated here. Now, variability in this line is high, and these are still studies ongoing. And so, but in the initial cardiac side screen, it definitely detected abnormality, and we're going to get at that in a minute, because that's really sort of more the goal for us, to be able to say whether or not a genetic variant has a pathogenic phenotype. Let's switch gears to the long QT syndrome. Most of you are probably familiar with that. It's caused by usually variants in ion channels, resulting in an action potential prolongation, either increase in gain of function of the sodium channel or decrease in function of the repolarizing potassium channels. And there's also some other genes that have been associated with one. And this in particular was reported not too long ago the triaden knockout syndrome. Now, triaden is actually a interest SR in the sarcoblastic reticulum protein that anchors to the intracellular calcium release channel. And we had reported in already 2009 that this should be a candidate gene for long QT, because we made a mouse because we were studying EC coupling, and the main phenotype was a long action potential prolongation. Turns out it was indeed discovered in 2013 in humans that a complete knockout of this protein causes severe long QT. And Lily went ahead and knocked it out in our IPS lines, and indeed it causes an action potential prolongation, which can be easily reproduced also on the cardiac side. And so we're still working on out the exact mechanism in human cells. So in principle, it works really well for that. And then finally, the Brugada syndrome. Brugada syndrome is another ion channelopathy that's characterized by a loss of function mutations in the sodium channel, a Brugada-like phenotype in EKGs. And the question was, does it also cause a phenotype in the cardiac side? And one of the characteristic features is that compared to an isogenic control, this is Kevin Purcell working with Dan Roden's group, the beat-to-beat variability of the action potential is quite drastically increased. And he could also show this in the beat-to-beat variability of the beat-to-beat variability of the EFP direction. Again, here we have a sodium channel loss of function variant that caused Brugada in humans from a patient at Vanderbilt, and the key feature was this beat-to-beat variability. So far, so good. And what I've shown you is that in principle, we can use the cardiac side to rapidly detect both contractile as well as electric phenotypes of genetic variants in IPS lines. So now, the reason we're interested in this is really to decipher ion channel variant pathogenicity, because we have patients in the clinic that come to us with some form of arrhythmia disorder or cardiomyopathies, and then a gene panel is done. Multiple genetic rare variants are being identified, and it leads to what Mike Ackerman has termed the genetic purgatory. Yeah, so you don't know whether this variant is pathogenic or not, and whether it is responsible for the phenotype of the patient, the disease of the patient. And so we were looking all for fast ways and ideally high throughput ways to provide functional information. Now, there's many approaches how to do this, but given that it's so easy to introduce a variant into IPS lines, my lab has sort of been interested in, and we're looking at a number of lines to use the IPS technology to provide that pathogenicity information. And now I'm going to show you the case study, which we published using this patient-independent model of a calcium channel variant that was identified in a kindred with long QT. And this family had been followed at Vanderbilt for a long time. You see the timeline here. When initially they couldn't find a disease gene, there was an ICD placed, and then which is defibrillator, and then the family was thinking about whether to remove it because it wasn't clear if the patient really had a disease mutation. And then the patient was tested again, and now at that point, this fairly benign-looking calcium channel variant was identified. Yeah? And it's a very conservative change. It's not in a classic disease region, so it was absolutely unclear whether or not this calcium channel variant was caused for the long QT phenotype in this family. So Lily in my lab again made a knock-in IPS line using our standard control line to really identify whether or not this variant is pathogenic. And there's a common misconception in the field that we need to use patient lines. Now, in this case, the patient line would be completely irrelevant, because all we would see that the iPS cells have a long action potential, a long QT. And so in order really to show mechanistically that the variant is causative, we would have to go in and repair or rescue it. And so it's a much, if you simply want to know whether or not this particular genetic variant causes disease and has a phenotype, then a very straightforward approach is to simply put it in a control line with a different genetic background and see if there's an effect of that variant. Okay, so we did this. We used our cardiac site, and we didn't really find anything, because these iPS lines, the mutant ones, did have a prolonged EFP or FTP max, but they were also beating much slower, which is a common problem, and everybody in the field knows that the beating rate determines the duration of the action potential. That's a very physiological response. All of us have different adaptation of the repolarization when we our beats get faster, so to improve the filling of the heart. And then, so these were the spontaneously beating ones. And once you correct for the faster beating rates using those correction formulas, Bazet, Fredericia, Framingham, which is used for human QTs, there was no more difference. So did this mutant now cause a phenotype, or did it not? And I'm making this point here because there are so many studies reported of spontaneously beating cells that actually don't provide much information, because even the different population lines that we have in the lab, they all beat at different frequencies. So for me as an electrophysiologist to look at electrophysiological phenotypes, I need to pace these cells. We had an electrical feature already in our cardiac site, but there is some toxicity with chronic pacing. So we went with the optogenetic approach. And so I've been actually asking now for that for quite a couple of years, because it's such a simple way to introduce a channel rhodopsin into these cells using AAV1, works like a charm. And then you can beat them, you can pace these cells optically for long periods of time at different rates. It's really well tolerated, and it doesn't give you any electrical artifacts. And so we use that the SOL system for the study, paced electrically, paced optically. And this shows also the difference between the spontaneously beating cell and the action potential. What you reduce is your depolarization time. The time it takes for the activation rate to spread across the monolayer. And this is summarized here. And so compared to compared to the unpaced spontaneous beating or paced at 1 Hertz, you see that these depolarization time, the time to the onset of the upstroke of the electrical signal is drastically reduced. And there was no difference between the two mutants. But once we did the pacing, we could then clearly show that in all 3 independent generator lines, there was a dramatic prolongation of the electrical field potential duration, as illustrated here, consistent with the idea that this is a pathogenic mutation that causes bona fide long QT. We confirmed this by standard APD measurements using standard action potential measurements, again shown here. This variant in the calcium channel does produce a long QT. What is the underlying mechanism? So these are alpha calcium channel current measurements. As you see here, the IV is not, actually these are barium current measurements. So the current density is not changed. The inactivation kinetics are not changed. It impairs the voltage dependent inactivation, which is classic for LPT8 or Timothy symptom like mutation causing this phenotype. And in the most likely explanation, because this is actually, it removes, it introduces a phosphorylation site. And so most likely what it is actually that this threonine is now, that new phosphorylation site that's being introduced by that mutation is actually phosphorylated, giving this otherwise minor change, a very bulky residue. And in studies in HEX cells where we introduced a phosphomimetic mutation, that also showed that there was a changes in the gating of the alpha calcium channel. So with that, I come to the end, and I've shown you that human iPS-derived cardiomyocytes are really a useful model for a wide range of human cardiomyopathies that potentially could be assessed by high throughput approaches using the cardiac site and maybe even the SyncroPatch, as we heard before. Stay tuned, there's more to come. With that, thank you very much for listening, and this is the team who really did all the work.
Thank you very much, Bjorn, for this wonderful talk. Do we have any questions from the audience? Nathan?
Great talk. So my question is around the fact that in iPS cells, of course, the isoform changes that occur during development, like with myosin and troponin I, myosin light chain, et cetera, which are known to be key disease-causing genes as well. What's your experience in terms of being able to sort of accommodate or account for isoform differences that could complicate being able to disease model using iPS-derived cardiomyocytes?
Again, you pick your poison, right? So it depends on what mutation you're studying, and we're using it for genetic approaches. So of course we look at variants. If the sodium channel usually is the neonatal isoform. So if the variant is primarily an adult isoform, you wouldn't necessarily expect that. You know? And for troponin T, there's also not much of an issue. Troponin I, there's a huge switch from slow skeletal to cardiac TNI. Now, we have taken the approach to use the maturation methods, where T3 plus X, I didn't even talk about that, but you do get switches in the isoform to a more adult phenotype for a number of diseases. Delayed rectifiers are drastically upregulated. The mitochondrial function is drastically upregulated. And so each of the labs have sort of worked with their own little cocktails to get them into at maturity to study wherever the genes of interest are. Now, so for that, that's why for us it's not a big issue. But for somebody who studies diabetes in the dish, it's a major issue. Yeah, because you're looking at it a much more holistic approach, and whether that now still translates is a big question. So I hope that answered the question. Yeah.
Thank you. We have another one here. Will, please.
Hi, cheers. Really nice talk. I've got a quick question for you about the idea of stimulating these iPS cells as opposed to the spontaneous activity. And that was really great. We also stimulate our cells as well. You use those correction factors on your spontaneous field potential durations, and they showed no change. Would you then recommend, in general, that using these correction factors are really not a good way to go for analyzing spontaneous data?
Absolutely. And this is what I didn't know. This was right before lunch, so I took out a couple of slides. And so one of the first things, and we did this a while ago, was actually now that we have this pacer, now we can come up with a better correction formula. Yeah. And yeah, we felt that when you look at the slopes and the different cells, that a correction formula using the Federica correction should probably be better, but it doesn't work, and because the lines are so different from each other, and we don't even know in the first place what's the driver of spontaneously beating in iPS-derived cardiomyocytes. It's a multitude of factors, and it's clearly very different than what's happening in the sinus node. So the rate-dependent regulation is very different from a human heart. It's like the HCN channels, the IF currents is involved, but even with an HCN blocker, we cannot never completely block the basing rate. And it's most likely in the monolayer, interactions between a group of cells, and there's calcium dependent mechanisms, there's essentially IF dependent mechanisms, there's coupling dependent mechanism, there's action potential duration dependent mechanism. Because one thing we find consistently is that every LPT line, which has a long action potential, beats slower. Yeah. But what I wanted to show here is really these correction factors, they don't work, and they've never been designed for that. You could go ahead and come up with your own, and a number of groups do this. I have personally very little faith in the validity of those correction factors. And as an electrophysiologist, it's sort of like, just paste them. Yeah, correct the rate. And because if you're interested in the duration of the repolarization, have them beat at the same rate. And then you're comparing apples to apples, right? And so, yeah, but it is also striking that a lot of disease phenotypes actually manifest themselves in the beating rate. No? And a lot of my colleagues find that these beat-to-beat variability in the IPS-derived myocytes is often associated or found in Brugada syndrome, right? Where there's the loss of function of sodium channels, CPVT mutation, those that usually have an increase in beating rate. A number of, anyway, or beat-to-beat variability, that seems to be a common factor when you have genetic, patient-derived IPS cardiomyocytes derived from patients with rhythmogenic disorders. So I hope that answers the question and gave me my chance to show this, because yeah, did I show that? Yeah. So we found actually the Fredericia worked best, yeah, and pretty close to, was a great correction. But in the LQT line, it didn't work at all, and the Frederica wasn't any better than the other two. Yeah? Meaning that it just it works probably for one line, you can do it, and but it doesn't mean it will work for another when you have abnormal repolarization in some of those ion channels. Yeah, because their rate dependence, depending on what channel you alter, will be different. Yeah.
I will not stop lunch for long. We have 7 minutes, so I will allow myself also one question for you. So for the LQT data that you've shown, you looked primarily on the EFP signals, because as you mentioned in the cardiacs that you could look at both. Did you see any contractile changes in these cells?
So we actually, as we discussed, analyzing the EFP is tricky, yeah? And because of the T-wave waveforms are quite different depending on what you're studying, and I mean even that isn't a good example, right? So they don't always look like that. They can be biphasic, can be inverted. Where's the end of that signal? Just like in the human QT, we ended up using a slope method. So where we developed our own program where we export the cardiac side data and then use this, essentially put a tangent there that intersects with the isoelectric line. This is an algorithm that comes from human QT analysis that's been developed, because in drug safety, these QT studies are I mean, there's a huge industry, I mean, and development that went into trying to determine the end of the T wave. Yeah. How do you determine the end of anything that's sort of exponentially sloped into something? And so the problem is that it's so high variability. And so going back to your question, I said, well, look, I mean, we can easily measure the impedance signal. So, well, let's forget about the QT or EFP duration. Just let's measure the width 50 of the contractility system. It didn't really work that well either. There was just too much variability. Yes, there was a correlation between certainly EFP duration and the duration of the contractile, but then there's just some wells where had very large contractions, other had small contractions, and it was not less variable than at least in our hands, than the EFP duration. Yeah. And since I wanted to publish this at some point, and it was easier to simply report the EFP duration rather than to tell the reviewer that, oh, you know, we had trouble measuring the electrical activity. So here we are reporting the duration of the contractility, which is, yeah, anyway. So, and I don't have a good solution to it. And we've been discussing this offline, because that's right now the biggest challenge for us is that we have to do so many wells to get around the variability that the cardiac signs, which are supposed to be a high throughput system, is intermediate at best. Because we just have to do so many wells per for a line, like basically a whole plate, to just, or even more plates than that, just to define one variant. And I was hoping to be able to just a couple wells and really go through a number of variants in much faster fashion. So.
Thank you. Thank you very much. Maybe we can take a look at flexible substrates.
Absolutely. Now we should probably
possibility to look at also the maturation phenotypes of your cells that you mentioned without using pacing, and maybe seeing if that also helps.
And the flexible substrate, I mean, certainly for all the cardiomyopathy phenotypes where you have again, often those phenotypes are only evident in the cells when their cells are loaded. And so for looking HCM or DCM variants where the problem is even bigger because there's so many variants, there's a multitude of Titan variants. So patients come in from the cardiomyopathy clinic and have a variant in Titan, and Titan is the biggest gene. Everybody has variants in Titan. All of us in this room have variants in Titan, but we don't have heart failure. So it's the question of pathogenicity of those variants is even much bigger in that. And so by putting it on a loaded substrate, I would predict that this is something we need to talk about, because I think that's where there's an opportunity to use it there, and it might actually work much better as a high throughput system for those questions rather than for the long QT questions.
Thank you very much.
Thanks.
Thank you very much to both of our speakers. We also have a little present for you. So Bjorn, this is one is for you. You tried it last night. I think you liked it. And Nathan, you didn't try it, so now you can try it. Thank you very much. For everybody else here, we are 2 minutes early for lunch. So I guess if you would like to, and I guess you would, please, the food is in our common area or small conference area. So you can enjoy lunch. We will meet in 1 hour. So that would mean at 1 back here, CET, for our viewers online in other time areas or time zones. For you, we don't have lunch, but I hope you take care of that at home or at work. And thank you very much, everyone, for your questions and also for your talks to Bjorn and Nathan.
Okay, so hello, welcome everyone to the third session today, where we'll focus on solid supported membrane-based electrophysiology, SSM electrophysiology, using the Surfer platforms. So my name is Cecilia George, and I will be the chair of this session. So I joined Nanion in 2006. I'm an electrophysiologist by training, and joined the Surfer team a couple of years ago. I'm now responsible for sales. So a bit about the Surfer technology. It was acquired from IronGate about 10 years ago. And since then, Nanion has launched 2 platforms, the Surfer N1 and the higher throughput system, the Surfer 96 SE. And thankfully, during the past couple of years, we've noted a strong increase in the interest of transporter research, interest in the Surfer for scientific research, but also for transporter drug screening efforts as well. So with this approach, you record from membrane fragments sitting on an electrode surface, and thereby you gain access not only to the plasma membrane, but also to intracellular membranes, such as lysosomes, mitochondria, ER, etc., for the studies of electrogenic membrane transporters, but also ion channels, such as leak and ligand-gated ion channels. Just a brief notice before introducing the first talk and the first presenter today. I just wanted to let you know that in 2021, the Surfer team launched the Transporter webinar series, where we have a great lineup of scientists telling about the capabilities of the Surfer, ranging from hardcore mechanistic studies to transporter screening efforts. So if you're interested in learning more about that, you can check it out. It's all online. And the next webinar will come up on November 17th. So with this brief introduction, it's my pleasure to introduce the first speaker, my colleague, Andre Bathoni, presenting the talk titled Addressing Lysosomal Transporter and Channels with the Surfer, a TMEM 175 case study. So Andre studied biochemistry in Frankfurt, and already back then he had a strong focus on structure functions of membrane transporters and biophysical chemistry. He received a PhD from the Max Planck Institute, Biophysics, under the supervision of Professor Klaus Fendler, the inventor of SSM-based electrophysiology. There he studied proton-coupled sugar transporters, and he contributed to the unraveling of their mechanisms. So in 2006, Andre joined Nanion and the Surfer team, and he's responsible and the leading scientist for transporter-related scientific projects. But besides, he also enjoys supporting customers at any stage of their research or project, from assay development, data analysis, and interpretation. And with that, I hand over to you. And feel free to ask questions after Andre's talk.
Thank you, Cecilia. So I actually joined 2016. I'm not that old.
What did I say?
2006.
Okay, sure.
So since I'm kicking off the Surfer session, I actually start with some basic introduction about the technology. So as Cecilia already mentioned, we have instrumentation which is completely different to conventional electrophysiology. It's not using cells, it's using membrane vesicles or protoliposomes, and these are attached to a solid supported membrane, which itself lies above a gold-coated measurement electrode. So you are measuring lots of different liposomes at the same time on a sensor, which is about 3 millimeter in diameter. The instrument itself is actually a perfusion instrument. It performs a solution exchange to trigger the transport. Initial condition is at 0 millivolt. So in contrast to conventional electrophysiology, we don't have voltage steps to activate, but we have substrate steps, concentration gradients we apply. This is a typical readout. So you start with perfusion of a solution not containing your substrate, then you apply your substrate. This triggers the protein activity. You measure a capacitive current, which decays pretty fast in about 100 milliseconds due to the charging of the membrane. Then after a second of measurement time, you wash out the substrate again, and you see the counter transport. You wash off the substrate goes off the sensor, and you are measuring the off signal. After that, you can basically repeat with different substrate concentration. And the important thing, to understand is usually you either use the peak current or the peak integral to use as a measure for your transport rate in case of transporters or for the conductivity of channels. So what can you do with the instrument? Of course, you can measure different substrates, determine Km, Kd values, EC50 values in general. You can also do inhibitor or enhancer assays by providing these compounds in both solutions. So before you actually trigger your transporter activity, and also in the activating solution containing your substrate, and you can determine IC50 or EC50 values. What is very interesting is that you can also measure pre-steady state currents. I'm just introducing that because later on you will probably hear about them as well. For TMM, it's not so relevant because we mainly measure the conductivity. But still, as you can see here, this is a sequence of different pHs for proton-coupled sugar transporters. As you can see, for a neutral pH, the black line is a very slow decay of the current reflecting the actual transport, while at very acidic pH, transport is blocked, but you can still measure sugar binding, which shows pretty fast kinetics. So you can do also mechanistic investigations using that technique. Another important thing also related to TMM, I will show you later in the data, are artifacts. For all techniques out there, there is the discussion about what's the meaning of or the probability of false positive and false negative readout. Of course, if the target protein density is too low, or if the currents are too low, we might miss a signal. But on the other hand, you can also see that ion jumps lead to capacitive currents by interactions with the membrane. So here you can see potassium jumps in 100 millimolar range lead to some capacitive readouts, and that makes it important to have some negative control membranes to compare your actual sample readouts. So what are the instruments we were using for the TMM data? This is the Surfer N1 or single channel instrumentation. It's mainly used for all the assay development we do. You have here the 3 millimeter diameter gold-coated sensor where you attach your sample, you place it in the measurement chamber, and that perfuses sequentially different solutions across the membrane, measuring those capacitive currents. It's very easy to apply. You can exchange buffers on the fly. So we prefer that for the assay development. But then once the assay is ready, you can transfer it straightforwardly to the 96 SE, which is based on 96 well sensor plates. So we have here the possibility to measure 96 measurements in parallel. You have the possibility to, of course, also measure cumulatively. So you have the flexibility for drug screening to either measure one compound per well or do IC50s in well, yeah, depending on your needs. And with that instrument also comes the software, which enables automated analysis. So this was just the intro, the background. Take-home message: we are not measuring as in APC real steady states. We're measuring capacitive currents, and we use the peak or the integral as a readout for the channel conductivity. So now coming actually to our standard assay for TMM 175, we developed after some time of different assays we performed. First of all, we are using native lysosomes. So we have an overexpressing cell line, and we purify the lysosomes by disrupting the cells using a nitrogen power bomb just by pressure. And then we do a sucrose gradient centrifugation to prepare the lysosomal fraction. This was already shown like more than 10 years ago in that paper, the detailed protocol. We also tested another protocol by another paper preparing lysosomes, which just uses low-speed centrifugation steps. Actually, both lysosomal samples are not perfectly pure, so there are still some impurities from plasma membranes, but both are similarly pure, showing some nice bands for lysosomal markers. So this is our standard assay. Actually, in the next few slides, I'm showing you also data not using the standard assay, because I also want to show you how we actually got there. But the standard assay uses this assay buffer, pH 7.6. And then you have in the non-activating solution, which is basically the solution before you apply your substrate. This contains additionally 50 millimolar sodium chloride, making it 140 millimolar in total. And the activating solution contains additionally, instead of the sodium chloride, the potassium chloride. So basically we trigger potassium conductivity in TMEM, and the signal we are measuring reflects that potassium conductivity. So now I'm going to start with some basic characterization of TMEM, talking first about our very first trial, which already looked very promising. Again, this is not using our standard protocol and also not the cell line we are using at the moment. So here we actually have choline chloride in the base buffer and the non-activating buffer, and then jump to cesium chloride. So TMEM also conducts cesium, and what we see here is cesium conductivity in the blue line. This is an overexpressed cell line. And then we did the same assay using a knockout cell line, and you see almost nothing in that trace. So we know this is really the TMEM activity we are measuring. We then extended this kind of assay to other monovalent cut ions, not only measuring cesium, but also potassium and rubidium and sodium as well. And as you can see, in blue is the overexpressed cell line. Cesium, potassium, and rubidium are similarly conducted, while sodium is not conducted. Interestingly, if we look at the knockout, the red bars here, all the monovalent cations show a pretty small background current, and the background current also matches the sodium current and the TMEM. So there is not a single, not the smallest sodium conductance we can actually measure with TMEM. The fact that the sodium artifact here with the knockout is basically the same as with cesium, potassium, and rubidium, makes it possible to use the sodium jump against choline as an internal control when you just look at TMEM activity. So basically you can subtract your potassium conductivity from your sodium conductivity, which actually measures the potassium artifact, and then you get your pure TMEM signal. So we made use of that. We've measured, this is done all from the same sensor, basically. We measured the cesium jumps as well as the sodium jumps. Cesium jumps, you see different concentrations here in black, sodium jumps in red. As you can see, that's what I mentioned in the introduction about the artifacts. Ion jumps do generate some capacitive currents reflecting ion binding to the membrane, but these can be subtracted from the actual sample membrane, leading to the greenish curve here, which actually shows the pure TMEM activity. And you can see that it's actually saturating in the range of 200 millimolar concentration jump. This is still work in progress. It was just done actually beginning of this week. So we also wanted to test proton conductivity, since there was some rumor that TMEM actually also conducts protons in a pretty high degree. So for protons, you need to know membranes are naturally leaky for protons, right? If you have an artificial membrane system, pH gradients are not perfectly stable. Protons go through, and these generate, of course, artifacts. So what we need to do here, since these artifacts are larger compared to potassium artifacts, we need to really have the control membrane measured and the TMEM sample measured, and then subtract. And this is basically the subtraction of TMEM minus control for different delta pH jumps. Starting pH was always 7.2. And as you can see here still, I mean, standard deviations are still quite high. This is, again, work in progress. But here you really see significant proton conductivity, TMEM specific, and also this can be inhibited with 4AP, which is known as described as an inhibitor for TMEM. So this is quite promising. We also looked on the road to the standard assay to the pH dependence of TMEM activity, and that pH dependence is shown here. The experimental workflow was that we equilibrated internal and external pH in the lysosomes to the given pH. So this is basically symmetrical pH inside and outside. What we find that the pH optimum of something around 8, and then you see a reversible inactivation with that PK here. So this is actually a regulatory binding site. And then you see irreversible inactivation with a PK of 4.4. So now I should say these results are effects of external pH, not symmetrical pH per se. So meaning, of course, TMEM is stable regarding the lysosomal site at that pH. I will show that in the next slide. But it's quite sensitive to pH from the external side, which makes sense. This is a cytosolic side. Here we looked at pH gradients. So we tried to mimic the lysosomal pH conditions. So outside, there was always pH 7.2. Lysosomal pH with that data point, I did the normalization, was also pH 7.2. And you can see if we acidify down to pH 4.5, you even get a slight increase in potassium conductivity. If you go beyond that, you will again lose activity, but this is a consequence of the procedure because we need to equilibrate the internal pH by incubation with external pH. So this again is actually a delayed response to the external pH. So now we understood pH dependence and what ion jumps reflect the nicest currents. So this was basically when we established the standard assay using the 50 millimolar potassium jump at pH 7.6. And this standard assay was then used for some compound measurements. So first I wanted to show you or get you an idea about how a kind of standard assay using the Surfer 96 SE could look like. So you have a compound plate. We can measure like 4 different inhibitor concentration, 4 different inhibitors or enhancers compounds at each 6 different concentrations using repetition of 4 wells per concentration. Usually we use sample membrane containing TMEM and then 1 line control membranes on top and on bottom so that we have n equals 3 for the actual TMEM per concentration and n equal 1 just as a control. So in that assay, we can get 4 IC50 curves basically within like half an hour. And amplitudes are very stable. Success rate is above 95%. I mean, again, we have here a very stable sensor, solid supported membrane. So attaching the sample basically works every time. It's not like that you have huge efforts for assay development regarding getting cells nicely sealed or something like that. And also the consumption of compound is reduced in our standard protocol to about 30 microliter of 10 millimolar stock per compound and run. So this is like one workflow, one sequential workflow we applied. First, you have the baseline recording, then 3 times activation using 50 millimolar potassium. And as you can see here, the signal is quite stable across repetitive substrate jumps. Then we have a compound pre-incubation. You don't need that every time. You only need that for compounds which need to insert into the membrane and act from the membrane side. If compounds are soluble, you can actually skip that. After the compound incubation, we have 2 times against the activation with potassium, but here in presence of the compound. Here an enhancer is shown. Here just a different well with the control is shown, where of course no compound, just DMSO is present, so nothing is changing. At the end, we again do the 3 times activation without compound. So you see this compound here is washed off. Not every compound can be washed off. It depends again on the solubility. If it sticks to the membrane, then this will not happen, and you of course cannot measure then another compound on the same well because it's contaminated. But if you can wash it off, you can actually start with the same well again and use another compound. So we then applied these protocols to SYNC and 4AP, which are described as pore blockers, and we could see nice inhibition curves. Again, this is all the standard protocol measuring potassium conductivity. We also tried SC79, which is described as an enhancer, but we could not see an effect. This is due to the fact that SC79 is not acting directly on TMEM175, but actually through protein kinase B, which is not present in our in vitro assay. So actually we confirmed that there is no direct effect of that compound to TMEM. Finally, we also have some enhancing compounds as work in progress. And here is just a slide of very preliminary experiments. As you can see, we didn't fit the correct concentration range here. So this still has to be optimized, but we can see some nice enhancements for some compounds. So to sum everything up, the surfer is a really good tool to detect TMEM175 conductivity induced by different cations, including protons. Proton is still work in progress, but this is in general doable. We had a detailed look at the pH dependence and found 2 different inhibitory effects of intracellular pH. We also checked inhibitors and also enhancers, could reproduce some data from literature. And we think that using our Surfer 96 SE, the assay is suitable for secondary drug screening approaches. So yeah, I thank you for your attention. And I also want to mention the people who actually did the work. This was completely done non-ion internal. And the actual measurements was done by Maria, Nina, Valentin, and myself. And I also would like to thank the rest of the Surfer team. Thank you.
Thank you very much, Andre. Are there any questions?
Thank you. Great talk. I've got 2 questions. The first one is, I'm just wondering, with your lysosomal purification, is it whole lysosomes or is it fragments of the lysosomal membrane that you're attaching the lipid layer on the
Good
electrode?
question. We don't know for sure. We believe that using the cell disruption method, we are using mainly breaks the cells, but not the very small lysosomes, which are 200 nanometer in diameter. So I would say yes, these are intact lysosomes, but we don't have a measure for that at the moment.
Okay, and with the second question is more about TMM175 itself. Do you think, considering the potassium concentrations in the cytoplasm versus the lysosome, it's considerably greater in the cytoplasm, do you think it's more likely that TMM175 will work as a proton or a potassium transporter?
Well, yeah, I mean, we lack the possibility to do pH jumps with these huge delta pH, because as you see, I mean, even delta pH of 1.5 makes some noise of nanoampere range. But if it goes linear, then proton conductivity is much higher than potassium conductivity, in our assay at least.
Thank you.
More questions? There is another question here. Do pH gradient affect compound effect on TMM175?
Yeah, so we checked for that, actually only for a few compounds, but as far as we can tell, there's no large effect on IC50 values we determined when we have just pH 7 inside and outside compared to like pH 4.5 inside and pH 7 outside.
Okay, thank you very much.
Thanks.
Andre. As you see from the program, the next speaker should have been Professor Ulrich Hammes from the University of Freising. He unfortunately couldn't make it. But for those of you who are interested in Ulrich's research on the pin-formed auxin transporters found in plants, which he recently published in Nature, I highly recommend that you find the webinar that Ulrich and Björn Pedersen made for us with us previous this year. I am also very thankful and impressed by our next speaker, Emanuela Colucci, who with very short notice was able to record a presentation and step in and contribute to the user meeting. Thank you very much for that, Emanuela. So about the next two speakers, they have a couple of things in common. First, they both are pursuing their PhD studies. They both work with glutamate transport, and both started using the Surfer N1 merely a year ago, which I find is very impressive. So hello, Emanuela, are you there? Would you like to say hello?
Hello, yes. Thank you
Hi,
for having me.
very much for joining. So I'll introduce you. You have a Master of Science in Molecular Biology from the University of Parma. And during a Erasmus-supported internship in 2017, you visited the University of Groningen, where you contributed to the findings of vitamin B transport. Emanuela continued with her PhD studies in the X-ray crystallography and membrane enzymology group under the supervision of Professor Dirk Slotboom, working on both structural and biochemical characterization of glutamate transport. They use a combination of cryo-EM, X-ray crystallography, and assays designed to study kinetics of transport. Emanuela will present part of this research, including the use of the Surfer N1 for the characterization of glutamate transport. And the title of the talk is New Insights into Mutations Leading to Episodic Ataxia 6 by Using Archaeal Protein as a Model, GLT-TK. I hand over to you, Emanuela.
Everyone, my name is Emanuela Colucci, and I just finished my PhD in the groups of X-ray crystallography of the Professor Albert Kuskov and the membrane enzymology group from Dirk Jan Slotboom. And part of my research has been around the characterization of the archaeal homologue of glutamate transporters, GLTTK, and we used it to have new insights into a mutation that in mammalians is responsible for episodic ataxia 6. So first of all, why glutamate transporters are so important? Glutamate is the most abundant neurotransmitter in the brain, and it is responsible for activating a series of different neurotransmitters. Its concentration is it's the homeostasis of its concentration is very important, as a low or high concentration of glutamate at the synaptic cleft are responsible for different neurotoxic conditions and different diseases. As for the proteins that are responsible for keeping this concentration constant and at levels that are compatible with life, there are the EATs, which stand for excitatory amino acid transporters, that are differently expressed in different parts of the brain, and we find different homologues in presynaptic, postsynaptic terminals, and also Bergman glia. EATs have been widely studied, as I said, for their importance in keeping this concentration constant, and their malfunction is associated often with diseases, pathological conditions. For the EATs, they are able to symport 3 sodium ions together with 1 molecule of glutamate. They can transport 1 proton and counter-transport 1 potassium ion, and they are capable of creating a thermodynamically uncoupled chloride conductance. Most of the information that we have for the mechanism and the structure and all the other information come from the study of the archaeal homologue GLTTK and GLTPH from Thermococcus kodakarensis and from Pyrococcus airoshi. These proteins share very low sequence identity with the EATs, but they are capable, they have the same structure, and they are capable of transporting 3 sodium ions together with L-aspartate, for which they have a higher affinity than the L-glutamate, and they are capable of creating a chloride conductance. So the study of these proteins brought us to the resolution of the structure that consists of a homotrimer, in which each monomer is constituted by a scaffold domain represented here in yellow, that is also called the trimerization domain, as it is responsible for the contact with the other monomers, and a transport domain that contains all the elements for the substrate binding and the transport that happens from the outside to the inside. This mechanism has been obtained and has been by the different states that were resolved in the different proteins, and we know that each monomer behaves independently from the other and goes from the outside to the inside of the cell with a movement that is being compared to an elevator. So the transport and binding of the amino acid is strictly dependent by the binding of sodium ions. So here we see the sequence of events. So the transport domain exposes the binding side on the outside of the cell, and it can bind two sodium ions followed by one molecule of aspartate, and then after the last the aspartate binds, the last sodium ions can enter. After the closing of the binding side, the transport domain goes through the inward facing state in which the substrates are released, and the transport domain can undergo again through the same cycle. In between, we know from electrophysiological studies that in between the outward facing state and the inward facing state, there is a chloride conducting state. That happens probably thanks to the lateral movement of the scaffold domain that allows the formation of the channel in between the two domains, but for which we still miss a structure. This double function of the glutamate transporters of glutamate and or aspartate and chloride conductance is very important and very well coordinated by these proteins, but a single mutation, for example, the mutation from proline to arginine at the position 290 in E81, is responsible for the dramatic change in this behavior, as this transporter cannot transport glutamate. The glutamate transport is heavily reduced, while on the other hand, the anion conductance is increased. To study this, why this happens, which is striking from a biochemical point of view, because as I said, the chloride channel formation is substrate gated. So only when the transport domain goes inside the cell, there is a formation of the channel, and this happens only when the transport of the other substrates is happening. We wanted an explanation from a structural point of view. So we wanted to see what implications this mutation has on the structure. And as you can see here, the proline is very well conserved throughout the family, and it is responsible for formation of a kink in the transmembrane helix 5. And some people have hypothesized that this change from proline to arginine could disrupt this kink and then also could induce rearrangement of the architecture, therefore impairing the transport of glutamate and chloride conductance. Therefore, we used GLTDK as a model. As explained before, it has been used to obtain many information, and also being thermostable is possible to express it in high amount in E. coli, and it can be purified in high amount in detergent. And for the biochemical studies, such as studies also to study the transport, it gets reconstituted into protoliposomes. Before ensuring that this model was good to study the structure implication, we also wanted to validate GLTDK as a model by confirming the presence of the chloride channel, as it has never been proved in GLTDK, but only in the other homologs. And in order to do this, we started from optimizing the electrical signal, and we started from the signal that is obtained by the co-transport of aspartate and sodium. So being in the ratio 3:1, so 3 being the sodium ions and 1 being the aspartate, the electrical signal that derives from it is positive. So we went through the optimization of the right lipid to protein ratio, in which 75 was the best ratio to obtain the best signal. Then we also optimized the right concentration of the protoliposome to get absorbed on the layer for our studies, and the type of buffer to exchange the different solutions for the experiments. And we see that phosphate buffer works better than the chloride buffer in our cases, also because chloride probably is one of the substrate, and its negative electrical signal might affect the positive one connected with the aspartate and sodium transport. So we used a double solution exchange. Here you can see the composition from a resin solution that contained potassium. The resin solution is the solution that is contained in the protoliposomes and outside the protoliposomes, gets exchanged with a non-activating solution with an increasing concentration of sodium. In the activating solution, we have the same non-activating solution, but we also the substrate to create the jump. And in our case, it's a fixed concentration of aspartate. And then it gets again replaced with the non-activating and the resting solution. By increasing the concentration of sodium, we obtain that with the increasing concentration of sodium, the amplitude of the signal increases. And by plotting these peak currents and using Hill equation to fit it, we obtained an apparent affinity that is in the same millimolar range of the ones that were measured with different experimental setup in other studies. Confirming the function and stability of this type of protoliposomes that we used for the assay by the aspartate and sodium transport, we also wanted to, we proceeded with optimization of the signal obtained by the anion current. And in our case, of course, the anion transport creates a negative signal. And we went through some different anions, and we selected thiocyanate as an ion of choice over nitrate and chloride. And this is consistent with literature, with other homologues that see thiocyanate as the most permeable for this type of transporters. Also, we studied the effects of the presence and absence of the substrate, and as you can see, comparing with the case in which both aspartate and sodium in the sodium thiocyanate are present, the absence of aspartate and the absence of sodium don't allow the detection of any signal that is different from the artifact that is obtained on empty liposomes. We use the same experimental setup that I explained before, the same double solution exchange experiment to determine the kinetic parameter for the sodium dependence of thiocyanate transport. In this case, aspartate is being included in all the buffers, and in the activating solution, there is a fixed concentration of also thiocyanate. And also in this case, we use increasing concentration of sodium. In this case, we also see that increasing the concentration of sodium, we have an increase of the amplitude, and with the same idea, we plotted and fitted with the Hill equation and obtained an apparent affinity, which is again in the same range of the one measured for aspartate. These results altogether confirm not only that the chloride channel is formed, but also that it is strictly dependent by the transport of aspartate and therefore is defined as a substrate gated. At this point, we use this tool and this experimental setup to study the implication of the mutation from proline to arginine, which in GLTTK is at the position 208. In the case of the jump of L-aspartate, we see a dramatic change in the transport, and we see that the electrical signal, positive electrical signal given by the aspartate in the mutant is heavily reduced, while for the anion conductance is slightly increased. We plotted these peak currents and we determined this sodium dependence in both wild type and mutant. And we see a dramatic effect as in the mammalian homologs for the aspartate transport and slight increase in the anion conductance. And this difference is even more visible if we compare the ratios of the induced currents of thiocyanate over L-aspartate. Hinting to the hypothesis that GLTTK is indeed a good model to study this, the effect of this mutation, because also it gets logical to think that it is logical to think that with the lower transport of aspartate, the anion conductance might be affected in any other situation. And also this problem is also not directly in the binding site, but is in the scaffold domain, which also poses the question as for why this impairment. And we try to explain this by looking at the structure. So we obtained a structure of the mutant at 3.25 angstrom resolution of the protein embedded into nanodiscs. So in the case in which the protein is in the lipidic environment, and we also were able to solve the lipid densities. And as you can see, the replacement of the proline at the disposition with an arginine creates a salt bridge that might be responsible for keeping the chloride conducting state more stable. As I told you before, the chloride channel is formed in between the transport and the scaffold domain, and this salt bridge could stabilize the channel formation, explaining why the anion conductance both in GLTTK and in the human homologs is increased while the amino acid transport is reduced. To conclude, SSM was a valid tool to confirm the kinetic parameters of aspartate transport that we also could measure with other experimental setups, such as radio-labeled uptakes. It was, SSM was useful also to confirm the presence of the chloride channel in GLTTK, which is unprecedented as it was confirmed only in the mammalian and in GLTPH, but with another experimental setup. The mutation from proline to arginine at position 208 in GLTTK is able to create effects that are similar to the ones that are measured in the mammalian homologs. And also, we used the structural tool to reveal that this arginine creates a salt bridge that could lead the protein to linger in the chloride conducting state for longer, impairing therefore the aspartate transport. And with this, I would like to thank the people that collaborated with me, from my supervisors, Albert Guskov and Dirk-Jan Slotboom, and our postdoc, Milligan, who was responsible for bringing the server Nanion to our labs, and also contributed a lot for these experiments and for the designing. And Zaid, who is a PhD in our lab, who helped with the optimization of the experimental setup. And I would like to thank you for your attention.
I would like to thank you. I would an extra big hand again for Emanuela for stepping in with such short notice. Thank you so much. Thank you. Thank you. Do we have any questions? I got one. Can you rule out the possibility that the reduced transport current in the P208R reflects a reduction in reconstitution efficiency?
Yes, so for every experiment, before the experiments, we always loaded preparation of our protoliposomes in STS gels, and we compared reconstitution efficiency of the wild type and the mutant every time. So we assume that reconstitution efficiency is the same for both.
Okay. Does this mutation have any effect on the protein stability? Did you study that?
Yes. So from both a structural point of view and also the purification of the protein was always with the same efficiency as the wild type, and also we ran other experiments that I didn't show here, where we also measured the affinity for the of this protein for the aspartate, and it's in the same micromolar range as the wild type, meaning that the whole protein is stable, and the only thing that is affected is the transport rate when measured in the protoliposomes.
All right. Excellent work. Thank you very much. Thanks once again.
Thanks. Bye-bye.
And now the third and final speaker today on the server. We're happy to have Natalia Dmitrieva here. A warm welcome. So Natalia has a Master of Science from the in Biophysics from the Moscow Institute of Physics and Technology, where she worked on the heterologous expression of retinal proteins, membrane protein crystallization, and structure determination. In 2019, Natalia joined the research group of Professor Christoph Balke to pursue her PhD studies at the Institute of Biological Information Processing, Molecular and Cell Physiology at the Research Center in Jülich, Germany. The focus of Natalia's PhD studies is the molecular basis of the function of vesicular glutamate transporters, and the title is, as you see, Exploring the Transporter Cycle of DGOT, a bacterial homologue of vesicle glutamate transporters. Thank you very much.
Thank you for the introduction. So yeah, today I will speak about transport cycle of DGOT. SLC17 family is a group of transporters that are involved in accumulation of anionic neurotransmitters such as glutamate or nucleotides. The family includes lysosomal sialic acid transporter sialin, vesicular glutamate transporters VGLUTs, and a vesicular nucleotide transporter. Their bacterial homologue is D-galactonate transporter DGOT, whose crystal structures in 2 different conformations were resolved recently. DGOT shows striking structural similarity with glutes. Both proteins belong to major facilitators superfamily and share common for MFS 12 transmembrane helices fold. There are only 4 charged residues in transmembrane region of DGOT, and with the exception of D46, they are highly conserved in glutes. It has been proposed that DGOT transports galactonate together with at least 2 protons, and it has been shown that mutation of either of 2 negatively charged residues or arginine 47 leads to loss of transport activity of the protein. We wanted to understand transport mechanism of DGOT in more details, and for that we used a combination of experimental and computational approaches. And in my talk today, I will try to show how SSM experiments and optimistic MD simulations complement each other and allowed us to reconstruct a detailed model of DGOT's transport. For our SSM experiments, we used purified protein reconstituted into protoliposomes. The technique itself has been introduced by Andre, so I don't have so much to add here. I will only briefly mention that for the results I will show today, we will also be interested in differentiation between transport currents and fast pre-steady state currents. When activated by substrate concentration jump, DGOT demonstrates transport activity in a wide range of pH conditions. This experiment we did with 3 mm sensors, and we saw nice large transport currents. Transport activity is inhibited by extreme alkaline pH, and this effect can be attributed to proton depletion outside of the vesicle. Transport is also reduced by acidic pH, but in contrast to what we saw at alkaline pHs, here fast pre-steady state currents dominate at pH below 6. And that suggests that in this case, transport activity is limited by impaired proton release inside of the vesicle. If we plot normalized peak currents against the pH, we can get estimates of apparent proton binding and release. We measured transport stoichiometry with reversal potential assay, and for that we used the idea that for coupled transport, substrate or proton gradients can be used as a driving force for the transport. So this way, by combining these gradients, we can observe either negative or positive transport currents. When these gradients are balanced, then the transported charge as well as free energy for the transport reaction should be 0. So in our experiments, we kept proton gradient constant and varied substrate concentration gradient, and we were able to drive transport activity in different directions. Then to analyze transported charge, we should make correction for solution exchange artifacts, because since we changed the pH, these artifacts are not negligible. So we performed experiment with exactly the same solutions, but with empty liposomes. So after we make this correction, we can plot transported charge against ratio of chemical gradients. And x-intercept of this plot gives us our transport stoichiometry, which is indeed 2 protons with 1 galactonate molecule. So now we will try to understand how this transport works at atomistic level. As typical MFS member, DGOT uses alternate access mechanism to perform its function. Members of the family undergo major conformational changes to allow access to central binding site from either side of the membrane, but not simultaneously. As has been shown for many family members, these major conformational changes are coupled with local rearrangements of flexible gating helices 1 and 7. And in our simulations, DGOT behaved consistently with this model, and its TM1 and 7 appear to be indeed quite flexible. In fact, there are 2 pairs of gating helices that gate access to the binding site from different sides of the membranes. From periplasmic side, it's a pair of TM1 and 7, and from cytoplasmic side, it's TM4 and 10. and the distances between the most flexible parts of the helices, which are shown here in red, we will call gates distances. To study binding of galactonate, we performed all-atom unbiased MD simulations with outward-facing digot structure and checked whether in these simulations distance between TM1 and 7 would change. And we noticed that if we manually protonate either E133 or D46, distance between gating helices increases. And this effect is even larger when both residues are protonated at the same time. So it might seem a bit counterintuitive how gate distance is influenced by protonation of the residues that are relatively far from the gate itself. So if we take a closer look, we can see that deprotonated glutamate 133 interacts with arginine 47. And when we protonate manually glutamate, this bond breaks. And apparently it gives arginine 47 more room to interact later with negatively charged substrate. A similar interaction we can find between deprotonated D46 and another arginine 126. When we protonate D46, this interaction is disrupted, and arginine instead interacts with asparagine located on the same helix as aspartate. So this interaction leads to rotation of whole TM1, and apparently it's part of gate opening mechanism. So now, assuming that double protonated protein with completely opened preplasmic gate precedes substrate binding, we performed the same simulation, but added the substrate to the solution in our simulation box. And here we again observed opening of preplasmic gate, and shortly after, substrate enters binding site. After substrate enter binding site, gate closes again. And we also checked if the gate would close if we keep one or both key residues deprotonated. And it seems that it's also found in closed conformation. So substrate binding induces this local rearrangements and leads to preplasmic gate closure. We studied galactonate release from inward-facing structure with similar approach. So here we performed unbiased MD simulations with inward-facing structure, with substrate bound, and changed protonation state of key residues. And what we saw here that substrate does not leave binding site when both key residues are protonated, and instead all spontaneous release events were observed in simulations where D46 was deprotonated. So we can conclude that at least deprotonation of this residue is needed for a successful substrate release. So yeah, finally, the only part of transport cycle that remained undiscussed is major conformational changes. To describe them, we can follow the movements of gating helices, and if we plot distances between two pairs of gating helices against each other, we can, in this representation, easily separate inward, outward facing, as well as occluded conformations. And what we saw is that with our unbiased simulations, by modifying protonation state of key residues and addition of substrate in the binding site, we were able to explore almost whole conformational space. So also we can assume how major conformational changes would look like projected on this space. So all these observations are summed up in this transport cycle model that we propose. Here we go clockwise, starting from the left. We will start, as I showed on previous slides, with binding of protons, then binding of substrate, then major conformational changes and substrate protons release, and we go back where we started. Then we performed SSM experiments with different mutants, and for this experiment we used 1 mm sensor to have better time resolution. So first mutant is E133Q, which showed no transport activity, but selective binding of substrate remained intact. Peak current did not really depend on pH, and overall these pre-steady state currents resembled what we saw in wild type at extreme acidic pHs. So here we assume two-step reaction, where first step is substrate of the binding to the protein, and second step is electrogenic conformational changes. Rates of recorded pre-steady state reactions can be derived from an exponential fit of current decay, and they depend hyperbolically on substrate concentration. So from this fit, we can extract KDs and forward and reverse rate constants for second electrogenic step. And we can see that for this mutant, all these parameters are similar to wild type at acidic conditions, although mutant seems to be a bit faster than wild type. Next mutant was D46N. Its current looked quite different from the other one. These currents were much slower and they were highly pH dependent. And since they were very slow, it was not obvious whether we observe some remaining transport reaction or it's pre-steady state one. So we performed the same experiments with different liposomes with different LPRs. And if for wild type protein, we saw systematical dependency of decay times on LPR, that was not really the case for this mutant. So we concluded that observed reaction is slow pre-steady state reaction. Similarly, we can fit rates of observed reaction, and we can see that forward and reverse rate constants for conformational changes are smaller than those for wild type, but KD remained quite similar to wild type, which makes perfect sense that substrate binding was not disrupted in this mutant. R47 co-mutant looked completely different from the first two. So since this arginine interacts directly with negatively charged substrate, we expected that this mutant would not be able to even bind substrate, but instead we measured very fast pre-steady state negative currents. And what we think the interpretation is that we can observe in this mutant fast binding of galactonate, so only first step of our reaction. And apparently this arginine is not strictly needed for substrate recognition. To check this hypothesis, we performed simulation with this mutant and compared with simulations with wild type protein. So in wild type outward facing conformation, substrate in the binding site interacts with arginine 47 and periplasmic gate is found in closed conformation. When we perform simulation with mutant, substrate is slightly shifted from TM1 and our newly introduced glutamine interacts with protonated E133 and arginine 126. And periplasmic gate is found in open conformation. Another way to illustrate conformational changes is to project our simulations on conformational space introduced before. And what we see here is that wild type outward facing apoprotein is found in the most open conformation, and upon addition of substrate in the binding site, we see that conformations are shifted towards occluded conformations. What we see with the mutant is apoprotein occupies similar conformations as wild type, but there is no change in these conformations when we add substrate in the binding site. So this observation somehow support our conclusion made out of experiments that in this mutant we indeed can observe only first steps of fast electrogenic substrate binding, but not following conformational changes. So to sum up, we demonstrated that DGOT mediates a symport of galactonate and protons with 1 to 2 stoichiometry. We confirmed that proton transfer indeed takes place through D46 and E133. However, if protonation of these residues doesn't take place, we still can observe binding and following conformational changes, but not transport activity of the protein. R47 is apparently not strictly required for substrate recognition, but is needed for gate closure and following conformational changes. And we believe that this mechanism might be shared within MFS, since this arginine is highly conserved in the family. So with that, I would like to conclude. I would like to thank Christoph Falti for supervising this project, all my colleagues who were involved, and Andre for some useful discussions on currency interpretation.
Thank you very much. Thank you. Very nice presentation. Questions? I have one. Do you see large conformational changes in your simulation, for instance, between inward, outward facing configurations?
So we do not really see major conformational changes within one simulation, because this event happens on timescales larger than our normal length of a single replica. So to describe these conformational changes with simulations, we use some enhanced techniques to be able to explore this conformational space more extensively and then analyze what we see.
Can you somehow determine what residues are protonated first?
That's also not so straightforward. At least it's not so obvious from the experiments. It's not clear whether one should strictly be protonated first or second, or it's somewhat independent events, and the same applies for protons release, because yeah, from point of view of the symmetry, we would assume that first both protons should be released, and only after that the substrate.
Okay. Yeah. Thank you very much. Thank you. Okay, and yeah, so coffee break, and we join at 12:30 again.
Yeah, so if everybody can hear me, we could start with the last session. So who wants to join us in our conference room should enter now. Right. Yes, so welcome everybody to the last session of this year's user meeting. Last but certainly not least. So moving even further away from living cells, we've now seen some research done on solid supported membranes. We will now see some examples of research performed on freestanding lipid bilayers, which is done on our Orbit devices. My name is Konrad Weidroth. I'm the product manager of the Orbit devices and the vesicle prep as well, which makes me the bilayer guy. So if anyone is interested in research involving these bilayers, please feel free to approach me. And our first speaker today will be Adam Dorey. He received his bachelor and his master degree and also his PhD from the University of Southampton. And he then went to the University College of London, where he joined Stefan Hovorka's group, who is mainly dealing with tailor-made DNA origami, for example, nanopores. And he will today give us a really nice example of how you can exploit these pores in particle detection on a single molecule level.
Thank you very much for the introduction, Conrad. Yeah, so I'll be talking about something a bit different today to what we've already heard. So as Conrad said, I'm going to be talking about work that we do in our lab, where we try and custom design pores that we make out of DNA and use these for sensing of diagnostic proteins. So biosensing is important in a number of areas such as patient diagnosis, personalized medicine, food safety, and forensic science. However, traditional biosensing platforms often require large pieces of equipment that are often very expensive and require technical specialists to use. So there is a need for sequencing and sensing to be cheap, portable, fast, and easy to use. So a good example of this is the Nanion from Oxford Nanopore Technologies. So here we can get, just on this small USB device, it can be used for DNA sequencing. We can get nice, long, specific reads using this USB device. The main technology in this device is nanopores that are stably embedded in membranes. So how do we actually do sensing and sequencing using nanopores? So nanopores, as you all know, permit the flow of ions. So what we can actually do is hijack this function to pass through small molecules such as DNA or short peptide strands. So what we actually do in our lab is we use the orbit devices and we use planar lipid bilayers. So just a brief overview for those who have never done these experiments before. These involve a cavity, microcavity, two microcavities that are filled with buffer, and they have an electrode in each microcavity. We can then form a membrane that separates these two electrodes, and then we can insert our pore of interest. So the main advantage of doing this is we can completely control everything that we do here. So we use purified lipids, so we can control the lipid environment. And we use purified protein, so there's no influence from any other proteins. And also it's a single molecule. So we're just recording the electrical output of one single protein pore. So how do we actually analyze and sense analytes? So first we get a background recording just in the presence of just our inserted nanopore. We can then add an analyte that is small enough to fit through the lumen of these protein pores. And as these analytes pass through the lumen, it impedes the flow of a certain number of ions, and we get these characteristic blockade events in the ionic current. Now, these ionic current blockades will be dependent on the analyte size, shape, and charge. So you can actually get a lot of information out of these recordings. And this is what occurs in the Nanion. So they have thousands of these stably embedded nanopores. The translocation of the DNA is tightly regulated by this helicase. These pores are heavily engineered. And this means that as the DNA passes through, each individual base has a slightly different structure. And we can actually see slight differences in the electrical readout that allows us to get some quite high highly specific nuclear base reads. So DNA sequencing and small molecule detection is very important. But there's also a need to detect folded protein. So typical protein pores are not suitable for this function, as their lumens are too small to actually fit these much larger molecular cargo. So this brings us on to what we do in our lab, where we actually construct artificial nanopores that are large enough in diameter to accommodate these much larger protein cargo. And we actually construct these using DNA. So we use a technique called DNA origami. And basically, we use the predictable folding of DNA to actually create these nice two-dimensional and three-dimensional shapes. So the method that we use is we take a large scaffold strand, so a large ring of DNA, and then we target smaller staple strands, and this kind of targets and tells the long scaffold strand how to fold. So there are many advantages of using DNA origami over trying to produce de novo protein design nanopores. First one being that these are relatively easy to design. There are many computer software available that we can actually design these structures, and it will tell you what strands you need to buy and to order. Secondly, they're very easy to actually fold. You don't need to go through complex expression and purification. All we need to do is get the strands, put them into a PCR tube, run them on a PCR cycle that heats them up and slowly cools them over time, and these structures will self-assemble. Another main advantage that I'll come on to in a bit more detail later is you can easily add modifications to these structures. So this is just a short example of how DNA origami, how we actually form these structures. So we've got this long blue unregulated, unstructured DNA ring, and you can see how these smaller staple strands are bind into it, and it will eventually turn it into this nice structured rectangular shape. So this brings us on to a paper that we published in Nature Nanotechnology this year, where we wanted to create some size and shape tunable nanopores constructed out of this DNA origami. So on the left here, we've got alpha hemolysin, which is a typical protein nanopore, and this has a luminal area of about 1.5 nanometers square. Our structures, so we've got structures ranging from a small triangle of 43 nanometers square luminal area up to this larger square structure of 400 nanometers square. So we can see that we can not only just change the luminal size, but we can actually completely customize the shape of the nanopore as well. Now, what we have to do to get these to actually bind and insert into lipid membranes is we have to litter the underside with cholesterol anchors. So this means that the pores can now bind to membranes and then hopefully penetrate the membranes as well. So first we need to confirm that we're actually folding and achieving the desired size and shape pores that we have designed. So we do this with gel electrophoresis. Once these structures have been folded in the PCR cycle, we purify them from the excess staple strands. And what we should see if the pore is folded correctly is we get a nice single band. And this is what we see here. And we can see that as the structures get bigger, the band is higher as it won't flow as quickly through the gel. And we also corroborated these with some TEM imaging. So you can see how the size and shape of these structures relates to the design shape. And this is particularly apparent in the large triangle and square here, as there's more DNA here for the stain to bind to, so we can actually see these a bit clearer. I then performed some electrical characterization using the Orbis 16. So we used the small triangle, small square, and larger square. And what we're expecting is as the luminal area increases, we'll have an increased flow of ions through these, and the conductance will therefore increase. And that's what we see here. So we can see that the conductance increases from the small triangle to the small square, and then up to the large square. So now we've got a background electrical characterization of these. What we wanted to do was to add some modifications within the pore lumen that will allow us to target protein and light. So that's what we did. In this green here is a receptor. It's a biotin modification, and then we could target antibiotin antibody to this receptor. So this is what we see in this current trace here. So before the arrow is just background, just the inserted pore with no antibody. And this gray arrow indicates the point of antibody addition. And what we see are these nice blockade events as the antibody is binding and dissociating from its receptor. So we can see how we can produce these pores to give us some nice characteristic sensing ability of antibodies. So we want to take this a step further, and we actually collaborated with Oxford Nanopore Technology to try and design a pore that we could insert into their flow cell membranes. And we wanted to try and sense an antibody that's a bit more clinically relevant. So we designed this triangle, large triangle structure. We managed to get insertions into their flow cell membranes. This is the first time this has been done with a DNA origami pore. And we included a modification within the pore lumen that related to COVID-19 spike protein. We could then target anti-spike antibody. And what we're hoping to see is that we get some blockade events so that we can actually sense this protein, sense the COVID-19 antibody. And again, this is what we see here. So again, gray arrows are the point of antibody addition. And we get these nice characteristic blocks as the antibody is binding to the pore. So it's a nice way to show that we can custom design these pores and include modifications that allows us to sense clinically relevant proteins. Simultaneously, we're also working with a group in Arizona State University, and we published this paper in Nature Communications this year. In this study, we wanted to create a similarly large-scale nanopore constructed with DNA, but we wanted to add a reversibly gated lid to this pore. So in blue is the large nanopore, and in red is a lid structure. And this can be reversibly opened and closed through use of key strands. So what we can see here are some nice AFM images. So on the left is the pore in a closed state. You can see the outline of the lid here. However, when we add the key, we can start to see some luminal openings as the lid is now open. We also corroborated these with some fret measurements. So we've got a fluorescent tag on the lid and a fluorescent tag on the pore itself. So when these tags are in close proximity, we get high fret readout. And when the lid is open and the distance between these increases, we get a decrease in this fret readout. And what we want to show with these experiments is that this lid opening and closing is completely reversible. So by adding opening and a closing key, we can actually reversibly open and close these pores. So one of the main reasons that we wanted to have this pore with a lid is to get some controlled protein translocation. So we first did this with giant elongated vesicles. We did it with the closed lid pore. We add the pore to the vesicles and then added GFP. As the pores are closed, we're not expecting any GFP to move through the pore into the GUV. And this is what we see over 3 hours. We get no movement of the GFP into the GUV. However, when we add the closed lid pore, and then after an hour, add the opening key, we can then see after 3 hours that GFP has managed to move through the open pore and into the GUV. We also corroborated this with some electrical recordings. We did this using the Orbit Mini. We did this using trypsin. So we chose trypsin as trypsin has a net positive charge in the buffer solutions that we're using. And basically this means that because DNA has a net negative charge due to the phosphate backbone, that we get some interaction of the trypsin with the DNA strands. So this will basically impede the flow as the trypsin moves through. And this allows us to get the resolution we need to actually detect these proteins. So with the closed pore, we have a very small background current, and we see no translocation events in the presence of trypsin. However, when we open the pore lid, we get a massive increase in current as now ions are able to flow through. And we can see these nice translocation events as trypsin is moving through the nanopore. So in summary, these protein pores are very useful for high resolution, small molecule detection, DNA sequencing, but they're not suitable for much larger protein cargo. And this is where our DNA pores are really useful. But they both have potential for portable, high throughput, single molecule recordings. So I'd like to thank my supervisor, Professor Stephen Walker, and members of the Hawker Lab. Also a postdoc on our lab, Yongjin, who designed and did the TEM imaging of these pores. I'd like to thank you all for listening.
Thank you so much for this really nice update, Adam. Are there any questions from the audience?
Hi, thank you very much for a great talk. I have a little curiosity. You mentioned that you had to add some cholesterol to your things. How difficult is it to fuse those pores inside the membrane? Does it take a long? What is the success rate there?
Yeah, this is the main limitation of using these pores, is that they take a long time to insert. And this is why the Orbis systems are so useful. So the Orbis 16 has 16 channels, so I can, get 16 times the insertions I would if I was using a traditional system. So yeah, there's tricks that we have to do to try and get these to insert, especially with the large diameter ones. They really do not want to be in there, so we have to kind of force them in as best we can.
Hi, very nice talk. Thanks. I'm just wondering, when you look at these traces, you sometimes see that there's a lot of noise, added noise when the pore is there, and there seems to be some sort of dynamics going on.
Yeah.
Do you have any idea where exactly that comes from? Is that typical for DNA, or do you have a strategy for calming these things down? Because if you want to separate, if you want to discriminate between different types of analytes, you would probably like to reduce your low frequency noise.
No, this is due to the flexibility of DNA. So these things aren't rigid when they're in the membrane. You can actually see from the AFM images here how there's different states of kind of opening, and these things will kind of fluctuate between open and closed. So there are things that we can do to kind of add rigidity to these, and these are things that we are exploring. Oh yeah, much less rigid. Yeah.
All right, maybe we have time for one more question.
I'm just wondering if the properties of your pores are the same in lipid bilayer and the membranes on ONT, also on Oxford nanopore.
Yeah, from what we saw, we saw the same kind of slightly noisy channels. Yeah, they were basically the same in the lipid membrane and in the ONT membranes. Obviously, the ONT membranes were a lot more difficult to insert into because they're made out of polymer. The membrane's a lot more rigid. It's difficult to even get protein pores into their membranes. So, yeah.
All right, thank you very much, Adam. Oh, maybe one more question.
Sure.
Maybe a very naive one. So I understand the importance of the size of the pore, but you have really a lot of different shapes.
Yeah.
Do they have like reason why sometimes a triangle, why you use sometimes a square?
Yeah, I mean, for this paper, it was just to show that we could do it. But you could imagine that if you have an analyte of a certain shape, you might want to tailor it to that shape so that you get more of a block when it inserts. Or
So let's thank Adam again. So it's a great pleasure for me to introduce our next speaker. It's Andreas Borkas. He's originally from Cyprus and he went to Greece for his bachelor degree. And then he went to Berlin where he performed his master's degree, where he received his master's degree at the Freie Universitat. And he now joined the group of Ramona Schlesinger for his PhD. And we heard about optogenetics yesterday, so activating proteins with light. And what Andreas did on the Orbit Mini was not really activating the protein as such, but activating the lipid surrounding, which I find very interesting. And he will tell us about his work.
Thank you very much for the nice introduction. And a second. All right, thank you for having me also here. So my topic today is the photoactivation of a mechanosensitive channel. It's a work that we did this year in our group with collaboration with Professor Joachim Heberle. We tried to create a system, therefore I will try to go stepwise and deconstruct the whole system one by one, so we will all be on the same page at the end of the day. Starting with the basic terminology for the activation and mechanosensitivity. For activation is quite self-explanatory. You activate something by a light. Why? Because we are able to trigger biological systems and control the activity of those biological systems. There's great interest the last years because of the photopharmacology and how you can implement such methods. And I will talk specifically today about photo switches and the ASO-PC, but I will go later on, specifically on that one. And the second part of the terminology on the title was mechanosensitivity. Again, it's a response to mechanical actions and biophysical forces. Is quite important because a lot of biological sensors, such as sensing the touch, hearing, pain, and balance, they are mechanosensitive, or at least one part of them is mechanosensitive. So by studying them, we will understand better. There are a few categories about this from mechanosensitive channels, but I will focus on the MSCL. Now, to visualize a bit the MSCL, the mechanosensitive channel of large conductance, we use the one that is native to Escherichia coli. It has 16 kilodaltons size per monomer. We assume it's a pentamer. It has an alpha helical structure. It responds to mechanical stress. It's a non-selective ion channel, and it's the emergency valve of the E. coli in case of osmotic pressure. Now, on the left side of the panel is the Mycobacteria tuberculosis MSCL. Unfortunately, the E. coli MSCL structure has not dissolved yet. So just to follow a bit here, we have the N-terminus on the parallel of the lipid bilayer, then expanding the first trimembrane helix in alpha helical form into the lipid bilayer, going outside in the periplasmic loop, entering again with the transmembrane helix 2, and then we have the C-terminus at the hydrosolic site. Why are we so interested about the MSCL? It's a very well-established model. It's been studied for many years extensively, and structural and functional characteristics of the MSCL and how it's actually functioning, we can also reoccur in higher organisms. It has medicinal applications because there are drug targets that specifically for the MSCL, and also can be used as a nanovalve in nanosystem because at the fully open state of the MSCL, it has an opening of around 30 angstroms, which is quite large for a very small protein as it is. So we have the first component of the system. The second component is the photoswitchable lipids, the ASO-PC. So on the right side, just to visualize again the chemical structure of those lipids, we have the PC head group, as we all commonly know, the alkyl chain, and then on the other part of the on the other side of the lipid tail, we have the isobenzene moiety, which this specifically is our switch. By illuminating the transform with UV light, the benzene isomerizes to its C state. And reversibly, by illuminating with blue light, then we control it back to the trans state. In order to confirm that those lipids are actually reversible and we can actually control the system that we want to proceed in the future with our experiments, we did FTIR different spectra. I will not go into detail about FTIR and infrared spectroscopy because this is not our topic today. But what we can see here, the first experiment was getting the lipids at Cs, illuminating them with blue light to trans, and then we get the blue signal here as a different spectra of this transition, and then vice versa with the trans to the Cs. Although I don't want to go into details about the IR spectra, what you can see here, and you can agree, I guess, with me, is the mirroring of these two images is so perfect with each other. Therefore, we can control the switching reversibly in 100% with these lipids. So we have also our switch is working perfectly, 100%. It's a mechanosensitive channel. The next step, do we have tension? Do we have pressure in our system? So we have to, again, examine the ASO-PC monolayer in the water-air interface with the language project trap, starting either from the cis isomerization or from the trans isomerization. We go through cycles of 10 minutes each. The whole experiment was around 100 minutes, so there's a long time there. And we concluded that it's expected pressure of 13 millinewtons per meter. So the target is there, switchable lipids are there, pressure is also there. Now, how am I transferring an internal membrane protein to the system that I want to create later on? Traditionally, nanodisk assembly. Basically, we have our membrane protein in detergent. We introduce membrane scaffolds, the MSP, lipids in a secondary detergent using either biobeads or dialysis. We remove the detergent and we have the self-assembly of the nanodisc. All the nanodiscs that have been used in these experiments, they were with a percentage of ASO-PC, so they can mimic nicely or reach nicely what we want to see. Also, the same the same nanodiscs were used for the protein transfer in the lipid bilayer. Now let's put all of them together. Illuminating with 365 nanometers under the UV light, our lipids will choose to be in the C state. Therefore, the bilayer will have a big pressure, forcing our mechanosensitive channel to be closed, therefore no ion conductivity into the system. Illuminating with blue light, the lipids isomerize to the trans, the lipid bilayer is being relaxed, mechanosensitive channel senses these relaxations in the lateral pressure, then we have ion conductivity. The first experiment that we did, it was again with the FTIR different spectrum. Again, we'll not go into the details about this one, but the experiment, we started with nanodiscs that they contain the MSCL, and nanodiscs that they do not contain MSCL, with the same ASO-PC lipid concentration in both of the systems. We follow the different spectrum through this switching from trans to cis and from cis to trans. And we did the double spectra here. FTIR, it's a quite good analytical technique for many years now, because you can actually follow all protonation, deprotonation pathways, and you can also characterize structural changes that might be into the sample that you have. Therefore, by double difference spectra of these two samples here, we can see here at the gray region, at the amide 1 region, that there are some changes in the alpha helical structure. Although IR is quite good and specific analytical technique, yet is not a functional readout. We don't know, is actually this alpha helical structure changes there from the MSCL, or is it enough from the MSCL in order to be activated? And we can see what is happening into our molecules. Therefore, enter orbit mean. We use again the simple chips that we already know. We created the lipid bilayer, again with the ASO-PCs and the PHPCs in order to replicate the same conditions as the nanodisc. Illuminating with the UV light, there was no signal. Also at the negative controls, there was no signal there. Then illuminating with the blue light, we can see the spontaneous openness of the channel. That is actually okay. We have something inside there. To go further a bit more, zooming in into our sample and our recordings, these are the spontaneous opening of the channel with the blue light illumination. They're not very clear if these signals are actually the protein conductivity or if it's correct or not. So we had to analyze our data. But at this point, as I mentioned before, MSCL is quite extensively studied. From 1999, Sukharev and al. have identified the MSCL that is a non-binary channel. What the non-binary channel means is we don't have an open and a closed state. There is also subconducting states in the between. Each subconducting state has a different conductance. So we try to remove currents that they don't agree. So we have the baseline, the nominal current level, yet we have a small population with a mean amplitude of around 60 picoamperes, which agrees with a conductance of 0.8 nanosiemens and agrees with the first subconducting state of the MSCL. Now, again, to confirm that what we see is actually what we want to see, we proceed with introducing an again well-established mutation of the MSCL, the G22C. The G22C exchanges one of the glycines at position 22 with the cysteines. For visualization, again, this mutation is in the pore domain of the MSCL. By exchanging the glycine with the cysteine, we increase the hydrophobic character of the pore domain of the MSCL. Therefore, it stops being a mechanosensitive channel anymore, and you cannot control it via pressure. Yet, by adding MTZ, this trimethyl sulfur compound here, which reacts upon the cysteine, we create a trimethylamine end at the pointing inside the pore domain of the MSCL, increasing the hydrophilicity of the pore domain. Therefore, ions can pass through the channel. So to confirm our experiments that they actually work, that this is actually proving the first subconducting state, we did the same experiment, but this time with 100% DPHPC lipids at the lipid bilayer. We incubate our protein. There was no signal for a long time, as much millivolts that we applied. We added the MTZ compound inside, and after some time, we had the spontaneous opening, and the signal was recorded. We analyzed again the data with the same region here of around 17 picoamperes at the mean amplitude, agrees with the 0.8 nanosiemens conductance, but it is actually the subconducting state 1. Being very fast. And with our conclusions, there was photopharmacological approach to remotely activate mechanosensitive channels by light, opening a route to time resolved studies of mechanosensation by pulsed laser sources, and long-term goal is the understanding of the molecular mechanism of hearing and pain, hopefully in the future. And a big thank you all for your attention, physically and online, to my group, to my supervisor, Ramona Schlesinger, to Professor Joachim Hebele. Special thanks to Fuchsia and Ife that they did all the spectroscopic analysis. Konrad for the many calls that I've been calling him when I was trying to find out how to use this machine, and our funding, the FDA and the UNICEF. And thank you all again.
Thank you very much, Andreas, for these insights in your current research. Are there any questions from the audience?
Hi, thank you very much for your talk. Very impressive work. I have a short question regarding the photo switch of the lipid. When you switch it to the cis state, how long will it stay in the cis state before it switches back by itself? Is it switching back by itself at all?
It switches back with itself because it's heat with heat. So, I mean, if you leave it for an hour without any help with the UV light, it will go back to.
Well, okay, an hour.
It chooses always to go to the trans.
Oh, that's surprisingly long.
Yeah.
Are there any further questions?
Hi, Andreas. Fascinating talk. Thank you. Are there already any known blockers of MSCL, or are you going to be looking for specific blockers of MSCL?
I've read a bit in literature. I cannot recall them now because it was not our goal for so far. Yeah, I cannot say.
Right. Further questions? Okay, so let's thank Andreas again. Thank you. All right, so the last talk will be held by Tobias Enslin. He received his bachelor degree at the University of Reutlingen, and he then went to Freiburg, where he was one of the earliest adopters of the orbit devices. He was actually with Ionera, the company that produces these chips that invented this technology. and he received his master's degree at the University of Freiburg and did his PhD under the supervision of Professor Jan Behrens, who's here as well. And well, he was intensively studying bilayers and transport across bilayers on the orbit devices. And today he's going to give us a really nice overview about a new feature we introduced for the Orbit Mini. It's the coupling of electrical readout, which you would have under voltage control from an artificial bilayer, and optical readout. So it's a translucent chip. You can prepare these bilayers and investigate them under microscope while reading out an electrical signal. So Toby.
Yeah, thank you very much, Konrad, for the introduction. Well, hope it's working. Okay. Yeah, thank you very much for staying here for the very last talk of the day. I know it's been tough 2 days, a lot of different talk, very nice. My talk is about membranes, or it's called membranes, and especially planar lipid bilayers under investigation. So about myself, I'm, as Konrad said, currently located at the University of Freiburg at the Institute of Physiology, which is in the very southwest of Germany. And yeah, my main research are actually nanopores. And there I'm doing a lot of basic science, but I also have a strong interest into new technologies, which is exactly the point I'm talking today about. So the key question is, how can we investigate biomolecules? So we could do this either optically by the use of a microscope, which brings us very satisfying and pleasing nice pictures. So it's always fun to look at those, but we can also measure distances very easily there. But microscopy has a lot of disadvantages. So you need a very intense sample preparation. It's mostly a discontinuous method, and it's not really live because most of the time tissue is dead when you look at it, right? So the other methods, which is available, is electrophysiology. So this is exactly the vice versa method. So you can do live measurements, very easy manipulation of your specimen, and you can do parallel experiments, very easy, but you don't see what is really happening inside your sample, right? So data analysis, when you get at current trace, like it's depicted on the bottom, can get very tricky because all these current traces can get rather complicated in there. So there's a very obvious elephant in the room. Can we do both at the same time in parallel? And in such a way that we get rid of all the cons and use the pros of both techniques. And there's a simple answer to it, which is already the conclusion of my talk. And this is, yes, we can. So, yeah, and this is exactly the Micro-Opto system. So all the rest of the slides I would like to spend to introduce you the system itself and to show you a few case studies we did with it. So the Micro-Opto is a extension of the Orbit Mini. It's a chip which holds 4 electrodes, ring-shaped electrodes, and those electrodes are located at the bottom of the cavities in this array. So you can span manually a bilayer across the cavity, which you can investigate and optically by placing the entire setup on a regular microscope. So here, this would be the Orbit Mini. So very small device, which makes it very easy and applicable to all the microscopes you can think of. And yeah, you can use the electrodes to stimulate then the bilayers or whatever you are interested in. So when it comes to nanopore science, first of all, you need to know about the lipids, because there is no nanopore experiment without the lipids, right? So this is exactly where I started the research here with this new device. And wherever I go and to whomever I talk about these systems, there's always one question coming directly. How long does it take to form a bilayer? And for this reason, I think the upper left graph is one of the most important graphics in graphs in the entire talk, because it shows you it takes seconds. And whenever I tell people it takes seconds, they start laughing, but this is really what it is, right? So you have an open electrode on the very left side, then you paint. We use a manual painting method here with an air bubble and lipid, and then you get already a sealing, and then it's a self-thinning of the bilayer within a few seconds, like 5 seconds or something, then you are ready to go. And that's really nice. And using the electrodes there, actually, we can characterize the lipids or the bilayers we are forming. So we can measure its capacity, and usually we find values in the range of a specific capacity of 0.5 microfarad per square centimeter, which is comparably with what you find for other painting methods in literature, but it's a bit lower than when you would do Montan-Müller, and this is simply due to a different content of the solvent in there. When you use, oh no, I hope it's working. When you use the electrostimulation, you could, for instance, do an experiment. Not starting. Oh, that's a pity. It was. Oh, no, okay. Yeah, so here you can see the beading. That's actually the black part is the real bilayer part. The white is a thicker part. It's not a full bilayer. Actually, I would not use such a bilayer for an experiment, but it's a very nice one to show people who are not used to this electrorestriction experiment what it is and what it means. So it's simply a triangular waveform of voltage, which is applied, and then the bilayer is answering to that. And because when you use a good bilayer, as it should switch now, it's not. Stop. Yeah, so if you would use a good bilayer, there's on these chips, you almost don't see any annulus. So it's full bilayer there, and then electrorestriction is very hard to see. So on the lower left side, you see a color stained bilayer. So it's post-processed. It was also black and white in order to make the bilayer visible, because otherwise its contrast is very, very low. But sapping of it and measuring the fluorescence activity there, intensity, we can clearly see a shift when we lose the bilayer on purpose. And it was very nice in this experiment by the use of a very fast camera. We were able to track the movement of the lipid directly after the zap. And we saw that within 50 milliseconds, which is a fairly long duration actually, the lipid retracts to the rim of the cavity in a dropwise manner. So that's what you see here in the lower central picture actually. So the next step was, we were very interested, thank you very much. We were very interested in how the membranes are looking. So we went to a confocal microscope, and this was very surprising to us. I was emphasizing the term planar lipid bilayer in the beginning. So everybody thinks all those membranes are planar, so they are completely flat. But this was the result of the first scan I did. So this is a stitch up of 3 individual scans, so set scans. And we saw there's a curvature in the bilayer, and this is really a significant curvature. So when you measure it, it's actually curved by 8 micrometers, which is half, or almost half of the remaining of the cavity size. So when we investigated further membranes, we found actually all sorts of membranes. So there were convex, so inward, outward carved membranes. There were perfectly planar, flat membranes, as well as bubble-like membranes on the lower right side. So the take-home message here is, when you do electrophysiology experiments with lipid bilayers and you are charging their quality simply by a capacitive measurement, this gives you no information about the shape of the membrane, right? And this, easy to imagine, will hardly affect your reconstitution of the nanopores or whatever you would like to get in there, right? So this was actually a very nice thing. And what we found out, that the shape of the membrane, so whether it's inward or outward curved, really depends on the way you move the bubble across the openings. So when you do it very fast and you lift the bubble up, then you get more readily an outward shape. When you do it with a lot of pressure, it's inward shape. And when you do it very slow and smooth, you get really planar bilayers. So it's good to take time on this, on this method here. Yeah, then we started to investigate channels on the membranes. So we modified the setup that we are not using anymore a labeled lipid here, but a labeled channel, which we would like to investigate here with this Meca-Optos tip. And in our case, what we did use here was ceratotoxin A, which you can find in the female Mediterranean fruit fly. This is a short peptide, and it has an antibacterial activity. And there's a working hypothesis that it undergoes an allomaticin-like insertion mechanism. So the C-terminal helix is supposed or hypothesized to kink into the lipid bilayer in such a way that you find transient pores, like you see here on the lower right panel. And what we did here, we extended the sequence by a C-terminal cysteine and labeled it with the ATO dye. The first question, whenever you do such an experiment, is for sure, is the dye hurting? Is it affecting the results of my experiments? And to answer this, we went on our high resolution setups with the Meca 16. chips where we have a very nice temporal resolution. And we compared native CTXA with labeled CTXA. And what you see here on the left side, so the lower panel is just a zoom in of the upper panel. We found roughly the same current levels, which was good. But if you look carefully, the labeled one has a high noise or high frequency flickering noise on there. And we attributed this actually to transient blocks of the channel by the flexible terminal end group of the die itself. All right. And another finding, which I will come back later again, is that membranes tend to break when there is a die on the C terminus after pore formation. So you see a short time a transient pore, and then the entire membrane ruptures by itself. So we said, okay, it affects somehow our results, but we can live with that. So that's fine. So we went back to our wide field fluorescence microscope, and we tried a very low concentration, actually 5 nanomolar of the toxin of the labeled. And we found also with the orbit nice transient current openings there. But what you can see on the two images here at the bottom, that the fluor, and also on the histograms here, that the intensity is very low. So when we zap the membrane, we can detect a shift in fluorescence intensity, but this is a very minor shift compared to what I've showed you before when we had the labeled lipids, right? So here, this is mainly due to the low concentration for sure of the dye. And we have a fairly high background here, actually, which is due to the glass. So that's maybe a next step here to thin out the glass of the Mecha Opto in order to improve the optical quality. But nevertheless, we went further and did some confocal microscopy here and did lifetime measurements on membranes of with a lipid dye, as well as on membranes with the peptide on top of them. So the top membrane here, this is really the stained membrane. And here what you see enlightened, this is just the lipid on top of the unstained membrane. So we see it's really accumulating there. And when we were measuring lifetimes, we found values which were for the peptide exactly what literature tells us, but also for the fluorescently labeled lipid, it was quite good and in the same range what you would expect. And what was very important to us during these confocal measurements, we were able with the orbit system to detect already the channel activity there. So we could say what we measure here on the membrane is really our fluorescently labeled toxin. So that was very nice results here. So we had an interest in putting the experiment further. So we started to equip our light path with a beam splitter, and we used 2 polarizers there in order to get a single photon counting experiment with polarized light, because the working hypothesis actually was when you have one of these helices labeled and it kinks into the membrane, we expected a change in one of the intensities down, so a decrease, and the other detector should give us an increase, right? However, we did the experiments, as you see in the middle, we did not find any of these things. So, and this was simply because, at least that's what I think, the concentration of the dye was still too high to get a single photon counting. That we really see a significant change here. But what we found, and this is really obvious, is an anisotropy. So when you look both of the spats, they showed an anisotropy as long as there's a bilayer. Whereas when we measure later on in the solution, so the free peptide, which is there's the anisotropy is completely gone. And what we think is the reason for this actually is that the dye is maybe interacting with the head groups of the lipid. And this might also be the reason why it's rupturing the membrane when it goes back to the cis side, because it's really attached there. So that was, in the end, a positive outcome. So we learned a lot from this experiment, despite we could not see what we initially wanted to see. Yeah, then I have 2 other case studies, also very fast. So the next thing is about real nanopores. So this is the OMPF channel. Maybe some of you are familiar with this. It's the outer membrane porin of E. coli. Project partner of us labeled this channel, and I reconstituted it on the Mecha Opto. And what you see here in the 3 yellow circles are 3 channels. And I hope this video starts. Yes, nice. So what we saw here was actually very nice. So we could measure the diffusion coefficient of these channels actually. And especially with the one here in the lower left circles, we found a diffusion coefficient of 1.2 square micrometer per second, which is also a value which is known in literature and is supposed to be a channel which is inside. Whereas the two other ones, you can see directly, this guy already has left a circle, but those are still sitting in there. They're diffusing very slowly. So optically, you would say, okay, I got 3 channels there. 1 is fast, 2 are slow, fair enough. But we also got use of our electrodes here, and the electrodes told us there's just a single functional channel in the membrane. So this is what we found on the electrophysiology. So this means, in essence, that the 2 slow guys here, they are just adsorbed on top of the membrane, but never inserted inside. Whereas the fast one, this is really in the membrane, and this is conductive there. So when we added an heptaarginine peptide, we also could translocate this heptaarginine through the on-BAF channel and reproduce more or less exactly the data the literature shows for such an experiment without the labels there. So it was really a nice experiment, and the optical quality to get single channels here was really fascinating for me. And a last slide here was another study. We got a project partner who labeled a hemolysin channel, and he wanted to do actually calcium imaging with it. But then he came to us and said, the intensity of the calcium is very low, and he has no clue why, and what he would like to try out if the channel is still functional. And we said, sure. So we threw the sample on our Mecha Opto chip. Here it's not a movie, it's just a picture, so it's a bit hard to see. There's a single bright dot in there, which is the labeled hemolysin. And when we added PEG to our polyethylene glycol, which is known to translocate through hemolysin for years, we found actually something very weird. So these upper 2 conduction modes, actually from this experiment, the top graph is what I measured there, and the second graph here was a second experiment in order to reproduce it. So we found a very strong gating, which an unlabeled hemolysin on the same system under the same conditions is not showing. So the idea here is they use the NHS dye, which is responsive for lysine residues. And when you look at the structure of hemolysin, you have lysine residues inside the channel, especially here at the stem, at the constriction, as well as at the outlet down there. So what we think here, and that's also something I need to mention, hemolysin is a heptamer. So what I suppose happens there is that you label really the lysins inside the channel. And when you see a very strong gating channel like the upper one, you might have simply more fluorophores inside, which interact with the PEG and simply do stronger size exclusion. Whereas when you have an intermediate gating, maybe only 1 fluorophore is in there. So with this, we could say that you also, or the take-home message here is when you label your channel for a functional assay, you really need to make sure that you find a unique position for your fluorescent label, because otherwise the dye will hardly affect your channel and its function. Yeah, with this, I'm at the end of my talk. My conclusion you already heard in the first slide, I think. And I would like to thank my supervisor, Jan Behrens, as well as all my collaborators for sure the funding. And yeah, I'm open for any question. Thank you very much.
So much, Toby, for this beautiful talk about the optochips on the orbit mini. Are there any questions from the audience? So I would have a question about the membranes that bulge in different directions. Did you ever look at this with like osmotic pressure, like with 10-fold higher buffer concentration on one side?
No, we did not do that yet. We also worked in isomolaric conditions.
I see, because I mean, I could imagine that for some proteins, it might be beneficial, it might be favorable to have, I don't know, a certain bulge, maybe outwards to get better insertion or something like that.
Absolutely. I mean, again, that's kind of the procedure EBS is doing with their system, right? Where they have a tiny pipette and they apply a pressure in order to create a bubble to capture a protein, and then you reduce the bubble and get it back. Then you have a very tiny patch of membrane there. So yeah, actually this would be very cool to do something like this on an orbit system.
Yeah, okay, perfect. Thank you so much. So if there are no further questions from the audience, we should talk about these proof of concept studies. I really love those. I want to show them to a broader audience as soon as I can. So then I would like to thank Tommy again. And that was a great journey.
Okay, we are a little ahead of schedule, amazingly, but nevertheless, that does conclude our Nanion user meeting for this year. I'm sure you'll agree we have been treated to some outstanding talks over the last 2 days. We've heard everything from fluoride-free recordings on the SyncroPatch, lysosomal channels. We've heard about toxins, stem cells, nanopores on the orbit systems, and everything in between. So I'd like to thank all of the speakers for joining us from around the world, for making the journey here to Munich, or for joining us online. So thank you very much to our speakers. Thanks all, of course, to our chairpersons, to Niels, Marc, Konrad, Elena, and Cecilia for moderating the sessions. Thanks mostly also to our guests who've come to join us on site and online. It's for you that we organize this meeting, and it's because of you that it's always so much fun. So thank you so much for coming, and to our collaborators for helping to promote the meeting, and for being with us here over the last 2 days. Of course, this meeting doesn't organize itself. We've had a whole team behind making sure that the organization works as well as we can hope. In particular, I'd like to thank Eleanor and Sonja. They've done a load of work, the rest of the marketing team, also the IT department, in particular, Christian Grad. Christian always answers me with a smile on his face when I ask stupid questions or I ask him to do something for me. So thank you so much to him for helping me out with all the little technical glitches for getting the talks loaded up onto the laptops. So big thanks to him. Thanks to Ulrich for taking care and making sure we're all from the health and safety aspect, and for organizing our students and colleagues who've been running around in the background. In particular, I've seen Stancy and Martin running around, making us coffee, making sure we have coffee and tea and biscuits to keep us going. So thank you very much for rearranging the tables and the chairs, but also for clearing up after our very nice dinner that we had last night, which of course brings me to a very special thank you to Patrick Mum. Patrick is not only product manager for the Porter Patch, he's also an incredible cook, and he did us proud yesterday. I'm sure everyone who was here last night who was treated to that meal will agree. It was really amazing. So thank you so much to Patrick for doing such a great job. So thanks again to everyone who joined us online. Thanks to everyone who's come on site despite the uncertainty around illness and the dreaded COVID. Of course, we did have some people, oh no, my nose is running, but I don't have COVID, I did test. We did have some people, of course, had to cancel at the last minute, including one of our speakers, unfortunately, who did have COVID, so he couldn't join us, but we wish him all the best. We still have some product demonstrations going on downstairs for the people who've joined us on site, in particular, a SyncroPatch demo of the Piezo 1 channel. So please don't miss that. That's going to be great. Stick around a bit, drink some beer with us. I know Patrick is already organizing the next little snack for us, for those of us joining us on site. So please do stick around. You'll have noticed the beer mugs. Make sure you take one of those with you. Take one for your colleague who maybe couldn't come with you, and a t-shirt if you can still find one in your size. I think all the honey is gone, but I hope you managed to get a jar or two. There is a couple in there. Oh, there's more in there. Okay, so please make sure you take a jar of honey with you. This is a special edition honey. It's made by the bees from my home. And it was harvested by myself, my husband, and my kids. So please do take one of those with you. just remains me to say thanks again for joining us. Have a good journey home. For those who are joining us online, I will be stopping the broadcast now, but thanks for joining us. Goodbye for now and see you next year.